to make it possible for me to make a remote presentation about our current research project. To tell the truth, I'm double vaccinated, but unfortunately with Sputnik, it makes the international traveling rather impossible for me. So that's why I'm not with you, even if I wish to be with you in Prague now. So what I'm speaking about today is the first insights, or the first results we get on the genetic structure of the invasive oak lace bug in Europe. I would like to thank to all my co-authors from Boku, Vienna, also from the Hungarian Forest Research Institute, which is now a part of the University of Sopron, and of course to all of my colleagues at the Institute. As a first slide, some nice photos from Yuri Choka about the various life stages of these insects, well, the eggs, the larvae, the nymphs, and the adults. So if you are in the invaded area already, so you are definitely familiar with this species. Interestingly, the species has its native origin in North America, where it's a very low importance, both in economic and ecological sense. So what we know about this species now, the first introduction happened in 2000 in the northern part of Italy. However, there was more or less definitely independent introduction to Turkey in 2002. It's interesting that the next invaded area was in 2002 in Switzerland, and even in certain parts of Iran in 2005. But in Central Europe, the central part of Europe, or the southeastern part of Europe, it's rather a kind of mixture of various dates of the first funds. So like in the next date for first funds in various countries was 2013 for Hungary, Croatia, and even Serbia. So it's a rather interesting history of an invasion in Europe. So, however, it's only 21 years ago. There are already several publications already published, so we know already a few things about this species. So first of all, of course, the date of introduction. The publication was made by Iris Bernardielli and her colleague. And of course, we know already how wide the host plant spectrum of this species is. So most of the European oaks and many other plant species are suitable host plants for the oak lace bug. Of course, the effect of this species on the photosynthetic activity of the plant is also considerable. So it was investigated by Natasha Nikolic and her colleagues in 2 years ago. In Europe, oaks are one of the major tree species having a large distribution area across various ecosystems. So approximately 30 million hectares of suitable oak species are here in Europe now. So which makes the species a very considerable invasive species for various parts of Europe. Colleagues from the Hungarian Forest Research Institute investigated the overwintering mortality of the species, and this survey gave the final result approximately 30% winter mortality for the species in Hungary. And of course, there were several publications also on the possible natural enemies, or even the possible use of these natural enemies as a biological control of the species, like here by Marta Kovac. Even there were already certain trials to use some chemical control against this Corytuca arquata across the oak forest. And of course, there were several other publications already available. Just one slide about the damage the species can cause. You can clearly identify the oak trees here with this yellowish, grayish, yellowish colors. All the other tree species are green at the end of the summer, so in August. So that's a typical damage symptom for the species. And what we do not know about this species, the origin, I mean, within North America, where it is coming from, the pathway, how this species came to Europe. And of course, the number of the introductions, 1, 2, or maybe even more. And of course, we don't know anything about the genetic patterns, neither in the invaded area nor in the native area. And of course, we need to know the future as well, at least regarding this, the damage and the role of the species in Europe. So what we did, but before I start with this, just a few information again. So first introduction 2000 in Italy, second introduction 2002 in Turkey. There are more or less clear directions of the range expansion in both parts of Europe, but there is really there are many questions about the spread of the species across Central Europe. It's not really easy to follow up the spread of the species in this area. So what we did, at the moment we have samples from 9 countries, so we collected individuals from 9 countries, 18 geographic locations, altogether 37 individuals. We sequenced the CU1 gene of the mitochondrial DNA and analyzed the approximately 550 base pair long fragment of this gene. And what we got, what are our results? We could identify altogether 10 haplotypes of this species. What I would like to highlight that in the Central European part, you can find only 3 haplotypes. So these 3 haplotypes are dominant across all these populations. If you look at Turkey, the genetic diversity is much higher than in any other part, and it's a rather interesting situation in the Balkan Peninsula. So this is about the haplotype diversity. If you look at the dendrogram, where you can see 2 distinct clades, so distinct groups. The one group includes all the Central European haplotypes, plus the haplotype number 9 from Turkey. So it's a rather, on one hand, interesting, but on the other hand, also a little bit confusing result. And if you go to the haplotype network, what you can see on this picture that the most common haplotypes, which is green on the map, is common almost everywhere. And the haplotypes from Turkey, so the highest diversity of the species is in Turkey at the moment. If we are in a lucky situation in Europe that we have another invasive Corythuca species, so we can compare these 2 species regarding the genetic diversity. So if we compare the oak lacebark and the sycamore lacebark, then we will find some similarities, but definitely also some differences. The first one regarding the haplotype diversity is we got 10 haplotypes across Europe, across the current distribution area of oak lacebark in Europe. Now we found with the same fragment of the CU1 gene only 2 haplotypes. So there is some difference between the 2 species. If you look at the 2 dendrograms, while on the oak lacebark you can see some structuring, on the sycamore lacebark there is no structure at all, at least regarding all haplotypes we found in sycamore lacebark. If you look at the haplotype network, so I already explained the haplotype network for the oak lacebark, so which is represented by a very common haplotype across all regions and a very high genetic diversity in Turkey. Unfortunately, these 2 graphs are not so easy to compare, because in the case of the sycamore lacebark, we had already populations from North America and various other parts of the globe. So what you can see here that the highest haplotype diversity was found in North America in the native area of the species, while there are some common haplotypes across the invaded areas. And yeah, what are our conclusions here? So we found altogether 10 haplotypes for the oak lacebark across the current invaded area in Europe. We found a rather high haplotype diversity in Turkey and a moderate one in Central Europe. And I think this is one of our major points, that the supposed colonization routes of the species and the genetic patterns we found does not really completely overlap in the current invaded area of Europe. And it should be also mentioned that we found a much higher genetic variation for the oak lacebark in Europe than what we found for the sycamore lacebark a few years ago. So we definitely need some inputs here. That's why the title was called The First Insight. So definitely we need to analyze some core population from the native area, also from North America. We also want to add some additional population from Europe, from all parts of the invaded area. And of course, we are planning to use some additional methods, some next generation sequencing method to have a much more robust data set. But what I would like to highlight here, is just this one, that we need we have to concentrate on the core population from the native area. And here I'm really looking forward to make some cooperation with colleagues from North America. And that's it. I hope I could finish it in 10 minutes, and I'm open for questions.
All right, thank you. We have time for a handful of questions, if anyone has any.
Hello, Frank. This is Jean-Claude Grégoire. This is maybe a naive question, but among the haplotypes that you showed, there is one in Bulgaria, which doesn't appear anywhere else. Doesn't it suggest a third introduction?
Hello, Jean-Claude. That might be. That's why I put it on my slides. So we know about two definite introductions, one in northern Italy in 2000 and one other in Turkey in 2002. But at the moment, there are no real data, no exact data about the possible introduction in the Balkans, somewhere around Bulgaria. That's true. We have to check this opportunity as well. Thank you. Thank you for the note.
We have time for one more question. All right. Thank you, Frank.
Thank you very much.
Up next, we have Nicholas Maries from New Zealand, speaking on New Zealand's forest biosecurity surveillance system.
Hello. Just checking, can you hear me okay?
Yes, we can hear you and we can see your screen too.
I had a few troubles with the connection just before, so I probably just stopped my video to be on the safe side and hope for the best. So this presentation is on the development of the forest biosecurity surveillance system in New Zealand. We have a primary goal that is to detect invaders, insect pests, and pathogens as early as possible after they have established, and that's to provide more opportunity for a rapid response, including to attempt eradication when that can be possible. What I'm going to present today is our modeling works and also our risk maps that are used to inform the allocation of surveillance. And no big surprise, the system implements surveillance at obvious points of introduction, like for instance, the port of Tauranga that is pictured here. But it also shows that there are many other plausible places of introductions across the country, and we now do some biosecurity surveillance in these areas, which are areas where commodities carrying insects and diseases are transported. But moving to the next slide, and first a very quick overview of New Zealand forests. These are in fact quite varied. You have native forests pictured here on the left, still covering about a quarter of the country's territory. And by the way, on the bottom left, you can see a forest dominated by a giant kauri tree, which is quite iconic from the north. And then in stark contrast, New Zealand also has large areas covered by commercial plantations of exotic trees. These are completely different landscapes. This accounts for about 2 million hectares, or about 7% of the territory. One species there clearly dominates, and that is Pinus radiata. There are a few other cultivated species, including Douglas fir, cypresses, and eucalypts. Here is another picture of a typical Pinus radiata monoculture. This one is called Kangaroo Forest. That's located in the central North Island, and apparently one of the largest softwood plantations in the world. This one is characterized by very short rotation, average harvest age being only 28 years. This one, Kangaroo, like other plantation forests in New Zealand, is relatively free from pests and pathogens, but there are some. I present a couple here. First illustrated on the left is the wood wasp, Syrex noctilio, that has been there for a while. There are also a few European bark beetles, Cerambycid and aphid species, notably, and perhaps a bit more worryingly introduced pathogens. One example here in the middle is Dotistroma needle blight, but in recent decades, foresters also had a few bad surprises, especially when they found pathogens that weren't on any list of unwanted organisms. And some examples there are the causal agent of the Nectria flute canker, and a little bit more recently, Phytophthora causing red needle cast. Looking at pests and pathogens that continue to establish in New Zealand forests, some are pictured here. And you see there are quite a few with probably no obvious sign of a slowdown despite all the restrictions and the phytosanitary precautions that have been implemented. Invaders can clearly still arrive by a number of pathways, most of these associated with human activities, primarily internationally traded items and international travelers. We also know from international and New Zealand experience that these pathways tell us something about the most likely locations for future introductions, and these are the areas we should probably have a good look at. As an example, for New Zealand, we have recently conducted an analysis of 37 introduced forest species, and that interestingly shows a majority of the insects and more than half of the pathogens have been first detected in urban areas, and that is largely pointing to Auckland. Now, when we look at forest pest surveillance from a more historical perspective, historically, surveillance has been first conducted in forest areas. So there was more monitoring the abundance of well-established forest pests. That is, these main production forests where historical sort of surveillance have been conducted are pointed here in green on the map. But in recent decades, biosecurity surveillance activities outside of the forest have gained more momentum. Quite notably, a dedicated surveillance program for pests of trees and shrubs was launched in the 90s, with surveillance this time based on estimated entry risk levels. This is the so-called high-risk site surveillance system. It is illustrated by the red dots on the map, and you can also see it on the picture below the detail of the surveillance locations in the Auckland region. Quite clearly, this is focusing on ports and urban areas. Then from the 90s, and also reinforced over the last 30 years, there's an increasing recognition that most resources should be targeted towards surveillance at high-risk locations. Especially with that sort of objective like early detection for elimination as opposed to more traditional forest health monitoring. So more recently, in 2015, the government and the industry signed an agreement to capitalize on resources and clearly pursuing the same biosecurity objectives of prioritizing early response and elimination. One of the agreed objectives is to develop pre-agreed contingency plans, as well as quickly actionable response activities, all that should a new organism be introduced. Another goal was to further develop the forest biosecurity surveillance system to improve early detection of new forest pests, and especially these ones affecting commercial forestry. The system works alongside other national biosecurity surveillance initiatives, but I can highlight here 3 of its key characteristics. First, surveillance is to be risk-based and considering available knowledge of pathways of entry. So that's why we have the port of Tauranga illustrated here again. You see containers, and clearly focusing on most plausible entry location should favor detection of small established population that could possibly be eradicated. Second, this redesigned forest biosecurity surveillance targets both forest insects and tree pathogens, not a particular species. So the surveys are therefore based on transects and visual inspections of trees for damage symptoms, and these eventually followed by sample collections and the identification of the causal organisms. Lastly, third, this surveillance is focused on the protection of plantation tree species. So I mentioned Pinus radiata, exotic conifer eucalyptus, and that is a little bit different to the other national surveillance network. So what was the plan? I hope you can still bear with me. Here is an outline of the general implementation with the boxes with this red outline highlighting the 3 main activities. First one being risk mapping. So that's about the development of a model that could be based on entry pathways and then predict pest arrivals, and also take into account habitat suitability for establishment. So that model is called SPARE, and it generates national pest establishment probability maps. The second activity is a surveillance allocation. That is another model integrating the risk maps for different species or groups of species, but also some information about the cost and the efficacy of different survey methods. And that is generating an optimal surveillance allocation across the different parts of New Zealand. And then the final activity, which is the one that has been going for 3 or 4 years now, is operational surveillance. So that's working on the annual surveillance plans based on the model results and the optimal surveillance allocations suggested by the model. And these steps also incorporates operational feedback from the operator. But the risk mapping, that one's called SPARE, that's for Spatial Pest Entry Analysis Runner. It has been developed by our colleagues from Bayesian Intelligence, a team of ecological modeler experts in Melbourne. And what SPARE is not tracking tracking the pests entries directly. Instead, and that's shown here, it tracks the commodities to which the pests could be associated. You have an example here with the container pathway that could be a mode of entry for, in this example, gypsy moss egg batches. SPARE incorporates information on import volumes by the countries of origins, information like infestation rates by moss egg batches, and then it's tracking the movement of these containers as they transit through the entry point, which are the ports. Here it's the Auckland port, and then through pathway intermediates and endpoints. And in New Zealand, for containers, you may have transitional facilities, cleaning depots, and of course, all populated areas across the country. The model also uses pest biological information as input. I have already mentioned infestation rates, but there are several others, such as mortality, egg development rates, and eventually that models predict escapes or entries at each pathway point. I'm going to show you an example. First, I want to mention here that the tool doesn't only work for containers and gypsy moss. It can be actioned for across multiple combinations of pathways and representative or poster pests. That is really to capture the range of possible entry locations across the country. You can see here as well that pathways include multiple items a pest can be attached to or transported on. We have vehicles, containers, wood packaging material, even travelers, overseas visitors or returning residents. And then there is also the list of our poster insects and pathogens, the ones that we have used so far. How does the model work? It's based on Bayesian networks. So that's where we needed the help from this group, Bayesian Intelligence. It has several advantages, allowing, for instance, the quantification of both these pathways, items, and also the pests, and also to account for uncertainty associated with some of these input parameters. Overall, we can say we have relatively good data for our pathways factors, trade volumes, passengers flows, pathway points, but the pest biological factors are extremely uncertain, and many that we have incorporated are estimates based on experts' elicitation. A little bit more visual here is a representation of the model again with the container pathway example. This is how the model tracks the transport of these containers and the movement of these containers through the different pathway points. And these are the blue boxes. And at each step, it estimates a probability distribution across the number of escapes or hatching of gypsy moss larvae. And an example is provided on the right with this this area circled by the dotted line, where we can see a probability distribution around the estimated number of escapes of gypsy moss at a particular pathway point. There has been a software developed for running the model. I'm not going to detail on that, but this is how it looks like. And probably more interestingly, how is the output? I have shown you where the potential entry locations are, but the output after the model is run typically looks like this. We have entry maps, which are expected number of escapes of, let's say, gypsy moss in each one of a large number of territorial units in New Zealand. So this is the example for Auckland, basically generating a propagal pressure map. Going to be a little bit shorter on the two following steps. These two being surveillance allocation and operational implementation.
Hi, Nicholas. Can you
Yes.
hear me? You have about a minute or so left.
Okay, now I'm going to be short on this one. The surveillance allocation process, it's basically an optimization model. So that one's primarily based on the establishment risk maps and on some information on the cost and efficacy of different surveillance types. I can skip that one and show you my two last slides. Implementation first. So this is showing the green areas that are the areas where surveillance is implemented, and that table is showing the number of operational area units that are surveyed and the number of surveys of the different types. Then an example here for Auckland. So that's showing some variability across a city landscape. You can see the dark green areas are where there is more surveillance done. And on the more practical side, you can see one of these typical area units and the exact locations of these transects that are implemented. So thank you.
All right. To keep us on time, I'm afraid we don't have any time for questions, so we'll have to go right into our next talk by Etsuko Shotakagaya on current status of 3 invasive cerambycid pests in Japan. Are you there, Etsuko?
Wait a moment. I'm trying to share the.
Okay.
Sorry. I'm trying to share my PowerPoint but failed. Wait a moment. No, I don't know why. If it is possible, please proceed the next presentation. I cannot find my PowerPoint presentation in the Windows. Test was successful. Sorry, I failed now.
Okay. Well, maybe we could. Sandy, should we go to the next presentation, or do you want to take a pause? Rebecca, are you there?
Yep, I'm here. Shall I attempt to share my screen?
Yeah, Rebecca, if you're able to present now, that would be great.
Okay.
And Rebecca will be presenting on international insect interceptions.
All right. Is that in presenter mode now?
Yes, we can see it.
Cool, great. Okay, kia ora everyone. I'm Rebecca Turner. I'm from down in New Zealand at Scion Forestry Research, and I'm a biosecurity scientist. So today I'm going to talk to you about insect border interceptions to give you an overview of the data, what's in it, and some of the things you should be aware of if you're looking to use it yourself. So I'm sorry to the pathogen and plants people. This is just insects. And we published on this data set recently in the paper you can see here. So this is the research group that I've been working with on this international data set. Across the top there are my 3 colleagues from New Zealand, across at the University of Canterbury, and the rest of the people are international researchers. You might be able to spot a couple of people there in the room with you. There's Eki Brockerhoff and Sandy Liebold. So insect border interceptions. So by these, I mean insects that are detected as part of inspections of generally air and sea cargo, including the containers themselves and the vessels. There's also quite a lot of international passenger baggage, and a small amount of international mail. So the 6 main groups of insects that are being intercepted at borders are the ones down the bottom here. So we've got Coleoptera like beetles. Well, they are beetles. Diptera like the fruit flies, Hemiptera like the brown marmorated stink bug, which New Zealand is particularly interested in. Hymenoptera like the ants, Lepidoptera, so moths, and Thysanoptera like thrips, which are commonly intercepted with cut flowers. So in an ideal world, there's lots of questions that we'd like to ask using border interception data to do with biosecurity risk assessment. But unfortunately, in general, border interception data was not collected with scientific analysis in mind. For example, one of the prime reasons that it's collected is part of compliance testing of cargo that's just arrived to see if it is meeting its phytosanitary requirements. So today, I just want to give you a sense of the data, what's in it, and some of the limitations that you should be aware of if you're looking to use this data and when you're interpreting it. So these are the countries or the regions that we got our interception data from. And the data sets don't span exactly the same time frame, but they roughly cover the last 2 decades. So I'd like to acknowledge all the different government agencies that have made their data available in some form or another. Some of these data sets are only available under data agreements. And there's a few reasons why they do that. Partially it's to protect importer privacy, but also they're cautious about the potential for misinterpretation using this data. So for example, these are border interceptions. It doesn't mean that that insect has established in the country that's doing the reporting. And equally, if there's a interception record of a species being recorded on cargo from source country A, it doesn't mean that that species is necessarily established in source country A, because there is potential for contamination of that cargo on the journey. to the country doing the inspection. So those are just a couple of things to look out for. So what's in the data set itself? So in the combined international data set, there are just under 2 million different interception events, and just under 1 million of these identified down to species level. So the remainder identified generally to family or genus level. And one of the reasons why they don't always get identified down to species level is that the diagnosticians don't have a lot of time. So for example, sometimes in New Zealand, the diagnosticians might only have 8 hours to make the identification, and then they have to move on to the next sample because the importer has already decided to fumigate the cargo. But despite these limitations, over 8,000 different insect species have been identified in the international data set. And it's a very right-skewed data set. So 2% of the most commonly intercepted species do account for 81% of all the interceptions. And then there's thousands of species that will only intercept once in the entire data set. So the 3 most commonly intercepted species are these ones here. So we've got a mealybug and some thrips, and that is common of the data set generally. So there's a lot of Hemiptera and Thysanoptera being intercepted. So that's partially what you're seeing here. So here we're looking at the number of interceptions in an insect order relative to the number of species intercepted in that insect order. And the blue line there is a line of proportion. And you can see that the Thysanoptera and the Hemiptera orders are overrepresented in terms of the number of interceptions relative to the number of species being intercepted. And then if you look across at Coleoptera, so our order that contains lots of our forestry species, you can see it's just the most species-rich order, and we would expect it to be very species-rich given it's a large order, but it's not overly represented in terms of the number of interceptions in that insect order. So apart from some differences in the insect orders, there's also differences between the countries, and often this is due to operational differences, policy differences, and different priorities. So what you're looking at here is a mosaic plot where the size of the box represents the number of different insect species intercepted in that insect order in that country. So straight away you can see that the USA was the largest part of our data set, and it's overrepresented in Hemiptera, so that's the blue box there. You should also note that the USA had a larger proportion of passenger baggage interceptions compared to any of our other countries. And then looking across the top there at our Coleoptera order again, you can see that unfortunately for me in New Zealand, we actually have quite a low proportion of Coleoptera as part of our data set. But part of the reason for that is that we have a very general biosecurity inspection system. So we have a relatively large number of species being intercepted, given that New Zealand's a small country. So now if we go on a search for forestry insects, well, if you look in the Coleoptera order, the top most frequently intercepted species, so these are species intercepted in the thousands and not forestry insects. So the top 3 there are storage pests. And then a little bit further down, you get the mango seed weevil. And I'm just mentioning that here because it's an example of one of the biases that you find in these data sets. So in our full data base, 99% of the interceptions of mango seed weevil are actually from the US part of the data set. And within the US data set itself, 5% of their interceptions are on mangoes. And so I've been told that border inspectors there, when they've got a quota that they need to meet, they know that they can find insects on mangoes. So that's just one of those biases you have to watch out for. And then a little bit further down still, being intercepted in the thousands, you've got species in the Bostracoidea family, or superfamily rather. And these species in this family are actually overrepresented among global insect establishments overall, relative to the number of global species. And so that's coming up here again in the interception data. There's lots of interceptions of species in this superfamily. Okay, and then going down to the next level, so these are now species intercepted in the hundreds. Now you start to see some of the forestry-related pests or species. And a lot of the ones at the top will be the cosmopolitan species, so forestry species that are already established in quite a few places around the world. So that's reassuring that that's what's happening. And then lastly, I just want to encourage you that if you've got a particular species of interest, to consider that species in the context of what's happening with its whole family. So here you're looking at the different insect families along the bottom there, where we've got data, establishment data of at least 25 species in our establishment database. What percentage of those species that have established have been intercepted in the international data set? And you might be a little bit disappointed that most of them are actually under 50%. So you can't fully rely on interception data to predict historical establishments. And you'll see that our forestry orders there, our Curculionidae and our Cerambycidae are sitting around the 50% mark. So in summary, just be aware that the data is messy. There's lots of things to look out for, but I'd also encourage you to consider your favorite species in the context of its entire family. And these are a couple of the papers that you can go to look for some border interception data. So the first one, there's our one that just got published, and also the South African data came out this year. So thanks.
All right, thank you. We have time for 2 or 3 questions, I think.
Hello, my name is Jean-Marc Henin. I'm from the Lab of Wood Technology in Belgium, and I would like to know if you have information about the pathways used by the insects to travel, and in particular the danger that represents wood chips. I would like to know if wood chips is considered as a common pathway for insect invasions or not. Thank you.
Okay, so
yeah,
and sure.
Sorry, a last detail. Could you speak slowly because my English is not so good? So.
Yep, sure. So the different data sets from the different countries are quite different in what types of things they record. So some of them do record pathway information. Some of it's a bit patchy. There's not interceptions on the wood pathways make up quite a small percentage of the data set. So you could do a search for wood chips, but I don't think there'd be that many of them. What else was I going to say? Yeah, so the countries that you might want to look at in terms of pathways that might have wood chips would be like the US and Australia. So they had quite good records on the wood pathways. Yeah.
Do you have do you know some species that were proven to have been exported with the wood chips? Do you have practical examples?
I'd have to do a check. Yeah, because there's thousands of different insect species in this database. Yeah, sorry.
All right, if there are no more questions, I think we'll move on to the last remote talk for the morning. Thank you.
Sorry, can I interrupt you?
Yes.
My PC read my PowerPoint on the Zoom, and now I can do my presentation.
Okay,
Sorry.
great, thank you. Oh, I'm sorry, did you say you can't do your presentation?
Yes, can I start it?
Yes, you can start it.
Yes, thank you. First of all, I'd like to thank all the local staff hosting us. I know how difficult to have such an event today, and I really thank you. What I'll be talking about is invasive boring pests in Japan. You can see 3 longhorn beetles here. Maybe all of you know this Asian longhorn beetle, but others might not be familiar to Western country people. I'm working at the Japanese National Forestry Research Institute, and we are committing research about these beetles and developing deciduous tree protection plans. The long-horn beetles are a large family group, including more than 36,000 species in the world. Here in Japan, they are very popular among kids and insect collectors. The larvae grow in the woods, and it makes them easy to be carried by trade unintentionally. They invade through many pathways: plants for planting, wood packaging materials, timbers, and finished wood products. Most of the longhorn beetles are not a pest. They use dead woods. Occasionally, they become forest primary pests and kill living trees, especially in the new arrival areas. Here we present the current status of invasive longhorn beetle in Japan. The first beetle I talk about is this, the red-necked longhorn beetle. It is native to China, Korea, Mongolia, and Vietnam. This species has invaded Germany, Italy, and Japan, mostly at the same time. In Italy and Japan, it has become a serious pest. The first detection in Japan was in 2011. In these 10 years, it has spread its range into 12 prefectures. Since then, more than 6,500 trees have been attacked by this beetle. With the red-necked longhorn beetle population establishment, we have to conduct a strong tree protection plan to save Japanese apricot, peaches, and cherries. The two trees are fruit trees, and cherries are ornamental. If you come to Japan in spring, you might be surprised to see cherry trees everywhere, on the roadside, gardens, parks, and near harbors. It is our national flower. Such intensive planting might have helped the establishment of the beetle population in many places. The number of damaged trees is higher in cherries. It is due to the abundance of the tree. To develop the controlling methods, we have been working on this research project for 4 years. I'm in charge, and more than 50 researchers have joined. There are two goals. One is the development of chemical and biological controlling methods, and the other is the practical application of innovative new technologies. In the former, we surveyed the ecology of the beetle in Japan and developed a pest control system. In the latter, we mainly worked on the information and monitoring systems, such as real-time online mapping of damages and pheromone trapping. This is the last year of our project. We are now struggling outreach of the results, preparing management manual, pamphlet for caution, and websites for citizens and kids. For kids, I used cartoons like this. Many new control techniques and findings have been obtained. These are some parts of our project outcomes. Trunk injection of chemical pesticide is very effective for larvae. Where chemical use is difficult, we can also use biological control methods. Artificial rearing provides insects for experiments. Real-time online mapping helps local governmental management of the beetle. We have developed chemical lure trapping using sex aggregation pheromone for monitoring. Now, it's time for the Asian longhorn beetle. Secondly, I'd like to talk about this newly invaded beetle. Indeed, we are among Asian countries, but it is not native to Japan. In 2002, It was detected in Yokohama City. Where is the port town? It was found in a limited area and then eradicated. Last year, it was redetected in Kobe City. This city is also a port town. Now, reports of invasion are from four prefectures, but I have heard about more detection reports from amateur entomologists. We cannot predict how severe the total damages will be accumulated in near future. These photos are ALB near our institute. The Asian longhorn beetle is well known for its wide host range. Here in Japan, many tree species are attacked by this beetle. Elms, birches, willows, cutlass, and these horse chestnuts. These are what we see in the damaged areas. It was surprising that the maple tree, which is one of the main host trees in North America, was not attacked. Because it was found in the last year, we shall gather more host species information soon. The last one is this beetle. It was detected this summer in the northeastern area of Japan. I couldn't find its common name in English, but in Chinese, it is called rust color grain shoulder longhorn beetle. Here we call it rusty spotted longhorn beetle. It is native to China, India, Korea, Laos, Myanmar, Thailand, and Vietnam. This species is known as the killer of Japanese pagoda tree. It is a major roadside tree in Asian countries. To prevent its spread in China, internal quarantine has been in place since 1996. The rusty spotted longhorn beetle is attacking two Fabaceae species in Japan. One is a host tree in China, the other is new host species. The rusty spotted longhorn beetle is causing severe damage to trees on the roadside and in gardens. It is a little confusing that the Japanese pagoda tree is not native to Japan. In ancient times, it was brought from the Asian continent. Amur macchia is native species. Both species are planted in the damaged area. Apparently, the beetle preferred Japanese native tree species. While the beetle attacked more than half of the native trees, only one for the non-native Fabaceae. This pest is called the Japanese pagoda tree killer in China. It is possible that this invasive species causes devastating damage on Amur macchia, both in plantation and natural stands. On the one national road, most of this tree was heavily attacked and many of them were killed. It is a problem that we don't have a system for early detection and rapid response for such beetles. As you can see, we are experiencing a series of serious tree pest invasion in our country. This might be a result of the increasing pressure of invasive pests. This graph shows import amount from countries in the original red next long-haul beetle range to Japan. They have increased rapidly in this decade. In addition, we had Olympic Games this year. The national mass event sometimes entails invading pests. For example, pinewheel disease invaded Portugal in 1999, and 1 year before the invasion, they held Expo there. It is difficult to examine, but I guess some impact by the national mass event for this sequential invasion. Let's summarize this presentation. In the last decade, 3 newly invaded longhorn beetles have harmed a wide variety of deciduous trees in Japan. Early detection and rapid response systems for them are strongly needed. Thank you for your attention.
Thank you. We have time for a few questions.
Alain Roch from France. Thank you for your presentation. I want to know if one or all these 3 species you mentioned are already present in forest and not only in man-made habitats.
Sorry, is it about red-necked longhorn beetle?
No, any of the 3 species. Is any of the 3 species present in the forest in Japan?
Yes. No, they are not in forest area, but in urban areas. And we are trying to prevent them spread into natural forests. Okay.
Thank you for your presentation. So my question is, you mentioned that you inject chemicals in the trunk. I wonder what kinds of chemicals do you inject to control the beetles?
Yes, it's neonicotinoid pesticides.
Do you know the specific name of the pesticide?
Yeah, it's sorry. We only know the name of the pesticide in Japanese name, so I can't tell the name of the pesticide.
Okay. If you can write it on the chat, that would be
yes,
fine. Thank you.
Yes, I will write on the chat later. Sorry.
Let's see, Rebecca just shared that some of the species, this is about the wood chip question from the previous talk. Some of the species intercepted on wood chips are Bostrichidae, Savannidae, for example, Dinodaris minutus, Synoxylon unidentatum, Ahasverus advena, Heterobostricus equalis, and Lacioderma sericorne. Are there any more questions, Sandy?
Thank you, Etsuko, for the really nice talk. And I was just wondering if, because there has been so many of these longhorn beetle invasions recently, I wonder if the Japan Quarantine Agency is considering new measures, phytosanitary measures for imported wood and wood packaging.
Thank you for your comment, Sandy. Sadly, our government have no new measure for the sequential invasion. And I tried to talk them about how it is problematic for our society, but they don't try to start new measures. Thank you.
All right. Thank you for all of those remote talks. I think we'll go ahead and jump into the in-person talks now. Yeah, Sandy. Oh, sure. All right. For our first in-person talk of the day, we have Eki Brockerhoff, who will be speaking on bridgehead effects in insect and pathogen invasions.
Thanks, Jake. And thanks everyone for attending, and to the organizers for the very nice conference and the fantastic field trip yesterday, which I really enjoyed, although I think I ate too much. And yeah, so bridgehead effects, and I should just briefly acknowledge my co-authors on this, most of which are part of the succinct group that was already introduced by Rebecca Turner a moment ago. So what are bridgehead effects? We can see here a simple scheme that shows the native range of species and thrust populations that may be translocated to a non-native range that may be on another continent, and like in those two non-native ranges here. And so they can become a bridgehead population from where they spread and form secondary introductions. If this is in the same continent, it's just a fringe spread, that's nothing too surprising. But these bridgehead populations can also lead to secondary introductions that are transcontinental, so that a new introduction in a new continent actually doesn't originate in the native range, but from an invaded range. And if we have a look at what that means, we can use this unified framework for biological invasions from Blackburn and co-authors, and you see at the top the successive stages that are very common and universal for invasions. It begins with transport from the native range or another range, introduction, arrival, establishment, and then spread. And these bridgehead effects basically enhance transport, and so they accelerate potentially this invasion process. And the term bridgehead effect actually comes from the military, but in this context, it's been used the first time in the literature for the harlequin ladybird, Harmonia exoridis, which is native to Eastern Asia. And it was introduced to North America, and then from there, it spread to multiple regions in other parts of the world. So I've done a review on the literature on this phenomenon, and there's quite a few papers that have been published, several in recent years. But in total, there are actually less than 30 published papers that I could find on this topic. And only some of them actually have original data, like several are theoretical or review papers. So there isn't actually that much information yet. And so what is the cause of these bridgehead effects? Many invaders are rare in their native range, and some are not even known as pests or not known at all, but they can become very abundant in their non-native range. And so the reasons are or can be that often there are large host resources in these non-native ranges, like in plantations or crops. There is often less competition, especially if the host is also non-native, which means there are fewer other species to compete for this resource. And often there are also fewer natural enemies, so they benefit from enemy release. And so as a result, these secondary invasions are theoretically more likely than primary ones. And we've already seen this, something very similar in Nicolas Meurice's talk a moment ago. This is a radiata pine plantation in New Zealand. There's a lot of this, and it is a huge resource with very little competition because the tree, and in fact, there are no native pines at all, no native Pinaceae whatsoever in New Zealand. So there's a relatively limited native fauna that colonizes this, and not so many invasive species yet that have colonized this. But one that clearly benefited a lot from this is Halorchis ligniperda, the red-haired pine bark beetle, which is super abundant there. And so how do we know that bridgehead effects can play a role in invasions? They're rarely seen or documented. So when a species turns up, in few cases, do we have some information how it actually arrived or when exactly arrived. So it's quite difficult to determine the actual source of an invasion. However, we have these border interception records Rebecca Turner already spoke about, and we also have evidence from population genetics. And so 2 examples on interception data that give us some idea on this on bark beetles. Ips grandicollis, native to North America, that was detected in Australia in 1943, and it's been intercepted many times in New Zealand, mostly and initially from North America, but after the establishment in Australia, also quite commonly with imports from Australia. And another example, this Halorchis ligniperda I just mentioned, it's native to Europe, mainly invaded many regions, including Australia, South America, South Africa, New Zealand, etc. And so we had very few interceptions from Europe, a few from Chile, and 19 from Australia. And so if we have a look at the world map here that shows in green the native range of Halorchis ligniperda, so it's Europe, parts of North Africa, and then it probably stretches across Russia. There's only one record of a native population occurring somewhere in southern Siberia. And we're not quite sure about the situation in Japan and northern China, so there's some uncertainty there. But you can see it's invaded in Australia. That was the first documented invasion in 1943. It was detected there. Then in Uruguay, Brazil, South Africa, New Zealand, Chile, in the state of New York, in California, in Argentina, and supposedly it's also in Sri Lanka, where there are pine plantations at higher elevations, but I haven't been able to confirm that yet. So you can see this has been a super successful invader, and I believe it's actually the most successful invasive bark beetle there is. And so I had a PhD student who you can't quite see there on this slide now, unfortunately, but hello, Etsuko. And so Lea Bischofberger, she did a master's project with me, and we got samples from all over the world, not quite all the regions where Halorchis ligniperda occurs, but a very large proportion of them. And you can see on the left there, the 257 specimens that she analyzed from all the countries listed there, and the haplotypes you see there are color-coded by country. So you can see there are quite a few haplotypes, and the largest, okay, for some reason the animation just got lost there. So yeah, so the largest haplotype groups here are the ones with specimens from Argentina and Chile, then one from Italy, New Zealand, and the US. There is this group here, Argentina and Uruguay, and one with specimens from France, Uruguay, and South Africa. And some of the European ones were unique that we didn't find anywhere in the invaded populations, but there are probably a lot more because we haven't been able to sample from all the natural range in Europe yet. And so what that means in terms of what we can learn about the likely invasion routes of Halorhis ligniperda, considering together the evidence from genetics, interceptions, and the year of the detections, is that the first invasion in Australia most probably originated from the native range in Europe. And the invasion in New Zealand quite possibly may have come from Australia, where we had the most interceptions, many more than from Europe. And it's quite close to New Zealand, and there's a lot of trade. Then Uruguay was next, where most probably this was an introduction from the native range. And then in South Africa, we have both a lot of haplotypes from Europe, but also some that are shared with Uruguay. So we're not quite sure. It could have been a mix of the two origins. Chile, probably from the native range. We haven't got any other evidence to the contrary. And the populations in Argentina are very similar to those from Chile, so they probably just hopped over the Andes there. Then in California, we have seen a large similarity with the specimens from New Zealand. And although there are also imports from Europe, obviously, we think there's some evidence that suggests that the Californian population may have come across the Pacific from New Zealand, with log imports possibly. So just briefly to conclude, an example from pathogens, which there's a few examples where bridgeheads have also been implicated. And so this Gryphonectria parasitica, the causal agent of chestnut blight, is a good example here, where a bridgehead in North America that was most probably invaded from the native region in Eastern Asia has served as the agent that invaded Europe. This was complicated, though, by a subsequent introduction of a different genotype coming from Asia, and more recently, the emergence of a new lineage that spread into southern Italy and the Balkans. So often you have this complex image here where it becomes harder and harder to ascertain where an invasion has actually originated. And so to conclude, bridgehead effects are a common phenomenon in insect and pathogen invasions. And the snowball effect that I showed with the Blackburn diagram at the beginning most probably enhances invasions and their impacts. So it's difficult to determine with certainty, though, where these origins are, because there are no direct observations, and the indirect evidence doesn't give us complete proof. However, they will likely become more common as invasions continue and spread, and we have more and more invasive species being very common in more and more parts of the world. So all stakeholders should do their best to reduce bridgehead effects and invasions per se. And basically that means across the entire biosecurity system, targeting prevention of introductions, but also response and management. And so with that, just another photo of the succinct group. And so I think I have not actually gone over. So thanks very much.
A few questions.
There is a message from Brenda Mike Wingfield. Thanks for your nice talk, Eki. I know that I have raised this question before, but I became excited at the thought that we can back up the movement of H. ligniperda with an understanding of their fungal symbionts. I believe there are patterns. The fungi can be quite good taxonomists of the beetles, Mike.
Yeah, thanks, Brenda, Mike. So yeah, thanks for the question. I agree entirely. I think this a really nice system to also consider the Ophiostoma and other associated fungi. And I look forward to working with you on that, Mike.
Thank you for your presentation. My question is for the red-haired pine beetle. Do you think it is primary pest or secondary pest as attacking the stress trees? What do you think about that?
So in Europe, it's actually not even considered a pest at all because it basically feeds on deadwood and it's not known to really cause any primary damage to live plants. And the only issue why it is a pest in southern hemisphere countries where have these plantations is that it can be a quarantine pest, so it can be exported with logs and wood packaging if it still has bark attached and so on. And so that's an issue because importing countries don't want to receive this pest or the species, even though it's not so serious. Yeah, so in this particular case, it's not actually a big deal. And in Europe, relatively few people have studied it. And so it's nowhere near as bad as some other bark beetles. I guess we're lucky that this is not Ipsipographus or some serious pine bark beetle that has spread in the same way. So this is a question for Mike, which we've already had. And the previous one, that was the answer from Rebecca. So I think there's no more questions in the chat and not in the room either. So thanks again.
Thanks, Eki. Next, we have Yuri Trombik, who will be speaking on true fruit fly invasions, history, and global drivers.
So good morning, ladies and gentlemen. Before I start, I will maybe briefly introduce myself in a context where I'm working on. Maybe you remember that at his introduction talk, Marek mentioned that we have this big EVA project, and one of the subgroups that works there is basically dedicated to the invasions or invasion history of mainly tree pests. And he was saying that, I don't remember the direct quote, that we are doing something nice there. Okay, so one of the hopefully nice things that we do there is that we were trying to basically replicate the history and try to figure out the global drivers of true fly invasions. Now, since I'm in the company of mostly entomologists, I would guess you probably know about the fruit flies much more than I do. But just some basics. There are estimation is that there are more than 5,000 species of true flies worldwide. What is important for us or for the true fly invasion is that most of them feed on fruit, which also means that fruit production and fruit export is the main vortex of the possible invasions. Some of these species that already invaded some parts of the world are considered as one of the most damaging agents of fruits and vegetables in the species. Just this one number, economic damage associated with just one of these species, Bacteria dorsalis, is estimated to be $1 billion annually worldwide. So what we tried to do with this group of species, the first step was that we would try to assemble comprehensive database of 10-day invasions. And based on this database, we were trying to identify the biological and socio-economical drivers that could possibly explain these invasions. Now, for the first step, meaning doing the database of history of all the invasions that we could find. Now this is a little bit confusing even for me, but in our case, what we meant by invasion is that it was species that was unintentionally established. So no eradications, no biological agents, and that we were trying to find the information on the country level. So country level was our base level for all the analysis after. For creation of the database, we use various sources, mainly classically scientific literature, but I think the complete baseline was the CABI invasive compendium. I hope I'm saying it right. If data was available, we will also try to find the first record for each country and each invasion. That sadly wasn't the case, but I will get to that later. And just for the quick summary, I also, for this talk, summarized the data for each of the 8 biogeographical or biological realms. So once we got the database done, we found that there were 44 non-native Tephritida species that were able to successfully and unintentionally establish somewhere else in the world. If we summarize it for those biological realms that you see on the map on the top, hopefully, the most invasions and also the region of origin of most invasive species, we found one was European Palearctic region, followed by Asian Palearctic region. There are 3 regions that were kind of interesting for us because Nearctic region and Oceania were the source of very few invasions, but were recipient of total number of 11 invasions. Sorry. The other scenario was that Indomalaya region was the source of 10 total of 10 invasion or invasive species, and only 6 species successfully established in this region. When we look at the country level, these 2 maps are the map A, or the map on the upside of the picture is the number of successfully established non-native Tephritida in each country in the world. You can see that basically there were documented invasions in almost every country in the world where there are some data, exception of course Antarctica, where you probably don't find much fruit. The most invaded country is US, of course. I know that Aljaška part looks different, but as I said at the start, we did this whole analysis on the country level. Also, too important that a lot of these invasions of fruit flies happens in a remote or on the islands. 3 examples: islands of Hawaii as part of the US, and also African islands of Mauritius and Reunion, which got 6 documented invasions in total. And the bottom up is the origin or number of invasive Tephritida that originate from each country. You can see that the biggest number of invasive Tephritida origin from Europe, surprisingly. Also, one of the main sources of invasions is India. Now, if we look at the invasions based on species and also through time, the half of the invasive Tephritida in total only invaded or have a documented invasion only in 1 or 2 countries. There are 6 exceptions, like the most notorious and the most well-known invaders, which is Carpomia vesuvinea, Aures tabulans, Bacteria ole, Zaugustus cicurbidae, if I'm saying that right, Bacteria dorsalis, and Ceratinis capitana. If you look at the number of invasion through time, here in this graph B, I summarize the invasion in decades. And you can see two distinguished peaks in recorded number of invasions of fruit flies. One is in the 1930s, right before the First World War. And then you can see sort of linear increase in found invasions since the 90s, basically. With the globe. There was this, on Monday, there was this interesting talk about the two ways of globalization. So I think the Teflitida invasion sort of followed this way. A little bit of the problem with this data is that I think from the total number of 400 invasions that we got in the database, we don't know the date of establishment for half of them. So sadly, that could possibly impact our results a bit. Now that we have that hopefully best database that we can get about the invasions, we were trying to figure out what were the main drivers or main patterns in these invasions and what influenced them. We came up with a bunch of possible predictors that we thought could have some effect on the probability of invasion. That were sort of human factors, I would say, or human or factors affected by human stuff. That is human population in each country, gross domestic product, number of visiting tourists, cumulative imports from native region to target country, fruit production, and country area. I think all of them are pretty straightforward. You can sort of imagine what these variables are. The only thing maybe that I would need to explain better is that we also use this climatic predictor, which we call climatic similarity. What we did was basically we used Shannon index to compare the target or the country in the model matrix with the estimated climatic niche of each of the Teflitida species. To define this niche, we basically took all the records from GBIF database of each species and calculate the fraction of the country that have suitable conditions for the invasion. Now, to figure out the most significant predictors of invasion, we used general linear mixed effect models. First, we basically tried full best fitting model with all the predictors that we could find and see what sticks. And then, because a lot of these variables that we use were correlated a lot, and also we wanted to sort of narrow down the number of predictors just for the most significant ones. We first removed, based on IC scores, we removed the predictors that were covariated. Covariated predictors, basically two groups. One, again, sort of affected by human stuff. We found that the best matching in our model was to answer the question about the possibility of invasion was GDP. And then the second group is this climatic similarity, or we used another variable which was suitable area in each country that country could invade. And in that, the most impactful factor that we found was climatic similarity. Now here is just the final model. Intercept is basically just if country was invaded or wasn't invaded. And based on our model, we found out that by far the most impactful factor that predicts the possibility of Teflitida invasion is climatic similarity between the native region and the invaded country, followed by GDP, basically the economic importance of the country, let's say. And then the final was slightly confusing with negative correlation, but distant between native and invaded range. In conclusion, what we found out about studying these Tephritida is that the number of non-native species that established somewhere in the world varies considerably among regions. Mainly, the main invaded regions are European Polarctic and Arctic regions. Almost, as I said before, there is documented invasion in almost every country in the world except Antarctica. Most of the invasive species were documented only in 1 or 2 countries, except those 6 most notorious invaders that were able to establish all over the world. We found out that the best predictor of Tephritida invasion is climatic suitability or similarity of climate between native and invaded region. From that, we can sort of conclude that since the most of the species, also based on that interception data that Rebecca was talking before, haven't yet established in their whole possible area of distribution. And if the climatic suitability is the key predictor, that we can sort of accept more of these invasions in the future, especially with the changing climate. Okay, that's from me. Thank you.
We have time for 1 or 2 questions.
Thank you for your talk. I was interested by the fact that you calculated the fraction of a country which was occupied by a certain Köppen-Geiger climate category. Did you build up a script to do that?
Yeah, I'm a GIS guy, so I sort of just work the model in the GIS facility. But no script. It's basically just common tools used in ArcGIS that I'm using. But if you're really interested, I could send it your way if you want, or we can talk about it later.
Yeah, thanks for a very nice talk. Just to comment, I believe there have been a few interceptions and establishments in New Zealand, but they were all eradicated successfully. So, and there are fruit there. So it's not only Antarctica, and there is actually a good track record in eradications if they're detected early.
Yeah. We sort of got also a summary of eradications, but I sort of didn't want to put it in because I found it confusing even as it is what we consider invasion and whatnot. So I didn't want to include that. And the problem is that since we didn't know so many of these first records, we couldn't sort of include the temporal or time in our analysis, which the eradications would make even like more messy, I would say. But you're right, there should be somehow included, I think, maybe in the future.
Hi, I saw you didn't find an effect of cumulative imports, but did you try looking at just fruit imports?
No, because I couldn't find the long enough timeline of fruit exports to sort of try to figure it out, because I didn't mention it, but the cumulative imports, we basically use this TradeHill database. I don't know if you heard about it. That's cumulative imports or imports for each year, like or pairwise between countries since 1850, I think, or 1890 until 2010, I think. But for the fruit like imports, I don't find any data. So we use only fruit production as a predictor.
Yeah, you can.
And I should also mention that actually the cumulative imports were sort of significant for the prediction of invasions, but were sort of biased, or the other predictors were much more dominant in our analysis. But if you know about any data about fruit imports since 1900s or something, Yeah. But for all the countries? Yeah. 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 For what I was looking for, I was able to find only like for the developed countries, let's say, not all the islands and stuff like that. And trade was the only one that covers the whole world. But you're right.
All right, thank you. And we will go now into our last talk from the morning, after which there will be a brief coffee break, and you're welcome to check out the posters that are downstairs. And this last talk is Richard Malley, and he'll be speaking on biography of Lepidoptera invasions worldwide.
Thanks a lot. Okay, hi, welcome to the last session of this morning session. I will present you another nice topic from our small invasive insects working group that Sandy is heading here at the Czech University of Life Sciences. I'm a trained taxonomist and phylogeneticist on Lepidoptera, so this is a very sexy topic for me. I hope you find it as sexy as I do. Lepidoptera, as you know, are one of the largest radiations of insects. There's about 140,000 species described as the last time I checked. Their evolution is tightly linked to the angiosperm evolution, mostly since these are major pollinators of angiosperms. And they're also one of the largest radiations in the animal kingdom of herbivores, since pretty much most larval stages feed on some part of an angiosperm or plants in general. Being a major radiation of plants of herbivores, you have a lot of pest species, of course. Among them, such notable things as Limantria, the codling moth. So you have silvicultural, agricultural pests. You have horticultural pests like the box tree moth that is now also spreading in North America, as I've heard. But you don't even have to leave your house. You have even pest species in your home, clothes moths and these notorious Indian meal moths in your cereals are most notable, probably. With this study, we wanted to look into several things. Apparently not the established Lepidoptera are not equally spread among families of Lepidoptera. So we wanted to look into other families that are more successful than others. Some families maybe not even established at all. We looked into this classical species area relationship. Is there, do we find land area to account for the number of established Lepidoptera in a certain area? And do we find disharmonies between established and native species, meaning are some families overrepresented or underrepresented as compared to the native fauna? And can we explain these over under representations by bringing on this different propagular pressure proxied by species richness in port intersections? And for this, I will we use, for example, Rebecca's interceptions data she talked about in the morning. We looked at 10 different geographical regions in the world with vastly different land areas, ranging from the biggest North America to, I think the smallest is the Ogasawara Islands, south of mainland Japan. And we looked into these regions because for those we have fairly recent and quite complete lists of total species, of total Lepidoptera species. Plus we had this assessing data that again, Rebecca and Eki mentioned, of lists of established species for these 10 regions. So we could easily calculate native species by subtracting the established from the total species and go into analysis with that. I mentioned we also used port interceptions to look into propagule pressure, if that plays a role in overrepresentations in invasion disharmony. I hope that won't happen again. Wow. Yeah, okay, we looked into species interception, and we also used Gida's trade commodities data. You've probably seen her talk on Tuesday. We used the Lepidoptera part of that to look for further clues what could potentially cause these invasion how did I call them, disharmonies, correct? Yeah. What could they? What could be? What could trigger? For our first question, we got this answer: 742 unintentionally introduced species in 59 families were among our data set. And for this, we actually excluded all the intentionally introduced species, which were about 100. That's quite a number. And surprisingly, the two largest families, Arabida and Geometrida, are among the ones that do actually the worst. Arabida, by the way, are now the largest family. There's a bit of rearrangement in the systematics of Lepidoptera a couple of years ago. Before that, it was Noctuida. Now a lot of things have moved to Arabida. Arctiida are there now, Limantriida. They are all in this big group of Arabids. Yeah, apparently they do surprisingly bad concerning regarding their, considering, that's the best word, considering their global diversity. Whereas these other families, Noctuida, Tortoisida, Crambida, Puralida, Tinida, and Gracilariida, we find they have exceptionally large numbers of introduced, of established non-native species. So that was quite surprising to find these species, to find these families doing so well. As for this species area relationships, that was not too much of a surprise. We expected there's this classical log-log linear relationship, especially for native species. As you can see in the non-native species plot on the right side, Hawaii is quite an outlier. So is North America. So there's a bit of more variety going on. Do we find disharmonies between established and native species? We actually do. Yes, we find these invasion disharmonies. We find, as we saw in this bar chart before, 5 families substantially overrepresented in the establishment data. These are again Noctuida, Crambida, Tinaida, Puralida, and Gracilariida. And these two largest families, Arabida and Geometrida, are substantially underrepresented. If we look into the 10 regions in more detail, we find a more varied pattern. Tinaida and Puralida, you will see them in many regions overrepresented. Australia, Japan, South Korea, they are doing really very, very well, the established species in many regions. Whereas again, Geometrida, for example, are underrepresented in many regions. Hawaii is exceptional in its diversity of families that are out of the expected distribution. If we now look into species interceptions of these established species, and compare them with established families, then we can see at least 3 of those overrepresented families, Noctuidae, Grumbidae, and Pyralidae. We also find them in interceptions. So they are over average intercepted at ports of entries, whereas Geometridae, whereas for Geometridae it's the same, they are mostly underrepresented. For these 2 families, Tineidae and Gracillaridae, we don't find that, so they are not overly often intercepted at ports, and neither are Arabidae underrepresented in the interceptions. What we also find is Sphingidae and Papilionidae. These are hawk moths and swallowtail butterflies, quite fleshy species with remarkable larvae. They are often they're overrepresented actually in the interceptions, and that does not translate into establishment of these families in non-native ranges. So they're probably picked more often in interception, found more often because there's a particular focus on them. They are just simply well spotted. We also find this interception bias. Macromoths are generally, so this is a comparison of established versus intercepted species. Macromoths are often underrepresented. They are less successful in establishment as would be expected from the interceptions. Whereas micromoths, these tiny things that you barely can see, 10 millimeters or less in size, and the larvae even smaller. They're often overlooked apparently in establishment in interceptions, and can establish much more successfully than you would anticipate from the data. Looking into trade commodities, this is the subset of Lepidoptera. We find these 5 families, these 5 successful families, as it's probably not surprising, mostly feeding on plant commodities, on plant products. Tineidae as mostly detritivores, they are also found a bit more on animal products and wood products, but the general pattern is the same for most of them. If we only look into the plant commodities, the picture is quite a bit of a different one. Gracillaridae, which are leaf miners. You've seen these Robinia leaf miners yesterday on the excursion, Parectropa and Macrostachys. So they are leaf miners. We find them in the commodities, mostly associated to fruits and nuts, which is to me a bit puzzling. I still can't explain this pattern. But it's an interesting thing. Maybe they are more screened for in nuts and fruits, and I will talk to you about this as well, Gita. I'm curious what you think. Yeah. So this raises a lot of future questions. If we can explain partly the success of these 5 families by propagal pressures, but for others not, is there some pattern maybe that we can look into in the future? Noctuidae and Grumbidae, for example, are often polyphagous species. They're in the noctuids, there's this particular pest clade that harbors most of the pest species in noctuids. So there's apparently an evolutionary lineage that is specifically polyphagous. Pyralidae and Tineidae, which do very well in many regions, mostly detritophagous, so they feed on they can feed on anything as long as it's somehow organic. And species from these 2 families have been established a long time in many regions because of their ease of finding food. For Gracillaridae, yeah, I'm still puzzled how they can be so successful, because they are mostly leaf miners, as I said. You should be able to spot leaf mines easily in plant commodities. So this is again a bit of a puzzle how we can explain gracilar success. Same thing for Arabic and geometry. Why are they so unsuccessful in establishing in different regions? What the host plant data suggests is that they're mostly relatively host specific, mono or oligophagous, with a few exceptions. Limantria dispar is probably, I think, recorded from 52 families, so this is really an exception. The same goes for geometrids. Geometrids are often confined to woody plants, which are probably less moved in global trade. And another feature of geometrids that might diminish their success in establishing is there's you find often wing reduction in geometrid females. So this is an extreme case of in the winter moth where the female is completely flightless. So this might actually hinder geometrids from establishing in a new region and spreading and establishing successfully in population. In general, in summary, we find this general classical rule of the larger the area, the more non-native species that are established. 5 families stand out as being very successful among established Lepidoptera. The 2 most species-rich families are underrepresented, and we still don't know really why. And propagules pressure can at least explain, in part, the establishment success of noctiids, crambids, and pyralids, but not for teniates and gracillarids. So this is something I would like to look into in the future, mostly through host plant data, through feeding guilds, and find out a bit more what the different success strategies might be for these families. And that's it. Thank you very much.
Another bomb here. Thank you for the presentation. Oh, here. Very interesting, basic information. I'm wondering how you see this applicable to the control management of invasions.
Yes. Well, we know now which families are quite successful in establishing, apparently. We know which of them are pest species. So this gives us a more focused view on what to look for. Especially, for example, leaf miners are maybe are obviously overlooked in the inspections. They were in this micromoth group. So that gives us certainly a bit of more focus, what to look for. We know noctiids are overrepresented. We know there's this pest clade in this pest lineage in the noctiids. So this is certainly worth looking more into, because there might be a lot of potential pest species. The same goes for crambids. We now know also a lot more about the systematics, the evolutionary, the evolution of these species. And there's a few lineages that are very confined to certain groups, like Poaceae, that they feed on grasses, and they might be potentially future species becoming established in different regions, because they feed on like target crops, for example.
Yeah, thanks for the excellent talk, Richard. I am wondering if you split Erebidae into the former families, you will get something else. For instance, Limantridae may be interesting to be considered.
I kept the data on subfamily level, so I can easily split them off. And this is actually a good point, because like I said, there's this pest clade in noctiids, for example, there's different lineages that are different, more successful than others. So we can split that more down, get it more fine scaled, and find out which actually which group in Arabics, for example, are particularly successful. Yes, because yeah, arcades, Arabics, they are not very common among these. Okay.
All right. Thank you, everyone. If you want to maybe give one more round of applause for all the speakers, and then Sandy.
back into the room, but apparently not. So I didn't realize I was also responsible for that. Anyway, so welcome back, and we're not much behind. I believe we also went over a little bit with the previous session. Anyway, so we have 5 more presentations before lunch, and so I guess you all know me from my presentation just before, so I won't say anything about who I am. And the first speaker is Jessica Roots from the Forest Entomology Lab of the very well represented University of Minnesota here. And her presentation is on relationships between regeneration, spruce budworm outbreaks, and wolves. Over to you.
Thank you, Eki, for that introduction. I appreciate it. Dobre rano. Good morning and welcome, everyone. I'm excited to be here today to discuss my research regarding the ecological role in forest regeneration of, and forest regeneration impacts of the eastern spruce budworm in both Minnesota and Isle Royale. The eastern spruce budworm, or Coristinura frumiferana, is a defoliator native to northeastern North America. And as Jean-Noel Cando described in his presentation on Tuesday, the insect goes through cyclical outbreaks that occur every 35 to 40 years and can last 7 to 10 years. The insect's preferred hosts include Abies balsamea and Pichia glauca, respectively. Damage caused by eastern spruce budworm can be quite extensive during these outbreaks. The insect prefers to start at the crown of the tree and work its way down. And it mines the needles, and after approximately 2 to 3 years of the outbreaks beginning, one can start seeing diameter growth loss of the trees. And after those 3 years, between 3 and 5 years, you can start seeing tree mortality. One of the landscape concerns of the eastern spruce budworm is general volume loss. In the last outbreak in the 1970s, there was approximated 52 million hectares of defoliation. It is also predicted that this insect will incur between $3.3 and $4.7 billion of economic loss by 2041. There are also ecological impacts that will be that are caused by the eastern spruce budworm, and that includes desedimentation and nutrient runoff that occurs due to salvage logging and general habitat loss. The objective of our research was to see whether or not moose herbivory is affecting forest regeneration in areas in areas of forest gaps following eastern spruce budworm outbreaks. And in order to answer this question, we chose 2 different locations, one of high moose density and one of low moose density. We were particularly motivated by Isle Royale, which is an international bioreserve and an island. The International Bioreserve, as I said, is an island, and it has approximately 132,000 acres of land and is surrounded by 450 other small islands. Because of this locked system, it makes it a perfect system to use for a comparative research study. The area or island is also the location of the world's longest longest standing predator-prey relationship study. And I'm excited to contribute to that by focusing on a lower trophic level. Eastern spruce budworm has never been studied on Isle Royale, and its population has only ever been monitored via aerial surveys. And so I'm very excited to participate. We are using Isle Royale to see whether or not wolf reintroduction will help forest regeneration post-eastern spruce budworm outbreaks. And in order to do this, again, we were relating different densities of moose populations to regeneration capabilities within the forests. Isle Royale specifically currently has very high densities of moose. There are 2 to 10 moose per square mile on Isle Royale versus the traditional 1 moose per square mile in northern Minnesota. Though there have been wolves on the island before, In 2018, there were only 2 wolves left due to a canine disease that was introduced to the island. And only recently did they reintroduce new populations that will hopefully help that regeneration. Again, the study locations that we've chosen are northern Minnesota and Isle Royale. Northern Minnesota is depicted here on the top left in the blue. Being one of these this is one of the locations, but there are multiple sites within that location. And on the right is the Isle Royale location, in which there are also numerous sites. On the left is the low density location, and on the right is the high density location. We chose these locations not only because of the differing moose densities and the fact that the island is a locked system, but because they have similar latitudes, and we are hoping that these similar latitudes will exclude any additional climactic factors in the study. Here you can see a map of Isle Royale, and these highlighted areas of various colors are all areas that were affected by eastern spruce budworm approximately 15 years ago. And it is within these areas that I and my colleagues, or my colleagues and I, chose sites for the study. And I have similar sites from northern Minnesota, though I'll spare you and not show you today. In order to perform the measurements for the vegetation, in each site, we set up a 100-meter long transect. And along this transect, we had 10 sections. In each section, we had each section had a 10-meter diameter. Data collected within these transects or along these transects were tree height and diameter. Diameter was taken at both breast height and knee height, depending on the tree height itself. We also took note of the browsing spherity of the trees and on using a classification that has been used in 2 previous studies. And on the bottom left here, you can see a branch that's been that has not been browsed by a moose. And on the bottom right, you can see branches that have been. And so seeing the 2 differences, I found it relatively easy to tell the difference. Additional data collected includes canopy density. And in order to measure the canopy density, we used a densitometer. And in each section, one of the 10 sections along the transect, we took 5 measurements, totaling 50 measurements for canopy density for each transect. We also took note of the stand composition by calculating the proportion of deciduous versus coniferous trees. Now, one of the reasons the canopy density measurements were important was to ensure that we were actually located and doing research within eastern spruce budworm created forest gaps. And I know it doesn't seem very intuitive, but the image on the right here shows how it is actually a forest gap. Now, there are trees, but they're deciduous, and traditionally we would expect coniferous trees in this stand as well. One of the things we were seeing after collecting data were that the tree heights for Minnesota were very similar. All of the trees ranged very little for the average height. And we're hoping, we also took note of whether or not the apical stems were missing and that impact on the height of the tree itself. And we're hoping that this will represent the forest regeneration capabilities. Additionally, we were seeing that very few trees actually experienced drastic browsing on mainland Minnesota. Drastic browsing here is represented in green, and slight browsing is represented in blue. And this is expected. Mainland Minnesota, again, is the location in which we expected very slight browsing due to the fact that there was lower or average moose densities. And also, and additionally, northern Minnesota approximately has 2,700 wolves. So again, drawing the connection between forest regeneration capabilities. This is a project in the works. I am a brand new PhD student. I started in July and I collected my data in August, and now I'm here. However, this is a very exciting study, and I have had a lot of help, and we've been able to collect data in one location and have been able to select sites in both Isle Royale and Minnesota. And we're hoping to collect the data in the sites of Isle Royale next spring. When we get the chance to do that, Besides being just a lovely location and semi a vacation, we expect to see very large differences. And this isn't just due to the expectation that results from the differing moose densities. As expected, or due to the higher density of moose on Isle Royale, again, 2 to 10 moose per square mile versus the 1 moose per square mile, we would expect to see very slow regeneration and a lot of moose browsing. But I actually did get the chance to notice this and visually and see this while I was there scouting for sites. On the top right here, you can see mainland Minnesota sites, and they were, these trees, though they had browsing, were regenerating relatively well and were not very cylindrical or dense. However, the trees on the bottom right are the trees found in Isle Royale, and they were extremely cylindrical and dense and experienced a lot of browsing and were much shorter. At this point in time, I'd like to take a moment to thank everybody who helped my research. That includes the National Park Service and United States Forest Service, the University of Minnesota, and my wonderful group at the Alchema Lab in University of Minnesota. Thank you so much. And I'll take questions. Sure.
So yeah, thanks very much for the very nice talk. So quite interesting and also fascinating that you can present already after such a short time. And also you seem very privileged to be working in such a cool place. And it's slightly off topic, but I was wondering, would you require any field assistance for your field work?
No, the work's very easy. I'm completely joking. Actually, yeah, I very much require assistance in both locations. Isle Royale, though beautiful, is relatively rugged. And this summer I backpacked approximately 20 miles in order to get even the sites found. I received a lot of help within my lab. Actually, there are a few people sitting here who helped me extensively. That would be Grace Haynes, who backpacked with me across Isle Royale. Patrick, who helped me collect data for over 400 trees. And Emily Althoff, who helped me also collect mainland Minnesota data. Thank you.
Okay, thanks. Yeah, so I was just hinting that if you need additional support, I might come in and help you. Okay, so no more questions. I guess we still have time because you didn't even get close to using up your time, but maybe not. So we'll make up a few minutes perhaps. Oh, sure, yeah, go ahead.
Thank you for your talk. It was really very nice. And maybe I missed something, but are you planning, or did you already compare the ground vegetation and the other plants growing in your two sites?
So that's thank you for your question. I appreciate it. The only other understory vegetation analysis that we're performing currently are whether or not there is brush just in general, and we did that by a percentage. So each section has 4 quadrants, and we calculated that percentage and what type of brush we were seeing within each quadrant. Thank you.
Okay, thanks. We should move on, but another show of your hands and appreciation. So next up, we have Romina Gazis, who is an assistant professor in plant pathology and the director of the Plant Diagnostic Clinic at the University of Florida. And she will be speaking on research and management for laurel wilt in Florida, or perhaps further beyond. Over to you.
Thank you. Good morning, everyone, and thank you for the introduction. My name is Romina Gazis, and I've come all the way from Florida. So I'm being a little bit cold here in this weather. So today I'm very happy to share with you the research that we are doing at the Tropical Research and Education Center. This is a collaborative research. For some of you that perhaps haven't heard about this disease, This is a very, very bad disease. It's a disease complex, and it involves fungus in the Opistomatelis group named Raffaella lauricola, and it is a vascular pathogen that is also a nutritional symbiont of ambrosia beetles. The main vector is a red bay ambrosia beetle, Oxyloborus glabratus. This is the original vector, and it carries the fungus in these specialized pockets called Mackenja, and also it gardens and it cultures and cultivates the fungus in the galleries it makes inside the tree. And this disease affects several species in the Lauraceae family, especially very susceptible the genus Persea, including the avocado crop. Now, very important to mention is that this beetle is not attracted to stressed trees. So apparently it is attracted to healthy hosts, apparently healthy hosts. Now, it's not attracted to ethanol, so it's very difficult to sample and to survey. The female mates prior to emergence from natal trees, so it's already ready to make galleries and put some eggs. And so one female can actually start an outbreak. Also very important to mention that in the avocado system, there is multiple ambrosia beetles carrying this fungus. In Florida, there is up to 9 native and naturalized ambrosia beetles that have been found carrying this fungus. And it has been proven for 2 of them that they are able to transmit effectively the fungus into healthy avocado trees. Now, the outcome of this host pathogen interaction results in the collapse of the vascular system and in the occlusion of this vascular system due to the induction of tylosis production. Now, once the fungus colonizes the xylem, and as the day passes and the biomass increases, tylosis really becomes prominent and extensive. And at the end, it really clogs the xylem and prevents the water to flow from the roots up into the canopy. So what you're gonna see is drought stress symptoms. And in avocado, you are gonna start seeing these symptoms after 14 days after the tree is inoculated. This is under control conditions. And then a little bit after a month, the tree will die completely. Now, these are some of the symptoms that you will see in the field. Completely wilt of the tree. Usually the leaves remain attached to the branches because of this rapid death of the vascular system. Eventually these leaves are gonna fall off the tree. Now, in the avocado system, this is how it works. One of these beetles, any of these alternative beetles, because Xyloborus glabratus does not like avocado, but the other ones do very much. So they can inoculate one tree in an orchard, and then only remember when I mentioned that only one female is able to, even one only female is able to produce an outbreak. Once this tree is inoculated and infected, the fungus can move through root graft through the row of avocados. So if this disease is not managed, the grower can lose up to 100 trees in 6 months. So it's a pretty aggressive, rapidly moving disease. These are some of the pictures that I took around where I live. So there's an extensive damage that this disease has caused to the avocado industry in South Florida, killing over 140,000 trees since the disease was introduced into the producing area, which was in 2012. So in a little bit more than 8 years, a lot of acreage have been lost due to this disease. Now, of course, it has caused horrible damage to natural areas. And in the southeast of the US, we have lost 1/3 of the red bays. That's more than 500 million trees dead due to this disease, and it's spreading really like wildfire. Unfortunately, it has reached incredible reservoirs of biodiversity like the Everglades National Park. And this is the map with the distribution of the disease. Really, this broad host range, even though is specific to the family so far that we know, some of these susceptible hosts have really broad ranges. Like look at the distribution of sassafras, goes all the way up to Canada. So there is a lot of area that this disease can affect. And it has so far. It was introduced into the US in 2002, and now it's present in 12 states. And it keeps moving north and west. So it's not really a matter of if, but when it's gonna arrive to larger avocado producing areas. Florida only represents 10% of the national production of avocado. So it's really on their way to more productive areas. And that's, if you like guacamole, you also should be worried about this disease. And very worrisome is the threat that it represents to the highly diverse family in the neotropics. So there's a lot of endemism and a lot of different species distributed in tropical America, and all of these are under threat. So for this reason, at the Tropical Research and Education Center, we are approaching the disease from different angles. My lab is leading the pathology aspect of it, and we are working on understanding pathogenicity, defense mechanisms, and management. Dr. Daniel Carrillo is leading the entomology group, and he is studying transmissibility of these beetles, how many of these beetles can effectively transmit the pathogen, and what's their role in the epidemics, the details of symbiosis and management of the beetle. Dr. Schaefer is leading the plant physiology part, and he is studying how the pathogen affects the subflow of in the plants, and how this graft rootstock combination influence this outcome. We have a strong extension program led by Dr. Crane and our extension agents, and we work really hand to hand with growers, and we have a strong educational program. And of course, we always care about the economics of the management, right? It's all about the economics, and that will, the grower is gonna decide which management practice to take, depends on the economics. So we have economists in the group as well. So can we manage laurel wilt? It's complicated. So it's complicated because Rafaela lauricola is highly virulent. So you don't need much inoculum to start an outbreak. The symptoms, that obvious external symptoms, are only seen once the vascular system is already damaged. And by this time, the pathogen has already extensively colonized the host and perhaps already went through the root graft to the adjacent trees. This is also difficult, the early detection, because by the time that you notice, the pathogen already spread. There's multiple susceptible hosts, including natural trees distributing natural areas. This means that there's constant introduction of the pathogen into plantings. There's two ways of spread, through the beetle and through root grafts. So you need two or more tools to address those two pathways. And so it's very difficult to manage. Now, one of the biggest challenges that I have faced is how to study this disease, because we need field trees. We need trees with a canopy that will have enough transpiration so our experiments can work, right? We're working with a vascular pathogen, so we need this transpiration. So it has been a challenge to get trees in the field. Growers don't want us to be inoculating their trees, and I completely understand that. But getting a potted tree that resembles a field tree is very difficult. The one that you see in the picture in the center up of the slide is 3 years old. So 3 years old to get a plant like this, and it's not even a mature tree, right? So that's a big challenge. However, we have tried different management techniques. I'm not gonna go into detail, just wanna let you know that so far, none of these have worked. We heard yesterday about stamping not working. It also does not work with this disease. Do not stamp the trees. You need to remove them. So the only thing that so far we can recommend is the complete and timely removal of the trees, right? And this will imply an early detection. And I already explained that early detection is difficult if we are only considering external symptoms. We will also recommend to remove the adjacent trees, but that's a recommendation that growers don't like because they appear healthy. So how can you tell a grower, I'm gonna remove your healthy trees? So it's not a popular recommendation, but that's what we have so far. My lab is pursuing 2 lines of research in terms of management. We are testing some fungicide applications in the way of drenches. And we are screening germplasm for resistance and tolerance. We have been having promising results in terms of drenching propiconazole, which is very effective as a fungistatic in vitro. And now we are trying to deliver the molecule into the trees, which is very difficult because these are hardwoods, very complicated vascular system. But we are, working on that to see if we can find a formulation that works. There are some escape trees that we have identified in the landscape. So these are trees that we confirm they are infected with the pathogen. However, they are remaining alive in the landscape, and not only alive, but productive. Only few of them, but it's giving us hope that there's some genetic component to this tolerance. And we believe that is based on their ability to compartmentalize the fungus. So as you can see in your left, you will see the trees that collapse. The fungus has been able to move laterally and colonizing all the new xylem, the xylem ring. Now, in the ones that we have seen, the trees that we have seen infected and that persist in the landscape, you will see symptoms like the ones that you see in the picture, with these only some vessels being colonized and showing these stylosis symptoms. So we think that there might be a genetic component to it. So we have partnered with the largest avocado germplasm collection in the world, which is located in Mexico. And they have sent us multiple accessions, and they will do so through the years. And we received the first 700 seeds. This is my greenhouse. And we just have to wait until they grow enough to inoculate them. We have a protocol to inoculate small plants. And we hope that by March, we will be able to screen some germplasms and hopefully find some tolerance or resistance. And with that, thank you to the funding agencies. Thank you guys for listening, and I'll take any questions if I have time.
Thank you. Enrico Bonanno, Ohio State University. So this disease has an ophiostoma component, very similar to oak wilt, very similar to Dutch elm disease. Do you do any trenching of the root systems to interrupt the root-to-root transfer? And also, how do you control the overland component of the spray? In other words, how do you control the insects?
So very good two questions. So the first one is about using trenching as a management. So because it's a transmitted root graft, and that's one of the worst ways that we have found the fungus moving through the orchard. And that's a very good idea if we were not in Florida. Florida has almost no soil, and we have limestone bedrock as a substrate. So it's very expensive to do trenching. We have done it experimentally, and it takes to really bring a big equipment to cut the stone, the rock, really. And so that's one of the reasons in Florida epidemics are really bad, because of the lack of soil makes the roots go more shallow and more laterally. So the chances of root graft below the ground are very high. But that's one of it would be recommended if it's feasible and cost effective. It would work, at least to cut that pathway. Bedrock. Yes, actually, yeah. I too bad I didn't include the video, but yes, I have seen it. It's pretty impressive the way they can do it. Our growers are very likely not gonna go for it. But I'm thinking that in the new plantings, the ones that people are replanting avocado, that would be a good idea to have the trees being independent, so to prevent the spread. But so far, yeah, they are not receptive to that kind of management. And about the insect, yes, the problem with this beetle is that it spends most of the time underneath the bark, right? More than other ambrosia beetles. So it mates everything with the siblings. So it really spends most of the life cycle within the tree. It does not eat the tree, it eats the fungus. So systemic insecticides are not going to work. But Daniel Carrillo, who leads the entomology part of the project, is testing some entomopathogens like Pauberia, which work once they have access to the beetle, right? In those really small times that the beetle flies to colonize another tree. I think he's also testing some insecticides, but we don't use insecticides as a management practice, only when we ask the grower to remove the tree, to chip it down, and then we recommend to spray the chips or to spray the adjacent trees in case these beetles are gonna leave that stump or that log that was removed before it dries with the sun and the fungus dies. So that's the only way we use insecticides for now.
Okay, so we should probably leave it there. Thanks again for a very interesting talk. And I would have had a question also, but I'll talk to you afterwards. So next up, we have Eva Franic, who is currently a postdoc at SLU in Sweden. And I believe she will present on her work that she's done at WSL previously on seed-borne fungi and their potential movement with internationally moved seeds. Some problem with that speaker. Over to you.
Thank you, Eki. So good morning. It's really nice to be here and to present the work that I did during my PhD at KABI, VSL, and University of Bern, and that I continued working on during my postdoc in Sweden. So it was mentioned on Tuesday and again today, plants for planting or live plants, which are the main introduction pathway for non-native pests and pathogens. They are because of that, because they are recognized as very risky, they are heavily regulated, and it's indeed not very easy to import live plants or big trees from other countries or continents. However, seeds, which are considered as the part of the same pathway, are considered to be very safe. For an example, there are more than 200 regulated organisms that are associated with plants for planting, and Fusarium cincinatum is the only seed-borne pathogen. And because of that, the only trees or seeds that are restricted for trade are pine and pseudotsuga seeds, and then there are some measures to reduce the likelihood of introducing Fusarium to Europe. All other seeds of all other trees can be imported to Europe more or less without any restrictions. And I need to do this. Yeah, and that is a bit worrying, or we at least thought it was a bit worrying, because we don't know which fungi are actually being traded in those seeds. So we decided to look at that. And as you can see in the table, we looked into 11 tree species that belong to angiosperms and gymnosperms, and that are native to one of 3 continents, North America, Europe, and Asia. These seeds were obtained from commercial seed suppliers, and we assessed fungi from all these seed lots, 100 seeds per each seed lot. We used plating and highly sensitive high throughput sequencing to look at what is in those seeds. So what we have found is that the infection rates were very high. We find 96% of these seed lots yielded fungi by plating. 100% of them had fungi as assessed with high throughput sequencing. Also, the fungal diversity was very high. We found almost 2,000, it was mentioned yesterday, ASVs, which are taxonomic units obtained by high throughput sequencing. And for comparison, only 6% of that diversity was obtained by plating. So for only 6% of them, we had live fungi. Also, fungal communities were differed between different origins, so continents or angiosperms and gymnosperms. And maybe a very interesting thing is that Asian seeds and angiosperm seeds carried highly diverse and unique fungal communities, which is something to remember in the case we will want to regulate this trade a bit more, which might be a good idea because we also found many fungal pathogens that were traded within those seeds. So after we did that, as I said, we saw these differences based on geography and taxonomic taxonomy of the host. So we wanted to look more closely into which factors are influencing these communities on the large scale. So we looked into similar tree species, again coming from Asia, Europe, and North America, but this time in botanical gardens in Europe and North America, so in their native and non-native range. And we were specifically interested in factors such as host identity, sites and site-specific variables, and distributional range, so not native or non-native range. So we know from previous studies that all tree-associated fungi are influenced by environment because most of them recruit these fungi from the environment. And that they are also host specific to a certain degree. Why distributional range is interesting? Because we know if we move trees from native to non-native range, they're normally moved only with portion of organisms that they have in their non-native range. So the results of this study show that host identity explains three times more variation in community composition than the site. You can see that in the first bar. So the green part is host and the blue part is site. When we decompose this site into site-specific variables, which you can see in the other graph, you see that the important variables are altitude and climatic variables. However, quite a big portion remains unexplained, which is probably because of other environmental factors that might be important, such as geographic distance between locations, for example. Number of congeneric tree species at the site was not significant, which is surprising because other trees should be the source of the inoculum to the air and then to the plant. But it might be that it would be better to look into total diversity of trees. And also range was not significant, which indicates somehow that these seeds bring part of the fungi with them, and then, or trees, and then they recruit some in the new environment. So this heat map shows the importance of this host identity in shaping these communities. So what you can see is on the vertical axis, this clustering corresponds to seedlots, and then the horizontal one corresponds to core fungal communities. So we excluded very common and very rare fungi from this. And as you can see, these first colors, so orange, red, and blue, green, and purple, they represent host genus. And now if you follow the clustering, you see that the first horizontal axis or this vertical clustering, so horizontal axis separates angiosperms on the top from gymnosperms at the bottom. And then second branching separates Fagus from Acer in the upper part, and in the down part, Larix from other gymnosperms. And now if you continue, you will see that they group by species, and only later they will group by continent. And this was a bit surprising because we didn't know that host identity is this important. It's also informative from the phytosanitary perspective because the importance of site suggests that we will still move things because there are different between sites. but the establishment might be limited by really availability of the host. The other thing that we got curious about is the fact that these communities are so host specific. So we thought that it would be good to explore more vertical transmission of these endophytes, because seed endophytes in trees are considered not to be vertically transmitted very often. But this level of host specificity was somehow suggesting, and also the question that phytosanitary services or institutions very often ask is, okay, you have high diversity of these fungi, but are they vertically transmitted? So in the study that I'm working on now at SLU, we are looking exactly into that. So we are working with 4 tree species, with Pinus sylvestris, Abies alba, Acer pseudoplatanus, and Fagus sylvatica. We obtained seeds from commercial seed suppliers again, 5 seed lots each. And we are looking into fungal communities in seeds and then in seedlings that are grown in sterile conditions. And what we want to do is to measure different traits of these seed fungi that are associated to nutrient utilization and interaction, and see if we can use these traits to explain fungal dominance in seeds and their transmission into the seedlings. And in the first study where we look into fungi in seeds, we were very curious about one particular thing, and that's how many seeds can be, how many fungi can be in one seed. Because so based on the work of George Newcomb and co-authors, what they think is that we always have 0 or 1 fungus per seed. And that is based on very large study that they did on many tree species and lots of plating on PDA. And they, in most cases, find 0 or 1 fungus per tree, which was very much what we also found in our studies. And based on that, they believe that this one dominant fungi in the seed is the winner of the exclusionary battles within seed. And that one fungi will be the primary symbiont, which will then determine the destiny of the seedling. So if it's a pathogen or a symbiont or something else. So now we wanted to check if this is really true, or it's due to methodological bias. Because if we put one seed on PDA, we are putting it on a very rich medium. So is it the fungi that is the fastest under these conditions that it will grow, but does that reflect really what's really going on? So we grew these seeds, we used whole seeds and subdivided seeds. And then we put 30 of them on each of the medium. So we used water agar, which is very poor medium, PDA, potato dextrose agar, which is rich in simple sugars, and then malt agar that is also rich in some other sugars and amino acids. And these 3, because they are most commonly used in the lab for this kind of work. And then seeds were used as whole and subdivided. And we were subdividing them because we thought that by subdividing, we will reduce these interactions. So if there are more than 1 seed per seed, per 1 fungus per seed, if we subdivide them, they will easily grow than if we have the whole seed. So the first results show that nutrient media has no effect on captured fungal diversity or community composition. The only differences we see here are between tree species. As you can see, numbers of morphotypes per seed can be very low in gymnosperm seeds, especially in pines. But then in Fagus, we on average get 2, which means that we can get many more. There are cases where we have even more than 2 per seed. And then we looked into subdivided seeds in comparison with whole seeds, but this is based only on pines and Abies, because halfway we realized that cutting Fagus seeds in very small pieces is extremely time consuming. So, however, so what you can see is that black bars are subdivided seeds and gray bars are whole seeds. And again, most of seeds yield 0 or 1 fungus, but as you can see, the black bar is higher than the gray bar for 2, 3, 4, or 5 seeds, which means really when we subdivide it, we get more morphotypes than when there are not. However, the real importance of this will be clear only when we measure these interactions fungus per fungus. Yeah, so the summary of this is that seeds that seem a bit riskier than we previously thought for trade, that we have this high importance of host identity, which needs to be further explored, and that for more accurate risk assessment in future, we need to look into many things from very basic, like the identification and distributions of fungi, to RNA-based characterization of active communities and others that are written here. So thank you very much.
Thanks very much, Eva. So we're a bit over time, but maybe one short question if there's one. Alain?
When hearing you about host identity importance, I was wondering if the process is not the same as for seed insects. I mean that most of the seed insects are highly host specific. And we attribute this as entomologists to the short window offered by the plant for the insects to attack and infest. And maybe it's the same process for fungi.
In the same study, we looked together with Alan into insects, and indeed, they're just very specific. You will not find the other ones. But with fungi, there are many of these environmental things, so we need to exclude most of the abundant ones. But it is possible, indeed, that the specialized ones have specific ways of entering. Well, they believe that they normally enter the seed from outside, so they get through while the seed is being formated. So it's coming from other organs around, or from the air, or via insects, but that's not totally clear. Yeah.
And then there are endophytes. But we're out of time. So, but thanks again, Eva. Okay, so next up we have Simone Prospero, who is a scientist with WSL in Switzerland in the phytopathology group. And he's going to talk about an extension continuation of this topic, but not about seeds, but dormant woody plants. Over to you, Simone.
Thank you, Eki. Hello, everyone. It's a new experience for me to give a talk in the session led by my boss, but I think this will be fine. First of all, I wanted to thank the organizer of this meeting. It's a pleasure to have a meeting in presence after 2 years of only Zoom meeting. So thank you very much. And as you can see in the title of my talk, there is a question mark. So the title is a question, but probably, as all of you know, probably we could even remove this question mark. We had very nice talks on Tuesday and this morning that, based on the interception data, showed that plants can really act as a vector of invasive pests. Maybe pests here, just to be clear, I refer both to fungi, to pathogens and insects. So both categories together. And here I want to show you, using a different approach, that the question mark is really not needed. I want to confirm what was shown already using interception data. How do I? Okay, maybe just shortly, the current situation, everyone knows it. In the last decades, as a consequence of international trade, we are facing an increasing introduction of exotic species. The saturation point, the plateau, seems not to be reached yet. So in the coming years, we have to expect more introductions. And we also have a second component that we don't have to forget is climate change. Climate change can result, for example, in the expansion of the range of native harmful organisms. These are native organisms. We also need to consider them. Or climate change can also result in the establishment of exotic species, for example, thermophilic species, which has a difficult time in getting established. With the new condition, they may be promoted. Or we are talking about pests and pathogen, so they both need a host to survive. So the host is also, we don't have to forget the host, the trees in our case, and the host may be stressed by this climate change, resulting in an increased susceptibility against this pest. And the current situation, we know the successful eradication, we just heard it about in the talk before Eva from Florida, that the successful eradication of established invasive harmful organisms is difficult and expensive. You can see in the two pictures here an example, sudden oak death, Phytophthora ramorum in Oregon, where all the infected trees have to be destroyed. The same approach for Asian longhorn beetle, Anoplophora glabripennis in Switzerland. And it will be more meaningful to prevent the introduction of this harmful organism. And for this, we have phytosanitary measures like the plant passport. We have border inspections. But there is a main problem here is that all these measures target known harmful organisms. And as all of us know, most invasive harmful organisms were not known to be harmful before introduction, or in some cases they were even not known to science before their introduction. And this is maybe Hymenoscyphus fraxineus, one of the last example in Europe, the causal agent of Ash dieback, which was officially described after its introduction to Europe when the fungus was already spreading, so quite late to apply some measure to contain it. So the question is pretty easy, simple. Is it possible to identify potential invasive harmful organisms before they leave their natural range, so before they become invasive in a new region? And we have set different approaches which have been developed in the last year going in this direction. For example, sentinel plantings or horizon scanning. And in this specific study, we applied this horizon scanning approach to asymptomatic dormant twigs. So asymptomatic means that apparently healthy, showing no symptoms of attack by insects or pathogens. And we used dormant twigs because living plants are frequently traded in winter. In winter, they don't have leaves, most of them. So we can already exclude the spread of leaf pests. And we assessed fungi and insects associated with these dormant twigs. And then we tried to determine which ones may be potentially invasive, in this case for Europe. But of course, you could do the same study for other continents. Maybe very shortly, material and method. We analyzed twigs of 167 tree species belonging to 14 different genera from 49 locations in 33 countries. You see, we have a pretty global sampling design with a strong focus as well to Europe. And just briefly for fungi, we did a DNA extraction, and then we used a metabarcoding approach. And then we used online this tool, this FunGuild, with which you can associate your, let's say, sequences to trophic groups. And in this case, we were, of course, interested in plant pathogens. And for insects, a bit different. The twigs were brought to the lab, and then we captured all the insects emerging for these twigs or present on the surface of these twigs. They were then grouped based on their morphology. So we made morphotypes, and then we sequenced a few specimens for each morphotype to confirm or to determine the species. To the main results, I start with fungi. We had in total 352 samples. Maybe here important to say what we consider as a sample. As a sample are 20 twigs, let's say 1 centimeter sector of 20 twigs. that were taken from 1 to 5 trees per species and location. So this is a sample. We identified 12,721 fungal ESV, what were previously OTUs. Now we use these exact sequence variants. And then we started this filtering activities, what I tried to show here using these different funnels. From these 12,000 and more, we had 754, so 5.9%, which were pathogen based on fungal. 118 were not present in our European samples. From these 118, we could identify 25. We could give a name to 25 of them, so known species. From these 25 species, 11 were never found in Europe. And from these 11, 10 were associated with woody species. So 0.08% of the overall ESV that we detected. Maybe a bit deeper in these results, you see in the first table on your left, the country of origin of the samples. And what is interesting here, that is all 10 samples from Russia, so from non-European part of Russia, from the Asian part of Russia, harbored plant pathogen not present in Europe. And samples from Tanzania, from Africa, harbored the highest number of plant pathogen per sample, not present in Europe, so 2.9. In the other table, the same data set broke down by genus. And here we see that Betula and Quercus are the two genera with the highest number of plant pathogen per sample, not present in Europe. Maybe I want to show you now 2 examples of these 10 plant pathogens that we detected. The first one is Pheropsis tumefaciens. The host in this study was Quercus robur from Tanzania. The known host completely different, so mainly especially citrus species, but avocado and ornamentals as well. The known distribution, you see here, several countries. In, I think, not only Egypt is in Africa. And that's me, no. And the damage, this pathogen induces the formation of galls and shoot proliferation on stem and branches, as you can see in these 2 pictures. The second one is Macrophoma sophoricola. The host in this study was Fraxinus manjuricus, an ash species from China. Known host, again, something very different, is the Japanese pagoda tree, Sophora japonica, that we also found in our cities. The known distribution is China. In this case, we have no information about the pathogenicity of this specific species, but we know that other species in the genus can be plant pathogen, like Macrophoma teicola, which causes branch cankers and twig diebacks on the tea plant, on Camellia sinensis. So now going over to the insects. Insects, we had 154 samples. In these samples, we found 4,751 insect specimens. About 3,000, so 64% of them were herbivorous. These 3,000 were grouped into 208 morphological species. 47 of these morphological species were not present in our European samples. We could give a name to 20 of them. 7 were never found in Europe, and 2 of the 7 were associated with woody plants. So we come down to 0.07% of the herbivores. And here maybe they are not very spectacular. Oh, no, first I have a table again before giving the names. So the same as before, you see in this table, the sample, in this case of eucalyptus and myrtus, showed the highest incidence of herbivorous insects not present in Europe. And the 2 genera are Pinus and Quercus, the 2 genera with the highest number of herbivorous insects per sample not present in Europe. So Quercus, again, as we already saw for fungi. So now the 2 names, the 2 not spectacular name. I'm not an entomologist, so I don't know them. But the first one is Pseudoxenetus regalis. It's a plant bug. The host in this study was Quercus macrocarpa from Georgia in the US. Known host Quercus, so this nothing special, and known distribution US. So everything fits to what we already know. And this insect feeds on leaves. I could not find information about the damage that it really causes on these trees. The second one is Omita centrolineata, is a bug, which we found on Podocarpus elatus from Australia. Known host is Rubus ulmifolius and other plant species from Australia. The known distribution, Australia and Tasmania. Here again, everything fits to what we know. And in this case, there is no damage. So this is why I said probably these two species are not so spectacular. And just summarizing our results, so we could find 10 plant pathogenic fungal species and 2 herbivorous insect species, not yet officially present in Europe. Most of them were found in association with 3 species that occur in Europe or that have closely related species in our continent. So in the case of an introduction, they would probably find suitable host. And of course, here we don't have to forget something. The climate, again, as I mentioned in the beginning, the successful establishment, at least of some of these, of the tropical species, would be probably limited by climate. And going over, that was me, huh? Going over to the conclusions. So by using a different approach than the interception data, we could really confirm that the international trade of asymptomatic plants can spread harmful organisms. In our case, we found both for insects and pathogen, 1 potentially invasive species out of 1,250 species detected. So the incidence is not so high. We need to screen a lot, but we can probably find them. So studies similar to these can actually identify potentially invasive pests and also new host pest association, which is very important. But of course, we need additional analysis. For example, we have to determine the host range, the life cycle, and pathogenicity before to apply some measure. And of course, we also have problems. I just mentioned 2 of them. The first problem is what we do with the many unknown species. So how can we regulate an organism without having an official name? And we saw most of them don't have a name. And then especially for fungi, for additional analysis, we need cultures. So sequences are not enough. So we have to isolate these pathogens from our plant material. And I think that's it. Thank you for your attention.
Thanks, Simone and Eva, for the very nice data and presentation. And Renee, of course. Yeah, Renee, very much involved. And so we're actually out of time, but just a very short question and very short answer, perhaps. I saw Quercus robur from Tanzania. Did I see this correctly? Can you explain?
Yeah, I had the same question from Renee, because Renee is the one who organized the data from Tanzania. And this European species was introduced by German colonizers, probably. Yeah. That's a very short answer.
It's still surprising that it even occurs there, but okay. So yeah, so we come to our last presentation. We're running slightly behind, but I don't know if we can do anything about extending the lunch break potentially. But so next up, we have Benno Augustinus, who's also from WSL, doing a postdoc with me and Valentin Culot, if you know him. And he's going to talk about optimizing surveillance for non-native species quarantine pests in Switzerland. Benno.
Thanks, Eki. Yeah, I wanted to tell you about a few things we found out in the last year that I've been working at WSL. I think some of them might actually be interesting for you. So Switzerland has joined the EU in the efforts to monitor priority quarantine pests. So that's a list of, pests that it's a bit of a who is who of damaging pests that we can still do something about its establishment. And the Federal Office of the Environment then asked us to also look at 2 quarantine species while we're at it. And they also funded my postdoc with these 2 main questions. This is just a small part of the whole project. There's many people involved, but I just wanted to know this as well. So the first one is, where is the probability of establishment of the model pest highest? And I will talk a lot about that more. And the second, what is an economically suitable sampling effort? So that's more weighing off the costs of modeling against the benefits of it. So against damages. Yeah, for that we use this bioeconomic model that is developed by Becky Pension-Neal. She's now a professor at the University of Maryland. Let's see. If we start at this hypothetical pest population that is influenced by the probability of establishment and the natural dispersal of the pest. The bigger this pest population is, the higher the probability of detection. And that one is also influenced by the probability of random detection. So that is basically someone from the general public who finds a funky beetle or a dead tree. And also the surveillance efforts. This is what we are going to do, and we want to find out how much we spent on that. And for that, we weigh it off with this optimization against this invasion cost. But don't worry, I'm only going to talk about probability of establishment because, you know, we are all hungry. So the focus of this talk is this one parameter, the establishment rate, which exists of the introduction and establishment, because you won't have establishment without introduction. I will also only show most data for ALB, Asian longhorn beetle, but it's similar for the other pests, the systems. So if we start with the introductions, we were looking at the relevant imports, because that's really the things that go to places where we are interested in, and then the number of inhabitants, because once it is in the country, it will be spread over the country. These goods are also spread over the country, and mostly to the places where there are people. For that, I collected data on wood packaging from customs data from 2010 to 2020. Those is a whole list of products that are associated with a lot of wood packaging material, and therefore also imports of these wood-boring insects. We get this data to the municipality level, which is quite important because many of these containers with the wares are only opened at the municipality level and are not actually screened at the borders. And that's also where they're going to be stored. So that's really information we need to make a better map, right? And then we only looked at countries that are sources of ALB, so the natural range and the invaded range. We have gross and net weights of these goods, of these products, which gives us a packaging weight, but everything under 200 kilo is very unlikely to be on a wooden pallet. So we excluded those data. It was much easier to get the number of inhabitants. We just took the census. And if you plot them against each other, you see a relatively nice correlation. But this is on a log scale, and you see that there's quite a lot of variation amongst the data. Yeah, and that is coming from someone who is trained in experimental ecology. And if we plot this out on maps, it's actually even clearer. So on the left hand, we see the inhabitants per district, and on the right hand, the wood packaging as described before in tons. And there are some regions there, in general, that's a bit the same, but some regions really stand out. For example, here in the east of Switzerland, we receive a lot of imports, but there is not such a high density of inhabitants. And then there's also other places that have very high densities of inhabitants here, for example, that hardly receive any wood packaging imports. So this is quite important to realize when you plot it out, when you want to get these risks. I think you should really look at both and not one at a time. The next step in the establishment rate is the establishment itself. For that, we use climatic suitability, which is Climax models that someone else developed for us. The urban trees, which I will, so that is climatic suitabilities. For the ecological suitability, so host availability, we're looking at the trees present. We're looking at urban trees, I will tell more about that later, and forest trees, the trees, which is data we got from the National Forest Inventory. As you might know, urban trees are the place to go, right? This most first detections of forest pests are in urban environments. And that was a reason for me to go and try to find a data set on urban trees. And there is not one data set for that. So I basically called every city in Switzerland and asked them whether they had a tree inventory and whether I can have it. I got some 33 useful data sets from that, combined them, and that was an interesting experience. They're all in different formats. They don't agree on coordinate systems because you really need 4 coordinate systems in Switzerland. And that's also, they also speak all different kinds of different languages. But then in the end, that was enough to determine the genus for every tree I got. And then I reduced that data set, threw out all bushes and shrubs because we're not terribly interested in those. And also invasive species, because some of these municipalities use these urban tree inventories to monitor where they have the invasive trees to fight them. And that makes your data set extremely skewed towards that. I also removed all genera that had less than 10 trees in Switzerland because I think they're just not relevant. But then we still ended up with a pretty decent data set with a small 500,000 of trees and 131 genera. And we were quite impressed by that because if we look at forest inventories in Switzerland, they are talking about 130 species. So yeah, that's quite a hyperdiverse data set to compare. And I looked at spatial autocorrelation of that data set because you would expect cities that are closer to each other to be more similar, and they weren't. But then looking at the language regions, as a small reminder, Switzerland has 4 official languages, but 3 of them are really widely spoken. Those are about Italian, German, French, and then there are some small areas that are French and German speaking, so bilingual. And if I compared those, I found quite some statistical differences. So there's very probably some cultural difference, climatic maybe, but yeah, we can discuss it later. So we were quite happy to find that because it means we had for every language region, we had a whole bunch of data sets, and we can just take the mean for that for other cities in those language regions. We then looked at whether there were actually hosts for our modeled pests in our tree data set, and for every modeled pest, we found hosts in every data set. So that is quite new. There were quite some suggestions to leave out big parts of Switzerland because there are none of the important forest host trees in the forests surrounding them. So yeah, that was quite spectacular. But yeah, you can really see that that these hosts are available everywhere. Now it won't be that surprising for ALB, but let's keep it consistent. What you see here is all the bars are the different data sets from the different cities, and the fraction of trees that are hosts to ALB, and you see that all have that. And it looks similar for many other pests. Yeah, that's were the 2 things that I basically wanted to tell you about. For one, the trade flows of these relevant products, and especially of the wood packaging material, and the number of inhabitants are correlated, but they're certainly not the same. And especially with wood-boring pests, you should look at where the most wood packaging material goes. The vast majority of wood packaging material in Switzerland in the last 10 years went to stone importers. And nicely anecdotal, Eki the other day came back from northern Italy where he looked at ALB's outbreaks, and they're all close to stone importers. So yeah, just saying. Also, the second point, I mean, you know this already, I'm preaching to the choir, but I still think this is another reason on the pile of evidence, is that urban tree composition is different from forest tree composition. and you should really look in these areas and not in the forests. And in practice, we have a lot of problem telling the foresters that have to do the monitorings for these pests that they shouldn't look in their forest, but in the towns that are close. And that's a huge knowledge gap still, because I don't know how it is in your country, but it's quite a lot of work to get these data sets on urban trees. And I haven't even started talking about private trees that people put in their gardens. Yeah. With that, I want to thank these wonderful people who deal with me on a daily basis, and you for your attention.
Yeah, thanks very much.
Hi, this is René Eschen from CABI. I liked your last conclusion, and I just wanted to add that it's also very important to make people who manage pests aware of the risk from urban areas. I know from Switzerland that people who deal with pests in non-forested areas don't really care about them because they don't see the risk of anything spreading to forests.
Thank you, Benno, for this nice presentation. I'm intrigued by one thing. How can you get the weight of the packaging material? Are there databases for the weight of wooden packaging material?
Thank you, Benno, for this very interesting talk. And, and I fully agree with your conclusion. And this is just to inform the audience that we have a new working party at IUFRO, Division 7, about trees in urban forests. And so you are invited to the first meeting next year in Lisbon. And I hope you can attend. Benno, thanks.
Excuse me, your mic isn't working.
But or use that one. Thanks. So yeah, so we've gone 10 minutes over. Can we start lunch 10 minutes later?
Yeah, so I wanted to make a short announcement about the agenda item at 2 o'clock, which will be renamed because business meeting sounds very boring, and in fact, these are really nice meetings. So since many people are leaving, we were thinking of renaming this to the pre-after party. And I would recommend that you're there because Eki will talk a little bit about IUFRO, and I assume that you will chair the meeting, but actually the nice and interesting part of that short session will be that we talk about potential future meetings. So opportunities to see each other again in different settings.
And actual future meetings also. Yes, several are already planned. Yes. Yeah, thanks, René. And so, yeah, 1 o'clock, be back on time. Sorry, the lunch break's a bit shorter, but yes. So, and thanks again for all the speakers.
Thank you. It's a pleasure for me to chair this session. But before we start with the first lecture, just let me thank the organizers for having this in-person meeting, the first one over the last years. And it's really wonderful to see you all and spend some time with you, not only in the lecture hall, but also in the beer halls, I must say so. So our first speaker today, we have 4 talks to begin with, one of them remote, and then we have a keynote as well towards the end of the session at 2:20. And in between, as René has already announced, we have the small UFRO non-business meeting talking about new locations. But let's do the science at first. And the first talk will be by Enrico, and he will talk, well, it's something more like a call for action, as I understand from your side. The global forest health crisis, a public good social dilemma in need of international collective action. So, yes.
All right. Thank you, everyone. So I'm going to be really out on a limb with this kind of talk because it really veers into social dimensions and social science, and I'm not a social scientist. So, and also this is either thought-provoking, like Sandy said, or massively obvious to everybody. So we'll see how that goes. So I want to start by saying that this is an effort. It's a large group of folks that came together to write a paper on this topic that we hope we'll be able to submit to Bioscience soon. We're still in the midst of it. I want to point out that the first name there, Jeff Williams, is really the driver, the motor behind this, this paper. And he just graduated from Purdue University. He was a graduate student with Matt Ginzel there, and is now with the Forest Service in Oregon. So we all are aware that we have major issues with invasive species. And this is just one example that's already been shown before. We always look at these exponential growth in introduction and establishment of different kinds of agents, whether pathogens or pests or insect pests. And this is just about pathogens, but you know, it's the same story over and over. And theoretically, we scientists have some pretty good ideas of what's going on and what we could do from a scientific perspective. And the answer really is sort of this convergent science, as NSF likes to call it these days, of human dimensions, machine dimensions, and natural systems. And so under human dimensions, you can think about individual and collective behaviors that interact with trade policies and markets across scales. So these are international as well as local. The machine component is coming into view now more and more, and has to do mostly with the application of sensors and artificial intelligence and intelligent systems to either detect or predict the advent and establishment of these agents. And then you have natural systems, of course. It's very important to understand ecosystems, the resilience of ecosystems, what are the drivers, and so on. So we, like in this group, we can talk to each other, we understand each other, and we pretty much have a pretty good idea of what needs to be done, or at least what needs to be learned to advance our oops, this didn't come out very well. All the colors are gone for some reason. But this is a typical invasion curve. This has already been introduced before. You have a pre-invasion stage, you have an introduction stage, establishment and spread, and it follows this exponential kind of curve. You know, superimposed on those stages in invasions, we put what are the sort of the normal or common management approaches. And unfortunately, these had arrows that cover different spans of that curve. They're all gone. But I think you can get an idea of when those particular management tools are coming to view, right? The course of time. Now, this chart, which was very ably put together by Jeff Williams, also shows the trade-offs. So risks, uncertainty, costs, the importance of stakeholders. And you can pretty much look at all these different trade-offs in the 4 different axes that we have on this diagram. What this has, well, all these things have in common is that it's a very reactive policy approach. We always respond to something happening, and we're unable to really predict and be prepared for the next invader. Simone talked about, for example, the pathogens that we don't even know exist, right? Anyway, I was thinking about Rumsfeld, of course, that said, we don't know what we don't know and all that kind of stuff. It was a different era. So a very reactive policy approach that is not working by and large, right? We have some success stories, but by and large, we keep failing. So the point of this presentation is that human activities have fundamental social dimensions to them, particularly as they relate to international commerce, that are quite separate from the strictly scientific and biological answers. The answer should be think globally and act locally. Instead, we keep acting quite selfishly at the level of each different country or even at more local levels within countries. So the goal should be to be a good actor locally to benefit all of humanity, because this is a global problem. It's a humanity problem, right? The social costs that come from invasions of forest environments are really borne by everybody. Special national interest conflict in a major way with its aspiration, which makes the forest health crisis what is called a wicked problem, an almost intractable problem, if you think about it. Again, I'm sort of thinking very high level here. I'm not thinking about individual specific local level solutions that can be implemented and can be successful in some cases. So what makes this a wicked problem? There's disagreement about the definition of the problem to start with. So there's competing dollars and other issues, human rights even, climate change issues, and so on. There is no stopping rule because global trade isn't going to stop ever. If anything, it's going to increase over time. The solutions are only incremental, so a little bit at a time. And so stakeholders can't really see a difference. It's the same as climate change, right? You're supposed to be doing these incremental changes that eventually will lead you to a better place. But because you don't see the short-term results, it's very difficult to implement these solutions. Sometimes implementing solutions can reveal new problems. And again, you have to deal with conflicts of interest among stakeholders. What works in one place will not work everywhere. So you have local, social, economic, geographical, political contexts that create unique challenges. And causation is intransitive. So you have embedded in feedback with other crises such as climate change. So everything becomes very, very difficult to deal with. So what we're positing here is that fundamentally this is a public good social dilemma, which is essentially a prisoner dilemma issue. So the example here is if you have two countries that trade with each other, the ideal situation would be one in which both of these countries implement strong biosecurity, right? But that would result in lower environmental and societal costs, but that almost certainly would result in higher net costs to export markets and domestic industry, because you have to impose those costs to be able to impose the biosecurity. It also implies mutual trust, which is one of the things that are most fundamental in human interactions, but it's almost always missing, especially at the level of exchanges between nations. So in reality, what happens is that we find ourselves down here. What is the economically optimal strategy? Both countries, each country thinks that they're doing what they're doing in their own self-interest, right? And the bottom line is that this leads to complete lack of mutual trust and respect. This is the business as usual situation, and it leads to these unsustainable costs to environment and society, which is what I'm trying to talk about today. So it's free trade agreements versus protecting forest resources. How do you deal with that in the absence of mutual trust between nations. And then you have the whole issues of cheaters, which is a big kind of worms that it's not important to open right now, but is a real issue. So that's where we are. And it can be defined as the tragedy of the commons, which is not unique to this situation. And it's very well understood in social science. So the solutions come through the policy cycle. And there's different, sort of 6 different recognized stages to the classic policy cycle. You have to have the formation of an issue is to identify the issue. Then you build sort of a policy agenda. You formulate the policy. Hopefully you adopt the policy. You implement the policy, and then you evaluate it sort of iteratively over time to see what works and what doesn't work, what's most costly, what's less costly, and so on. So the first 3 components there, some of the sub topics that could go under there are issues that relate with strengthening international biosecurity, applying integrated pest management, providing sustained and comprehensive research funding, and essentially provide a change in policy stance from a reactive paradigm of managing ongoing crises to a proactive approach designed to avoiding them. And again, this goes back to mutual trust between different entities and countries in particular. What happens is when you go through the rest of the policy agenda, then you run into really major obstacles. For example, things as mundane as the parliamentary nature in most democracies, you have fragmented powers. When you're dealing with parliaments or congresses, you have committees, subcommittees, subcommittees of subcommittees. You have the electoral cycle and localism. Most of the politicians are scientific amateurs. They don't understand what we're talking about by and large. Then you have political stalemate and partisanship. And then you also have statutory settings. So you have inconsistent and incompatible laws. Some things just don't make sense. One thing works against the other. And there's administrative overload very often as well, which all leads to sort of a paralysis of action in the policy arena. Now, in terms of short-term actions that are important to try and resolve this forest health paradigm, some of these are very, very well understood by all of us. We should really have more research funding, more interdisciplinary efforts, because it's clear that I'm a pathologist, I have to work with entomologists, and we both have to work with policy experts, social scientists, and politicians if you want to make any progress in this area. We think that we could really focus in a major way on preemptive breeding for resistance. So for example, if we have an idea of what could come into our country, and for example, by way of what Simone was talking about, by screening the plant material that he was talking about, if there are pathogens in there that are potential dangers to some of our target species, maybe we really need to start thinking about initiating efforts for breeding for resistance before we even get to the point, or at least putting the infrastructure in place so that we can respond much faster than we can now. We can target high-risk pathways strategically, for example, by way of using sentinel trees, and one of my students will be talking later remotely about our project on sentinel trees. Anyway, we can increase monitoring now for rapid detection sensing using sentinel trees, and then leveraging emergency funding, customs enforcement agencies, and so on. In the long term, though, that's another thing that we need to do to address public good social dilemmas, is to have the long view. And there are some key components that would form part of this long-term view. I will highlight a couple of them. One of them is that very often all our efforts are highly decentralized and discombobulated. And that also is part of the paralysis that we find ourselves in, both internationally, but also very locally, nationally and subnationally. Last year, we came out with an idea for the United States, for example, of something called the Centers for Forest Pest Control and Prevention, which is modeled on the CDCs in the United States, which handles all the human and animal diseases, zoonotic diseases. But really, if you follow sort of the thinking in this presentation, we need to incentivize global coalitions and collective action to prioritize forest health, to ensure adequate focus and sustained effort. So this is really a key point for the long-term solution to this problem. And this process has to be inclusive and stakeholder-driven. We cannot just talk again to each other. We have to involve the people that are on the ground, the ones that have interests and special interests. They have to be brought into the conversation so that we can make advances in this area. So the bottom line is the paradigm must be changed, and we scientists actually need to be part of the solution. So we need to become political or more political than we are. We tend to be very reluctant to do that, but it seems to me that this is the time where we need to speak up and speak out and start acting. So here's an example. There's a new bill in Congress in the United States called the Invasive Species Prevention and Forest Restoration Act. It's making its way through Congress, was presented by Representative Peter Welch of Vermont. And finally, it's gotten the endorsement of the Society of American Foresters. And that's their statement right there at the bottom, justifying their official support for this bill. But we can also do that as scientists. So here's an example of a letter that I wrote with Faith Campbell and a number of others back in December of last year to Representative Welch to say that we strongly supported the introduction of this bill and its passage. And I think the more politicians hear from people like us, the people who know what we're talking about, the experts, the more incentive they will have to actually, they will give them, strength and courage in trying to pursue these actions. So ultimately, we must decide in what kind of world we want to live. We either go right, as we are in many cases now, or we try to go left towards sustainable forest ecosystems with all that implies in terms of, as I said, collective action and cooperation. Before I close and I say thank you guys and ask for questions, I want to remind you that we have this new journal in Frontiers, the section of pests and pathogens and invasions, which I am a chief specialty editor. I encourage you to consider publishing here. It's becoming more and more interesting to see what kind of papers are getting submitted. And I also invite you to be editors, associate editors, because we're always looking for help. So with that, I thank you all, and I'm open for questions.
Thank you very much, Enrico. We may have time for one short question, if there's one from the audience. Any online questions so far? Okay, so thank you for that inspiring and encouraging presentation. We'll move on now to an insect we already heard about, a longhorn beetle, Monochamus galloproveniensis, and we'll have two presentations on that. Remember, it was just 2 days ago when Jean-Claude Grégoire gave his presentation on that, and we'll follow up on that now. The first presentation will be given by Pedro Nunes, and it's modeling dispersal trajectories of Monochamus galloproveniensis, insect vector of the invasive pine wood nematode, to optimize monitoring by trapping networks. So.
Thank you very much. First of all, I just want to say that I am quite glad to be here. I will be presenting a work that I developed with my colleagues, Manuel Branco, Ing van Elder, and Hervé Jactel. We already published this work, so I'll just be presenting what we found out and all that. And like already said, the work is is around modeling the monocamus and its dispersal trajectories across the heterogeneous landscape, forest landscape. So Jean-Claude already pretty much introduced the whole, most of it Tuesday, but basically monocamus is a native species to Europe, apparently not in all Europe, according to Jean-Claude's work. And why it is getting so much attention in the last years is because it is a vector to the pinewood nematode. The pinewood nematode, so is an American native species. It started to invade Asia and now invaded Europe, specifically Portugal in 1999. It invaded completely Portugal, and already there are some outbreaks in Spain which are currently being controlled. And the main thing is the fact that it causes the pine wilt disease, which is a very deadly disease that very quickly can lead to the tree's death, which obviously implies very high ecological and economic damage. And so that is why this whole thing represents such a great threat to all of Europe. And which is why since I think 2012, there have been monitoring trapping grids put in all the European Union countries. I think also Switzerland, if I'm not mistaken, they are also included. So it is obvious that it is very important to understand how does this vector spread across our landscape. There was already some studies in only pine landscape. So in this study, we wanted to go through a more heterogeneous landscape, which would be more realistic to most of the landscape out there. And for this, we used the tool least cost path analysis. I want to be brief, but basically least cost path analysis goes through the concept of in the landscape, there are some elements that provide resistance or provide incentive for the insect to go through there, and therefore the most direct path may not be the one that the insect actually uses, but may go around, just like in the example. So the main objectives of this work was to analyze the effect of landscape heterogeneity for the species dispersal. And the second one was to try to develop a method to locate the origin of captured insects. The point would be to utilize these trapping grids that are being spread in Europe and try to give them a second objective. So instead of just finding out if the insect has already been introduced, with this we would be able to find out where did it come from, and so help cut down the right trees and not have to just cut everything. So for this, we did a mark release recapture. For the people that don't know what this is, basically you accumulate a lot of insects. In this case, we marked 2,747 individuals. We marked them with paint or something, and then we released them all in one location, our release location. And in our case, we had 36 trapping grid around the whole area. And the traps were baited with attractants. So this was the area. It was in a forest place in Bordeaux. So as you can see, it is quite heterogeneous. Like the different colors are the different type of habitats, which I using satellite imagery characterized. And so for the first step of our work, we calculated the friction values. The friction values are associated with the least cost path analysis. So with friction, I'm referring to basically if the insects, how difficult is it to cross by these areas. So I use the word friction, but you could use any others like resistance, because this is very similar to electronic resistance, the Fury. But since I don't think any of us are physicians, I don't think I can, yeah. Anyway, so for this, we created a lot of different scenarios, testing for each different landscape, the different frictions. So a bunch of scenarios. I don't really remember how many it were, but it was a lot. And so with these different scenarios, then we used what was the highest correlation between the least cost paths that we got from each trap using the scenarios that I just mentioned and the recapture values that each trap had. And with these criteria, we got to what we assumed was the best way the best friction value for each land use. The second step was the reversal. So now that we have these friction values, we want to pretend that we forgot where we released the insects, like, I don't know, and try to do the opposite. So now we have the frictions of each landscape. We have the recapture values, and I want to find out again where did it come from. So I put here the equation, the barycenter equation, very simple, just without any weights. Now the Euclidean distance method, we call it that, in which we use only the weight of the recapture values. So basically the recapture values of the traps will give more or less weight to finding out the barycenter or center. And finally, the least cost path method, in which we use the friction values surrounding each trap to add a second weight to it. So the results from the so many insects, 68 were recaptured. These were the friction values from the scenario that we found to be best. As you can see, I didn't put here the 13 land use types, and I put the most important ones, the most representative. As we can see, the open areas, so lower number means less friction, so in theory, more favorable for the movements. So we can see that open areas seem to be the more favorable, pines as expected. Clear cuts had a very low value, which is actually quite interesting, and I'll talk about it a bit more in the conclusions. And finally, broadleaves, mixed forest, and young pines had very high values. And so then going to the idea of getting the barycenter. So in here we have the least cost path. Sorry, I actually so I should have put one above the other, but doesn't matter. So using only the Euclidean distance, so not using the friction values, we had a correlation between the paths, so the Euclidean paths and the recaptures of only 0.26, so it's quite low. Using the friction values, we get a 0.627 correlation, which is much better. The estimation accuracy means basically the distance between what we predicted and what we really released. So using the Euclidean distance, it was 31 meters, which was already very impressive. Using the least cost path methodology, it was improved by, well, technically 2 times, but and those are the 90% confidence ellipses. I forgot to mention the methodology, sorry, that we also then tried to test how it would go if we reduce the number of traps, because we might just be spending too much money with 36 traps. Maybe we could reduce that. So for this, I basically did the simulations reducing the number of traps. More details can be seen in the actual paper. But, so we can see that when it was reduced to 25 traps, it was still quite good. 16 was started to get a lot of noise, but it was still decent. 9 traps is kind of questionable, because even though 100 meters doesn't even seem that bad in a forest environment, like with 9 traps, chances are you might just miss the actual, because we are in this type of data, we are assuming that we managed to somehow put the traps, so the release is exactly in the center of our traps, which is obviously not realistic. But anyway, so with this work, we can conclude, well, we hope to be able to shed some new light about how the landscape composition and configuration does affect the dispersal of the beetles. So they are affected by the landscape. And one of the most important and interesting parts was that clear cuts did not greatly disturb its flight. This is quite interesting because a lot of the management done so far is mostly through clear cuts. And recently there were some papers done by Christelle, which was already pointing towards the idea that clear cuts are not really that good. And our work kind of helped, in a sense, confirm that, yeah, maybe clear cuts, so which already are such a big cost. Clear cuts, I didn't put a picture here, maybe I should have, but they cut such large areas and maybe it's not really doing that much because the insect has such a high flight capacity. Anyways, the non-area areas with broadleaves were also avoided, which gives an idea that maybe mixed forest could be quite a good way to limit the spread of these insects. And we were able to, in a sense, develop an innovative method to pinpoint the origin. Of course, this is quite limited and it was just, as you could say, like a prototype, but still. And finally, we hope to have been able to highlight how relevant and how good maybe for the future using both landscape ecology concepts and methodology can be to try to understand and predict pest invasion processes. Well, obviously I'd like to acknowledge both Victor and Fabrice for their work with the trap and release mark recapture. I'm sure it was a lot of work. I didn't happen to be there doing that part, but I'm sure it will be. And in the future, I will be doing it. So, and all the other ones are helping have funded. So, including the OMED project, which I am now still working with. And yeah, I think I said everything. Thank you very much.
Thank you, Pedro. Perfect timing. Thanks for that interesting presentation. I remember I did some capture and release experiments many years ago as a student as well. And I was very desperate at that time. So good luck with your future work then. Do we have questions from the audience? Yes, Eki, first one.
Yeah, so thanks, Pedro, for this very nice talk. And I'm just wondering, is there a possibility that the traps are more attractive in an open area than when you have a trap in the forest? Because there's competition from trees, potentially monochemists releasing pheromone, so that what you're seeing isn't only the least cost dispersal, but potentially also differential attractiveness of traps. In other words, is it possible that there might be more beetles going through the forest, but you don't catch them as often? So just a question.
Yeah. So we do know that these attractant, from what I know, it is actually quite effective when they do get in its range. But so I don't know if I completely understood, but you are saying that in open areas there might be so the ones that are passing by the forest may just not be caught, even though there's more passing through it, I guess. Maybe I completely misunderstood. I'm sorry. Well, I'll be honest. I have no idea. Like I can't. Yeah. Yeah. But there's also a lot of different things that you can discuss because the least cost path is not exactly perfect for this, in the sense that you could argue that in pine woods, even though the insect is incentivized to go by it, it doesn't actually, it may not speed up its movements, because in a favorable location it will be feeding and not actually zooming away. while in clear cuts, maybe why it seemed to have such a low least cost path value was because when he is in the clear cut, all he wants to do is get out of there because I mean, there's nothing there to do, not even to rest. So yeah, because sorry.
Yeah, Pedro, when you're doing the modeling, do you have to have an initial hypothesis on the friction values of each land cover? Or can you just feed the thing to the computer and it will figure out which is the easiest?
So, so no, even though we could have pretended, we did actually do a systematic method where I manually did it all. I actually did not use any special program. This was with Excel. Very. So what we did was a test for each land use, maximum value, medium average value. And then with that, seeing how the correlation would play out, we would start to mix. So basically a lot of different combinations.
Yeah. So in theory, you could automate that and just let
so
the
even
thing
though
run.
I knew that pine woods would probably have low value, I did try them with the maximum value. I tried I didn't try every single combination because some of them, well, yeah, but most type of combinations there were done. For the trapping, yeah, sorry.
Okay, thank you once again. Thank you. Thank you. Thank you very much. We need to move on. We now have a remote presentation from Brussels, from Jean-Claude's working group, I understand. Jean-Antoine, are you here?
Yes, I am here.
Okay, wonderful. So the stage is yours. You're going to talk about distribution, suitability models, and pest risk assessment of monojamos, the main pine wilt disease vector in Europe. Okay. Are there problems, Jean?
No, just 1 second, please. Yes. Yes, thank you for the floor. I will speak unto you concerning the distribution suitability model and pest risk assessment of Monocamus sp, the main pine white disease, the main pine white disease vectors. The pine wood nematode Burza phelancus xylophilus is responsible for the pine wilt disease, a major threat for pine trees globally. And the natural vectors of this disease are longhorn beetle of the genus Monocamus, a group of xylophagus insects distributed worldwide. And during this talk, I am focusing on a species, Monocamus galloprovincialis. The nematode is native from North America, where this nematode is still very active. Each yellow dot on this map represents a pine white outbreaks, and this disease causes also great damage in Asia since its arrival in Japan in the beginning of the 20th century. And despite preventive measure to limit the spread of the nematode, cases of pine wilt disease were confirmed in Europe in 1999 in Portugal and in 2008 in Spain. And however, except these two cases, the spread of this disease is still limited in Europe. We observe only 2 outbreaks in Europe since 1999. So why should we worry about this disease in Europe? because we have the correct mix of condition in Europe for this disease. First, we already have host and vector. The hosts are the different species of pine, and the vector it's different species of monocamus. And but we have also second, you need to know that the pine wild disease is symptomatic under particular ecoclimatic condition. And the risk for pale wine disease may be higher in the south of Europe because the disease requirements are met in this area. So ultimately, we have the right blend, host, vector, and particular ecoclimatic condition in Europe. And pine wild disease could result in important damage to the pine stands in Europe if the nematode successfully spread in the south of Europe. And I presented the general context of the monodis project, and this slide here is to present more precisely aims and objectives of this study I am going to present. First, the first aim is to predict the suitable area for monocamus in Europe with species distribution models. But we would like also to evaluate the effect of spatial scale, the grain on SDM results. And for this work, and a trap network has been deployed in Europe to evaluate the presence, absence, and the abundance of monocamus vector. And I show in this slide an example of trap used in Spain. And based on this trap network, based on this trap network, I show you here a map map that represents the distribution of trap in Europe. On the left, I show the positive trap for the presence of Monocamus gallo-provincialis. And on the left, I show the negative traps for the presence of this vector. And this data set represents a strong sampling effort, and 4,000 traps from 36 contributors and in 29 countries were used for this study. And first, to analyze this data, we used the species distribution model and boost regression trees. Boost regression trees model were used to generate a predictive map for the presence or absence of monocamus in Europe. And boost regression tree use a machine learning algorithm, decision tree, and boosting method to generate a prediction for the presence of monocamus. And we used climatic data and data on the forest distribution are tested in this study as a potential determinants of monocamus distribution. This map here, on this slide represents the predicted probability of monocamus detection, and the green area shows a suitable area for this vector. And this prediction is mainly based on climatic variable as the annual mean temperature, the mean diurnal range, and the temperature annual range. And this prediction showed that a wide area of Europe is suitable for Monocamus gallo-provincialis. But the second objective of this study was to evaluate also the effect of spatial scale, the grain on SDM results. And to test this effect, multi-scale analyses based on quadra areas of different size were used, and this first model based on boost regression tree was repeated over 26 levels. And for example, in this slide, I show four levels based on spatial units of various size. And for this second analysis, we use Poisson's GLM instead of boost regression tree, and we try to explain the proportion of positive trap inside each quadrat based on a subsample of predictor variable. And we test also a control variable, the total number of trap inside each quadrat as a predictor variable to test and control some sampling effect on your results. And in this slide, I show you first result. In this graphic, the y-axis represent goodness of fit measurement, and the x-axis represents the 26 level of analysis based on the analysis unit of different the 26 levels of analysis. And the first result, we found that the models become more accurate as the size of quadra increased. And here I show the absolute contribution on the left and the relative contribution on the right of each predictor variable on the model prediction. And here I ask you to focus on the right graphic that shows the contribution of the predictor variable we used independently of the spatial scale. And what's important here is that the mean diurnal range is determinant to explain the distribution of Monocamus galloprovincialis regardless the spatial scale. But another important result is that the analysis show signs of sampling bias as the quadra size increased. Indeed, the contribution of the control variable in your model is very low in the model compute with small quadrats, and increase as the size of quadrats increase. And in conclusion, we have three main results in this analysis. First, the model become more accurate as the size of quadrats increase. And we have two hypotheses to explain this first result. The local level determinants of Monocamus distribution are very difficult to treat, to capture. And the basic resolution of raster images will not always be good for the analysis. A second important result is that your analysis shows signs of a sampling bias as the quadra size increase. And we can explain this result because we are sampling in area where Monocamus species are known to be present, and the number of traps is positively correlated with the proportion of positive traps. Or the forest Monocamus species management is country specific, and we have some effect of that on very large scale. And the third main result of this analysis, the mean diurnal range is determinant. The climatic conditions are determinant regardless the spatial scale. And climatic conditions are crucial to understand Monocamus species distribution. And that's it. Thank you for your attention.
Thank you very much, Jean. We have time for questions from the audience or online. Any questions at this time? Any online questions? No. Then oh, here's one. Eki, I'll just hand you my microphone.
So do I understand correctly? Oh, of course you're online. That was a good joke. Huh? Did you only look at Monocamus gallo-provincialis? And what about the other two Monocamus species that are common in Europe? They could be equally effective as a vector, presumably?
No, currently we did this analysis only for Monocamus gallo-provincialis because it's because we have a huge data for this species, and the number of observation for the other species are not so important. And but we could do this analysis for the other species, but to use this kind of analysis on different spatial scale, and to use machine learning model, we need an important data set. And currently we have only one species. We have data to do this analysis only on this species. So we could do that for other species, but currently I don't know if will be a good idea.
Thank you very much, Jean. Do we have more questions from the audience? No, that's not the case. So thanks again, Jean.
Thank you.
And we'll move on to yet another presentation from the University of Minnesota. Seems to be a huge group here, and that's great, I think. Many students. This is Dora Mangola. Okay, thank you very much. And on emerald ash borer again, preservation of urban canopies of ash trees using insecticide strategies to induce associational protection in Minnesota.
Hi, everyone. Thank you for the invite to be here. So today I'll be talking to you about associational protection in insecticide-treated ash trees in Minnesota, USA. And it's in relation to the management of emerald ash borer, this very beautiful but destructive insect. So emerald ash borer, or EAB, as I'll be referring to it for the rest of my talk, is an invasive wood-boring insect that was introduced to Europe and North America. In North America, it was introduced in 2002. It was first detected in 2002 in Canton, Michigan. And since then, it has spread to 35 American states and 5 Canadian provinces. And this is just the most recent map as of January showing where those areas are. So emerald ash borer feeds on ash trees in the Fraxinus genus. It attacks them and it'll feed during 2 stages of its life. During the adult stage, it'll emerge from underneath the bark, and then it'll feed on the foliage, and then it'll mate. And this is known as maturation feeding. And then secondly, during its larval stage, it'll feed underneath the bark, and it'll create these S-shaped galleries that I have a picture of up here. And this is the feeding that results in death of the tree because it disrupts the movement of nutrients and water up the tree. And we can, as a result of this, we can see crown thinning from the top of the tree downwards, which I have a picture of at the end here. So there are a number of different ways in which EAB is managed. The first approach is to sit back, do nothing, and let nature take its course. But another way of managing it is educating the public on the dangers of moving firewood. A third method is quarantine of infested areas, then the release of biological control agents, removal and replacement of dying or dead ash trees, and lastly, systemic insecticide treatments, which is the focus of my talk. And these insecticides kill the larvae feeding underneath the bark and also the adults when they emerge and feed on the foliage. So the hypothesis of our study is that treating a subset of ash trees in an urban environment can confer benefits to untreated trees. And we're referring to this as associational protection. So you might wonder why I have a picture of a cow and the flu shot. It's just a reminder, if in your countries you get flu vaccinations at around this time, you should do it. This so our study is kind of in relation to this, because if you have a population, for example, in this room, if many of us get the flu vaccination, then those of us who don't have the flu vaccination can still be protected because there's reduced spread of the virus in the room. So for our study, what we're hypothesizing is if we treat a subset of trees, that can lower the population of EAB in the area, and that will allow untreated trees to survive for a longer time. So in order to study this, we conducted our study in Minnesota, as you've seen many of us from there. EAB was first detected in Minnesota in 2009 in St. Paul. And just to give you some brief information on Minnesota, it's home to 10,000 lakes, really long and cold winters, in my opinion, and and maybe other people's opinions if you've been there. And it's also home to, what's important to our study, 1 billion ash trees. And most cities in Minnesota, or a lot of the cities in Minnesota, have up to 60% of their urban tree population consisting of ash trees. So it's a very important tree in the urban environment. And I will discuss how we set up our studies. So we had 12 sites. 8 of our sites we treated with the current gold standard treatment, which is emamectin benzoate. So this is what most treatment companies are using to treat against emerald ash borer. And then 4 of our sites have azadirachtin as the insecticide that we're using to treat. And this is a product that's similar to something that was very popular in Canada, and it's considered an organic compound because it's extracted from the neem tree. And most of our sites are around, as you can see here in the map, southeast Minnesota and the east central area of Minnesota. So I just wanted to zoom in to those sites. So I've said that we have 12 sites, but this will be important in the results section. For some of our sites, they are in the same city. So we have 8 cities with 12 different sites. And in some of those cities, we have an emamectin benzoate site and an azadirachtin site. So for each tree in our study, we started this project in the summer of 2017. We went out and we measured the diameter breast height of the trees, and this allows us to know how much insecticide to inject into the tree. And then we tag all of our trees with round silver tags, and we have been monitoring crown health, so looking at that crown thinning and reduction to see how the tree is doing over time. And then in order to treat the tree, what we do is we get to drill holes at the base of the tree, and then we use a pressurized injection system to apply the insecticide into the tree. And we've been continuing this treatment since 2017. So for emamectin benzoate, we treat every other year, and for azadirachtin, we treat it every year. We treat the trees every year. So I've mentioned this crown rating system that we're using to monitor tree health. So we use this method adapted from Smithley and colleagues. I have 3 different images up here showing how we're doing our ratings. So our rating of 0 is basically a tree that's dead and has no crown present. A rating of 5 is 50% of the crown is present. And a rating of 10 is a happy, healthy ash tree, and it has its full crown present. So this is a schematic of how we set up the studies at the actual sites. So we have a couple of streets with ash trees present. At the start of our study, we tried to start with trees that had a rating of 7 and above. So 70% of their crown was present, and that's relatively healthy. So what we did is we started one end of the site, treated a couple of trees, then we increased the density of treated trees as we moved along. And we're doing this so that we can see whether a tree on the densely treated area gets protection while one in the sparsely treated area does not, or whether even the one in the sparsely treated area can get some sort of protection. And this is important because it could help homeowners or even city planners to save on costs for treatment. And also it could reduce the amount of insecticide present on the landscape. So how we approached analyzing this data is we looked at the effect of the proportion of treated trees within certain distances on the crown rating. So we've been going back every year since 2017, and the data I'll be presenting today is our 2020 data. And the different distances that we used were 10 meters, 25 meters, 50, and 100 meters. So just to give you a better view of what that looks like on our site, I have it overlaid on our site. So we have our tree of interest circled here in red, and we look at what proportion of trees around it are treated. So is it a third or a fourth within those different distances that we had? And this could give us an idea of whether we'll see protection like at those different distances, 10 meters or 25 or 50. And eventually in the long run, what we're hoping to show homeowners or treatment companies is what they need to treat within the area that they have their ash trees. So for 2020, we wanted to look at, does this associational protection occur? So this is a table showing the results. We have 2 different insecticides in our different cities. So as I mentioned earlier, some of the cities have both an emamectin benzoate site and an azadirachtin site. And then at the top, I have the different distances. And then we have color-coded boxes at the underneath that show either if it's no associational protection is present in the white boxes, and blue boxes show associational protection was present. So as you can see, there are a lot of white boxes, and I'll get into why that is. But what I would like to bring your attention to is that we were able to detect this beneficial effect on crown rating from treating a subset of trees in at least 4 of our sites. And most of those sites were in the emamectin benzoate treated sites, and it was at this 100-meter distance. So diving in a little bit to our results, I want to discuss these 2 results that I mentioned. So we saw associational protection at 4 of 12 of our sites. So maybe you're asking yourself, why is that? And then also I wanted to discuss this 100-meter distance. What could be the reason for this? So 4 out of 12 sites might seem like, oh, well, maybe it's not working that well. Maybe most of the sites are not getting this, we're not detecting this associational protection. But the reason for this is because at a lot of our sites, the trees are looking pretty healthy. So I said that most of our trees had a rating of 7 and above when we started, or 70% crown present. And as you can see on the table that I have here of the percentages of trees that still have that crown level and above, it's at least 96%. So the trees are looking pretty good. And just to illustrate that further, I included a picture of one of my sites, or a street from one of my sites. And as you can see, just looking at those trees, you can't tell which trees are treated or untreated just based off of looking at the crown. So the reason this could be is that the EAB density in our sites is probably not as high as we had anticipated it to be, or it's not high. And the potential reasons for this could be that the associational protection range is wider than we think. So although I'm looking at distances of 10 meters, 20 meters, and so forth, it could be that it's covering a wider area of my study site. And it's not that it's not working, it could be working too well, which would be great, especially for homeowners and city planners. But we need to study that, investigate that further. A second reason for this could be that is high, or there happens to be level winter mortality. So as I mentioned earlier, Minnesota gets pretty cold. The winters can get pretty cold. So there's a forest health report from the Minnesota Department of Agriculture that showed 40 to 90% level winter mortality in some areas in Minnesota. Secondly, looking at that 100-meter distance, there's a 2009 paper, and Sandy is one of the co-authors actually, and they are looking at dispersal of EAB in newly colonized sites. These were areas with ample ash trees present, and they noticed that most of the larvae that they found underneath the bark were within 100 meters of where the adults emerged. And we're thinking that we'll see this associational protection at 100 meters because it has something to do with EAB dispersal in the area. And we're looking forward to what this year's data has to offer. It's my final year of my PhD. I'm very excited that I got to come to Prague. And I am excited to have talked to all of you. And hopefully in the coming months, you'll hear the final results of my study. And with that, I'd like to thank all the collaborators, all my lab mates for their support, and all of you for listening, and the funding bodies. Thank you.
Thank you, Dora. We have time for questions now. Any questions? Just a second, wait. Just wait for the microphone.
Blanca Kalinova, this university.
Mhm.
So I have a question about the injection techniques. Can you tell me more details about how you injected the insecticides into the tree? Was the number of injection dependent on the tree size? And so did you inject in different areas, or whether only one hole was enough? So these small details that are important always.
Okay. So for our different treatments, what we do is we drill holes at the base of the tree, like near the roots. And then for every 2.5 centimeters, you use 0.2 grams of insecticide. So we'll drill a couple of holes and we determine that from the diameter breast height of the tree. And then we'll use it's like a hammer to put in some plugs into it that will allow you to put the injection or the applicator into the tree. And then after that, we'll use that pressurized injection system that puts in the insecticide into the tree. Yes, it'll take it up, translocate. Yeah. Yeah. Or 0.2 grams for every 2.5 centimeters. 0.2 grams, yeah.
Thank you. Do we have any online questions at this time? No. Any more questions from the audience? Nothing? Then thanks again for this inspiring presentation. And also for the valuable appeal for more vaccination. That was very helpful. So now I will reluctantly hand over the microphone for the non-business meeting to Eki. You have 10 minutes to be on time.
Reluctantly. Thanks, Andreas. So could I have the presentation? So yeah, business meeting is kind of a boring name, and it's actually not only anything to do with business. It's really for everyone, because we want you all to participate as much as possible with upcoming meetings. And we also want to encourage you to perhaps consider hosting or co-hosting future meetings. That's why it's very important that you're here. But just starting out, I'll just briefly run you through a few things about IUFRO. What IUFRO is, the International Union of Forest Research Organizations. And it's not actually that well known. We often have people at conferences who are not so familiar with what that actually is, because they may have only seen a few meetings, but it's actually a rather big organization. And it's kind of a grassroots organization, and also it's global. And you can see the subtitle there, the Global Network for Forest Science Cooperation. And so there are about 15,000 or possibly more scientists that are part of the network, and most actually not through individual membership, as you may be familiar with in normal societies and so on, but rather most are actually a member through their employers or universities where they are working. So most of the members actually are the organizations as such, so forest research organizations like this university here, I believe. And so they are in more than 100 countries and more than 600 member organizations. The headquarters and the secretariat is in Vienna. And most of these people you may not see, but there is actually quite a few of them. There's a director and about 10 people or so who keep this running. But yeah, so we also have these small meetings which are happening mostly as a sort of individual effort or people working within the working parties. So in terms of the organization, there's like a number of elements. There are task forces, there are like various committees and so on. But most of the activity happens within these divisions, and there are 9 of them. And Division 7, Forest Health, is the one that most of us working in forest insects or pathogens are involved with and attend meetings of, like this one here. And so there's also involvement of a working party within the Forest Environment Division, which is about biodiversity and all kinds of other environmental things. And it also includes a working party on plant invasions, which is in Division 8. And so Division 7, we have this kind of organizational structure, and I'm the coordinator of Division 7 and have been for 10 years. And actually, at the moment, like yesterday, today, and tomorrow, we had the board meeting. And as of Friday evening, I'm no longer the coordinator because I've done it for 10 years, and I'm happy to pass it on. And Todd Ramsfield, who is one of the current deputies, is going to take over. So from Friday evening onwards, Todd will be responsible and taking care of things, and I'll relax and sit back. And so this will be my last kind of official activity here anyway, but I should save time and not talk too much. Okay, so there are 2 research groups within Division 7. There's pathology, and Todd, who is one of the current deputies, is also the coordinator of the pathology research group. And most of you pathologists will have probably heard from him, received his newsletters and so on. And there are these working parties. So this is kind of where most of the action happens. And then the equivalent thing in entomology, which is research group 703. Marci Klapwick is the coordinator, and Hervé Jactel, who's here in the audience, is a deputy, and Barbara Benz, who's not here. And so there are 2 working parties. Firstly, alien invasive species and international trade. And Renee Eschen is the coordinator of that. He probably spoke about that on Tuesday morning, I think. And then the other one is population dynamics of forest insects. And so we have some online participation from them. And so just a few other things about IUFRO. So in the past, like in the late 80s and 90s, we had less than 5% of the office holders, so coordinators or deputies who were female. So it was a very much male-dominated sort of group. But that's improved a lot over the years. This is actually one of the things we try to achieve, to have more equitable representation of gender, but also geographic representation. But so I'm happy to say that we're almost at 40% now of female coordinators or deputies, which is fantastic. And yesterday at the board meeting, it was also approved that from 2024 onwards, the next board term after the Congress in Stockholm, we can have dual coordinatorship where you can have a woman and a man co-chairing a division. And so we may actually have co-chairs from 2024 onwards. And actually a few of us are working on a paper on these kind of gender issues in forest entomology and pathology, which we hope to submit perhaps before the end of the year, where we talk about all kinds of gender issues in forest entomology and pathology. And that's an effort that's led by Jess Hartson in the US. And so we organize mostly meetings and interact at these meetings. So here's some photos from 2019 when we actually had the last in-person meetings with field trips and everything. This is another one that was in Italy also in 2019. And you'll probably recognize a few people there like Alberto Santini, and there's my colleague Danny Wriggling and Mike Wingfield in the bottom. This was another meeting. There's Milos in the foreground. This was in Romania. Anyway, then we had a break of meetings, no meetings but we did have some online activities. Anyway, upcoming meetings. So Todd and Marge and Jeremy Allison and I, we've been planning some webinars. There've already been a bunch, and there are more coming up. You see on the 13th of October. The first one, these will be online, obviously, webinars. There will be more information about web links and so on coming up, and more details in newsletters and on the IUFRO website. Then the Pine Mill Disease Conference was postponed and postponed again, and now it's going to happen online in November. Then actually planned as an in-person attendance meeting, we have the Phytophthora meeting in June 2022 in California. Then a meeting of the Working Party on Foliage, Shoot and Stem Diseases in New Hampshire in June. And then the big event in September 2022 in Lisbon, Portugal. We have an all-division entomology and pathology meeting. We've received close to 20 session proposals, including the one on urban biosecurity issues that Hervé mentioned. And yeah, all kinds of topics. You're all invited to come. In fact, we hope that we get lots of participants because we have to fork out a lot just to secure the meeting venue. And so you're most welcome to register early to help with that. The registration will be online shortly, so we're just about ready to open that up. And so we hope for good participation, so we don't make a loss. And it should be a great opportunity. And Lisbon in September should also be a nice place. There will be a field tour at the following weekend afterwards, also where we can mingle more and so on. And then also just after that, you can actually travel by tour bus to the next meeting. Then this will be just north of Portugal in Pontevedra, genetics of tree parasite interactions. And lastly, we don't have a membership list through which we communicate. So if you want to find out things and stay in touch, you should subscribe to our list server, email lists, ForPath and Forend. You can find those, for example, by Googling Forend and IUFRO, then you should find it. That's where we distribute a lot of information about upcoming conferences, also job postings and all sorts of things that are relevant for us. And so if you subscribe, then you'll get this information and you can get in touch with us and so on, or otherwise proactively look on the website, but it's a bit of a rabbit hole, so you may not be able to find what you're looking for. I understand there's a bit of a problem at the moment with Forend and ForPath because they've just switched to a new server a few days ago. So if it doesn't work right now, just send us an email, maybe you'll find us online, but it should be all up and running again properly in a few days. And so with that, I'll hand over to René, who's going to, no? So the next point was going to be that we talk about the next meetings of international trade and alien invasive species. The next possible meeting, which I understand isn't planned yet. So we're looking for, again on time, fantastic. So yeah, René is looking for volunteers and inspiration for where we can meet next. And I guess the same applies for the Population Dynamics Working Party and possibly, I'm not sure about the Division 8 people, they don't seem to have that many meetings.
All right, to start with the last bit. So during my presentation on Tuesday, I already indicated that the Working Party on Ecology of Invasive Plants, if that's what it's called, they are planning a meeting. I have no details, but I assume that they would be interested in having joint meetings with other working parties. Since my working party has been having meetings about every 2 or 3 years, I think, I don't think that we're looking immediately for planning new meetings, but again, I suppose for all these working parties, what we're looking for is possibilities to have joint meetings that people can interact also with similarly minded or differently minded people and learning about other fields. And other than that, I can only invite suggestions for meetings. And if you're willing to host a meeting, or if you have a suggestion for having a joint meeting, please let me know.
So yeah, thanks very much, René. And I could just say that if anyone already feels a spontaneous urge to organize or co-organize a meeting, feel free to talk to René or me. about this, and so you're really most welcome. We can give you a hand with some of the organizational questions you may have and so on. You can also think about co-hosting this with your local or national entomological or pathology society and so on. So, and I organized my first IUFRO meeting quite a few years ago, and it was a bit of work, but it was also a lot of fun, and it all worked out well, and it was fantastic for meeting new people and so on. So yeah, so let us know if anyone is considering it or has some ideas, maybe with others together and so on. So yeah, so with that, I guess I should hand back to Andreas. And or are there any questions or comments? You can also talk to us afterwards, of course. Yeah, but yeah, of course. Yeah. Thanks.
Thank you. Thank you, Eki, and René for keeping the time. We have one remote speaker now, but before we do this, I just want to let you know, Eki, there's a comment from Mike Winfield saying thank you for leading the division. You see that up there? So it's in the right corner. So he's sending best regards and thanks to you as well. So, yeah. So now we should have a remote session from Florida. Yuji, are you there?
Hello, everyone. Good morning.
Good morning. You just finished your breakfast, I guess.
Not yet.
Oh, but will you be able to give a talk now? Or?
Very happy. My understanding is that it's still a couple of minutes away, and so if people want to move around, get up and stretch their legs, I think that would be very appropriate at this moment.
Yeah. No. Oh, Sandy is right. Are you okay starting now? Because Sandy said if we allow people to leave the lecture hall, they will not come back in time. So
Sandy is always right. So I will have to do that.
Okay, so we're very happy to have you here, though it's remote, and you will talk about invasive bark beetles that are going to eat up our forests, very threatening. So just go ahead, Yuji.
Thank you. Good morning, everyone. Good afternoon, I should say. And thank you, Sandy, for inviting me for this talk. I am so sad I'm not there with you. You know, this was all supposed to be very different. I was supposed to be in Prague with you. This was supposed to be last year. And if not last year, then this year. And I just finished the sabbatical year in Prague. And I left just before this fantastic meeting has begun. So sorry about that. Have some people on me, and I'll see you in person next time. But right now, we'll talk about bark beetles, invasive bark beetles. I assume that everybody can see the screen, right? Well, hearing no opposition, perhaps. So I am talking on behalf of the University of Florida Forest Entomology Lab, which is my team. And we study a lot of things that happen on the bark. Primarily, we study the ambrosia beetles.
Yuji, one remark from my side. Could you just put it in presentation mode so that we can see it larger?
Ooh, absolutely. I believe this is the right one.
Perfect. Thank you.
Okay. Right. Okay, well, welcome again. Good morning. And so I was talking about my group, the University of Florida Forest Entomology Lab. We study lots of beetles, mostly the ambrosia beetles that you probably know about. They live in the trees. They harbor symbiotic fungi inside their mycangia fungus pockets. The fungi grow through the wood, aggregate nutrients, and feed the beetle larvae. And we study the interactions between these organisms and also the interactions with the ambient fungi. And it's all great. But what we sort of, our perspective is this perspective of metasymbiosis. So symbiosis of symbioses, many of them together interacting within those dead trees. But what we have to remember too is that we need to study the effect of these organisms on the tree. The poor tree colonized by these things, almost all of them, all of those trees, in almost all cases, already dead. We do this in Florida. This is, of course, the peninsula of Florida. And somewhere in here, in the midst of the greenery, is my university, University of Florida. Most of you know this place, perhaps because of the fruits and vegetables that we produce. In fact, agriculture is one of the strongest money makers in the state of Florida. You know, many of you may know it for its amusement parks and beaches and such, but agriculture is really big. If you zoom in on the map, you see thousands of little squares like this. Those are all orchards, mostly fruit and vegetable orchards. And there are really a lot. We span the tropical and subtropical zones, and there's a lot of different fruits and vegetables grown here. But do you know what the biggest moneymaker is in agriculture in Florida? It's not bananas, it's not oranges, it's actually pine. Pine revenue in Florida exceeds $25 billion a year. This is more than all the fruits and vegetables combined. And few people know that. What is the biggest problem, though, for both pine and the rest of Florida's agriculture? It is invasive species. And this is where forest entomologists like you and I come into the picture. Why is it a big problem? I will illustrate that in the case of the citrus industry. These are oranges. This is the quintessential produce of Florida. Fantastic millions of not millions, but thousands of hectares of citrus orchards. Well, the industry has been doing really well. It was one of the leading industries in the state with over $1 billion in revenue annually. But now it's not doing so well. And many of you may have heard of this. This is what the orchards look like this time. What happened? An invasive insect showed up. It's an insect. Well, the insect is not the most critical thing. It's the vector of a bacterium that lives inside the tree tissues and that it destroys the metabolism of the citrus plants. It's called citrus greening. It's essentially a tree health issue that is devastating the industry. But I'll tell you one other story. Maybe you've heard already about this. I'm sure you did. Somebody has talked about the invasive species invasion. I'm sorry, ambrosia species invasion. This is approximately what happened in about 2002, about 20 years ago in Georgia savannah. One little beetle flew out of some cargo that came from Asia. That beetle happened to be the red bay ambrosia beetle, and it carried a load of its symbiotic fungus. And we did not know that before, but now we do know that this combination of a beetle and a fungus caused laurel wilt, a disease that quickly swept through the landscape and totally destroyed all the laurel trees. Every year, there were more and more counties affected by this. This was one of the fastest and biggest epidemics of tree health or tree death that we have witnessed in the history. It reached the productive avocado groves in southern Florida pretty soon after it got here. And this is what's happening with the avocado plantations now. They are many of the trees are being cut down because they are affected by the fungus. The whole tree wilts within just a couple of weeks and it's dead, and that it is threatening the rest of the orchard. The problem, of course, is also environmental. Laurel wilt is common in forests. Well, I should say it was common. Most of the laurel trees are now wiped out, have been in my area here in Gainesville. They've been wiped out for about 10 years. Already when I got here, there were very few laurels left in the forest. This is the reality of invasive species in Florida. There are so many, it's crazy. And so the lesson number one from this talk is that there are lots of invasive insects, especially in a place like Florida, which imports a lot of things and has a lot of different crops as well as environmental species of trees. And some of these invasives can eliminate the entire tree species from the landscape and from the industry. Now, our biggest concern is, what if one such invasive insect or fungus happens to attack pines or oaks, the trees that are fundamental for the landscape of the Southeast United States or of Florida? We really don't want the landscape of Florida to look like this. We really want it to stay green and happy and functional. Is it possible? Are there bugs and molds and diseases out there somewhere in the world that can do that? Well, we don't really know. And the government doesn't know. Nobody really knows. And for that reason, we have embarked on a big project or a suite of multiple projects, which can be summarized under the question, is there any threat to Florida trees on the horizon? And this essentially takes two steps. One is the assessment of foreign tree pests and pathogens that might be important for the American species of trees. And the step number two is working with government agencies on prevention of these specific potentially invasive organisms. So our first and biggest question was, which bark beetles and fungi native to Asia are able to colonize and kill living American trees? How do you answer that question? We really don't want to do that. We don't want to test it by bringing the beetles in, of course, that would be crazy. So how do we do that? By many ways. There are many ways to do that. And the method number one is working with overseas scientists. So, and the first part of that is building trust. We travel to many locations, particularly in Asia and in Europe, but increasingly also in Africa and Latin America. And we provide our expertise, provide some funding, and we share publications with local scientists. And we also get a lot of things. We get local help, we get more friends, and most importantly, we get pest reports, which is something you rarely get in literature. The other way to do that is we inform entire networks of people across this project. It was in Asia particularly. We set up a web campaign. It's called the AsianForestHealth.org. There's a whole website about it. Feel free to check it out. It's actually bilingual, so we have also a Chinese version of the same thing. On the website, we have provided many resources such as videos on why that work matters, how to sample beetles, how to take photos of these pests with a smartphone, how to sample them, and that kind of stuff, how to get them identified. A lot of simple videos. A big part of the information flow these days, of course, is social networks online. And so we work with those. We've started LinkedIn network and WeChat, which is a Chinese analog of Twitter. And we use Twitter. Facebook is hard in China. Twitter too, it's sort of banned, but we're trying in many other countries. And we have quite a few hundred of members. We have over 1,000 members combined now. So lots of that is young professionals in the forest protection field. We go to conferences, of course. Well, I should say we used to before COVID. This was one of the last ones. This was in Baoding. This is our little group here. We went to many conferences like that where we spread the message. And we also provide free collecting kits. If anybody asks, anybody who would like to join the effort, and or anybody who is just basically interested in looking around and looking at tree health, we send them a collecting kit, which includes a box cutter that you can use to scrape bark or to open up bark, vials with preservation liquid. This is a clip-on lens for smartphone. Everybody has a smartphone these days, so why not use its capabilities? And a forceps, and people can actually collect and document these potentially threatening insect species. What do we get as a result of all that effort? We get reports of aggressive beetles. And one of the sort of most important one that some of you may have heard, because we've been beating the drum about this one for quite a while, is the sweet gum inscriber. There are multiple species that we've learned about, but I will show you this one because it really is the most important one, we believe, the most imminent threat to a tree, very important tree in the United States. This is the sweet gum tree. The leaves kind of look like maple, but don't think it's maple. It's actually a completely different family. It's its own genus Liquidambar steraciflua. There is no such thing in Europe. The nearest one is in Turkey, and there's one species in Asia. And we found a beetle that attacks it and kills the American sweet gum.
In collaboration with our colleagues in China, my team discovered a bark beetle that kills the American sweet gum. It is called the sweet gum inscriber. A tiny beetle, but it attacks our sweet gum in masses. This is what happens to American sweet gums that are planted here in China. Here is the biggest trouble. Look at this picture again. Right behind this levee is the magnificent Yangtze River, the largest export hub in China, and a quick route directly to the rest of the world. Just a few steps from logs teeming with aggressive sweet gum beetles. Just imagine our southeastern forests if this bug got a ride on one of these container ships.
So that's not a beetle we want in the United States. And the only way we got to hear about that was because we worked with scientists in China. Otherwise, we just would have not known. There's one other interesting spin, very recent spin to this story, which is the more we study the beetle, the more we find out how interesting it is. And, so this is the sweet gum inscriber under the bark of dead American sweet gums. By the way, we know that it kills American sweet gum because it was planted in China intentionally as an ornamental tree. It doesn't kill the Chinese sweet gum. So without scientists trying to plant the American sweet gum, we would have no idea that it's such a threat. But back to the beetle. These are the beetles. And what do you think is this white fluff around it in the chamber where it pupated and where it emerged? Can somebody yell it at me? What is this white fluff? Well, I can't hear anything, but maybe it's because you're quiet or because my speaker is bad. But I'm just going to tell you. It's, of course, a fungus. And when you peel off the bark, you see a surprising thing. You peel off a bark of even a tree that survived. This is the bottom part, the sort of the inside part of a bark of a sweet gum that was attacked but survived. This is the remnant of the gallery that failed. There is no beetle anymore. But look, the fungus is there. And this black stuff around is a big lesion that the fungus left behind. Does it remind you of something? Does it remind you of something? Well, I'll give you a hint. This fungus we identified as Geosmithia recently. This really reminds us of the 1,000 cankers disease of walnuts, which has swept through the United States a couple of years ago, killing lots of walnuts. And now it's already established in Europe, and it's been a major issue in Italy and surrounding areas. This is where a tiny little beetle, on its own inconsequential, carries a fungus, and the two together can girdle and kill healthy trees. So why is sweet gum important? Why it's important to protect this? Because it's an environmentally critically important tree in the United States, but also because it's one of the dominant pieces of this stuff, the pellets that are being produced here in the Southeast US and shipped all over the world for bioenergy generation. Many people in England and Japan and elsewhere are heating up their houses with these pellets. So the potential losses, if this beetle is to show up here, would be not only a devastating losses to the forest ecology, but also about $150 million a year to the industry, the bioenergy industry. So we don't want a beetle like that. Now, the lesson from this part of my presentation is that several exceptional beetles and fungi can kill living trees, and collaborations with colleagues overseas can result in the reports, something that is not published in peer-reviewed literature. Here's a second method for how we predict which beetles are going to be important. We check the climate forecast. We all know this. We all are paying close attention to the changes in our climate and its impacts on forest health. You know, with bark beetles, it's already well established that the warming, changes in precipitation, and silviculture, the dense stands and even aged monocultures, they result in increasing stress on trees, more beetle generations, faster beetle development, and that increases the size and frequency of bark beetle outbreaks. You all know where this is, right? You may have seen some of that on your field trip. This is, of course, in Czechia. This is the spruce forests that were attacked by the Ips typographus, European spruce beetle. But those are not just plantations in Europe. Those are, of course, the sort of the hotbed now of the political turmoil surrounding these outbreaks. But we see that all around the world, climate-related outbreaks of beetles. The same thing is happening with Ips morphosus in the western United States, southwestern. The southern pine beetle has been exploding in Central America, and not as much in the southern US, but more so in the northeastern US, actually. It's not so southern anymore as it's creeping up northwards. We all heard about the mountain pine beetle and many other beetle species outbreaks in the northwestern United States. This is one of the most documented outbreaks where in some places up to 80% of the tree cover was effectively removed, essentially. It's happening all over the place. Many of you have heard in Europe and Asia about Polygraphus proximus. That's also a sad story of a beetle that normally typically was restricted to the very far east of the continent, but then was brought apparently on the Trans-Siberian Railway to central Russia and all the way to Moscow now, Moscow region and elsewhere. So it's a combination of both an expansion based on climate change and also expansion based on sort of invasion behavior. The same thing is happening with ambrosia beetles, strangely. Many of you have heard of platypus. There are multiple species of platypus that are exploding now in their outbreaks. They all look kind of like this, like cigars. This is what typically an oak tree would look like if it's attacked by these timber borers. They're called pinhole borers. They're all ambrosia beetles that carry their associated ambrosia fungi with them. And this is a picture from Seoul, Korea, South Korea, where the oaks are being attacked by their native platypus beetle. But it's not that anything changed about the beetle. It's that things changed about the forest. They are now very dense. They've been all planted after the war. Now they're growing up to maturity, and it's getting hot and dry. So again, it's the intensive silviculture and changes in weather that are making native beetles becoming more pestiferous. You can see the same in the Mediterranean. This is from my trip to Israel, where we were looking at these trees that were planted with great pride and great investment. And now see all those pines being turned gray after 40 years of happy living. Again, it's a combination of drought and local bark beetle outbreaks. This is mostly caused by Orthotomicus staurosus, but multiple other species are involved too. In the tropics too, Cryphallus dilutus. It's a beetle, tiny little beetle that was barely known up until recently, when it was shown that together with a fungus, Ceratocystis, it causes mango wilt, because these beetles are boring right on the edge of a living tissue and dead tissue. And by that, they're sort of advancing the necrosis, the necrotic tissues, and eventually they kill the entire tree. So it's happening everywhere. There is another one from China. Some of you guys have worked on this one. Tomicus yunanensis, again, responded to several years of drought, and it has killed a bunch of pines in Yunnan in China. So the lesson here is easy. Climate change is awful, and it's impacting forests as we are watching it. Forests are, in fact, one of the basic barometers of the climate change impacts. We are recording many secondary beetles and fungi, I should say formerly secondary, that are turning into tree killers following tree stress. So there was invasion, there was a new pest, and there was a climate change effects. And what's the third method? Well, we are not just observing, we're actually doing experiments, trying to discover these dangerous cases proactively. We're not just waiting for reports. Here, we are trying to discover what some of you have already mentioned, the unknown unknowns, that there are species that we know are dangerous, but there are species we don't even know about.
How do you predict which of the thousands of Asian wood borers may be the next big invader in America? Easy. Just plant American trees in Asia. Our team is doing just that. We call these the Sentinel Gardens. We, the Forest Entomology Lab at the University of Florida, joined forces with biosecurity experts at the Florida Department of Agriculture. And here we are in China, checking up on our gardens. Here is Simon, the man who incredibly manages our operation in China.
Okay, so we have this seemingly healthy citrus, and if we look at the bottom, we can see frass and sawdust, and this looks to be a longhorn beetle emerging. This is one example.
Will this longhorn beetle eventually kill this?
Yeah.
So this is
we'll
actually...
show you dead ones.
Here is Leroy, the chief of Florida State's Bureau of Entomology. This is Yuli, a man with a unique combination of a passion for bark beetles and excellence in Chinese politics. And this is Paul Skelly, who knows pretty much everything about beetles there is to know.
So as you have seen, it takes a lot of work and a lot of politics, but we're making this happen. We're establishing the so-called Sentinel Gardens. Sentinel Garden essentially means a garden of trees that are put into the ground in advance, so we can monitor the emerging threats. We are trying to look at not just bark beetles, but borers attractive to live trees from all groups, not just bark beetles. We're also mostly interested in insects that are not just eating dead wood. There are hundreds of species of longhorn beetles and buprestids and bark beetles that are traveling on dead wood around the world, and people are busy monitoring those. But frankly, often many of those are completely inconsequential and are sort of a distraction. What we're trying to figure out are the ones that bore into live trees, because routinely those are the ones that actually need a response. Those are the ones where we should concentrate our response investments. And we're focusing on key commodities. Right now, we've planted pines, oaks, avocados, and citrus. Of course, citrus is not American, but it's Chinese, but it is an important commodity. Those pines and oaks are American trees, and avocado is also Central American, but it's planted wildly in Florida. We are now expanding those gardens. We have ran the ones that are fully in green in Fujian and Yunnan, and now we're starting expand further north in Shandong and Liaoning provinces. And we have results. After the first 2 years of doing this, we've collected a whole bunch of beetles, actually. And this is a result from 3 of those Sentinel Gardens. It's interesting on many levels. First, we are confirming known threats. You know, we've known that the Anaplophora chinensis, the citrus longhorn beetle, is a threat to citrus. It's been known, and it's good to confirm, it's good to show that with our method, we can confirm the ones that are already known. What's even more interesting, though, is that that same beetle was also found attacking American oaks. And nobody knew that. Nobody knew that this, we knew that it's a threat to citrus. We had no idea that it also has taste for oaks. And this is a female that's walking down a trunk of our planted red oak, and she's chewing an egg deposition niche. And then she's gonna turn around and lay an egg. And the larvae are developing in oaks. So again, this is a species that we don't really want in the United States. There are other species, and those are I would like to focus your attention to. This is a species of Cryphallus. These are tiny little beetles, bark beetles, that previously almost no one paid any attention, and I think that should change, because we found two separate species in many cases attacking our slash pine. Slash pine is a very important agriculture, I should say, forestry commodity in the Southeast US, the second most important pine. And they totally destroy them. These beetles totally destroy the ones that we planted. And look, I want to point out this column, incidences, you know, like the citrus longhorn beetle, that happened once in oaks. These guys, these beetles destroy all our pines that we plant out there many times. And it's not just there's one beetle, that this is usually dozens of beetles attacking these little trees. So two species of Cryphallus, one species of Heledius, which is a different species, different genus, entirely different group of bark beetle, but it also attacked our pines many, many times. The other scary thing is that it's not just an attempt at feeding. These beetles actually make galleries and successfully reproduce in these pines. So what are our results?
Here we've got a hole in our tree, and we're trying to work out what caused it.
Look like Hypothetimus gallery.
Bark beetles.
Yeah, with a live adult there.
There are some bad news. The citrus longhorn beetle damage, for example, that was worse than we expected. It destroys citrus and some of our American oaks. The little pygmy borers on our pines are troubling. They breed on slash pine while its needles are still green. But there are also good news. We are starting to see that many of the destructive pests are already known. In other words, there does not seem to be an endless number of these potentially invasive borers. It looks like we can detect them, study them, and even get ready for them before they invade the United States. It's just that somebody has to come here to Asia and do it. So here it is, our treasure and our concern, the wood borers that attack living American trees in China. So stay tuned as we keep identifying them.
We've talked about beetles so far mostly, but there is plenty of fungi too that we really need to discuss. I'll just slowly start finishing here. We are also looking at the pre-invasion assessment of fungi associated with these exotic wood borers. This has been many years of a project where we've collected many exotic ambrosia beetles from across the world. And this altogether was over 250 from Europe and Asia. And we selected 55 of them based on various criteria and grow fungi from them, fungi associated with them. And those we brought over to the United States to our biosecurity greenhouse and inoculated them into American pines or oaks and looked at what happens. We are essentially testing, is this going to be another laurel wilt? Is there another Dutch elm disease that's affecting pines or oaks? Something that is threatening to wipe out the entire species. Altogether, we've tested 111 fungal strains, and this is what we found out. Again, I said after 5 years of work, this is finally the results coming together. Almost all of the fungi were harmless. They didn't do anything with the trees, they just died there. The trees are easily able to defend themselves. That's sort of expected. As I said before, most of these organisms that travel around the world are not really harmful. We found a couple of mild pathogens that cause lesions and that can persist in a living host. And those are worth further monitoring. Those need further research to see if perhaps a mass inoculation or mass attack of their vector beetles would cause a tree death. We don't know that yet. The best news is that there was no tree-killing pathogen. We did not find anything like the laurel wilt fungus or anything like the Dutch elm disease fungus that would be a threat to pines or oaks in the US. So to us, that sort of confirms our biological prediction, and it's a pretty good news for the forest. But of course, we're far from having an exhaustive search, and this needs to continue, this work. So I would like to go back to my initial slide, asking, is there any threat to Florida's trees and forests on the horizon? And we talked about how we travel to Asia and we work with the local folks, and we assess the foreign tree pests and pathogens before they show up in the US. The second step is work with government agencies on invasion prevention. We want to make sure that our data don't stay just in a published paper somewhere and get forgotten. We want to make sure that people can use that, the right people can use that, the people who can actually make a difference. And for that reason, we are working with APHIS, the United States Department of Agriculture arm that is in charge of providing security or securing the borders and providing quarantines, checking incoming cargo and such. And essentially, it's a plant health organization. And we have put together a objective, so I should say, the APHIS itself, the agency, has a objective prioritization exotic pest model, which is a ginormous model that relies on a big amount of input data that an expert selects, and it's about 50 weeks. So I should say they have had this model before, and it's been developed for agricultural pests. And now that didn't work at all for bark beetles, ambrosia beetles, wood borers and such. It just did not have the right variables. So we have now sort of taken it over and are updating it for the wood borers. And what is the rest? What is the result? The result of that model is that it actually feeds into a simulation algorithm, and it simulates uncertainty. It takes the uncertainty associated with the answers and simulates the probability that your target insect falls into the category either no impact, which is this one, or medium impact, which is right here, this is the threshold, or a high impact. So we're essentially looking for species that would end up somewhere here. This particular one, when the simulation ran, ended up almost certainly a low impact. And so that's what we're sort of happy about. Ultimately, this is a decision support for action against newly invasive trees, invasive beetles. If we have one of those unknown unknowns, and some of those shows up on our borders, we should already know where that insect belongs on the scale of low impact to high impact. So thank you very much. I am very glad to be able to mention this to you, and I would love to see questions. I'm not sure if anybody's talking, but
can you hear me?
Now I can hear you, yes. Before I couldn't hear anything.
Yeah, we had a minor technical problem due to my incompetence, I guess. So, but now we're online here. We have 2 handheld microphones, and so we can take questions from the audience.
Please.
Jerry, congratulations on the very nice talk, and it's Ecki Brokerhoff here.
Hi.
And I'm sorry you couldn't be here in person. It would have been very appropriate since you've come from somewhere here, right?
Oh, tell me about it, yes.
Anyway, just a question. So are you also looking at Asian trees planted in the US that may be attacked by North American species?
Excellent question, Ecki. Yes. So this is something that Enrico has already started in Ohio, and we're starting to develop some of those reciprocal plantings too. They are not in the ground yet, but we have plans to get it started next year. We're going to take some of the most important trees of Chinese origin that we can actually get our hands on here and see what we can what we can detect. We would like to do that in Florida, of course, which is extremely biodiverse, but we can also do that elsewhere. Yes, so it's a plan.
Hello, Jerry. This is Jean-Claude Grégoire.
Hello.
I noticed that you are focused on Asia. Are there other regions of the world with climate similar to Florida where you might have also an interest?
Oh, we are interested in everything. You know, the advantage or disadvantage of Asia is that it's not only a similar climate, but it's also biotically very similar. Most of the plant genera have congeneric species in both regions, the Eastern US and the Eastern Asia, which I'm sure you guys have discussed that, but that is also one big reason why there have been so many invasions between the two continents, you know. And it's not just that Asian stuff is ending up in the US, it's also the other way around, that American beetles are, or nematodes are ending up in China, are invasive there. But to answer your question, we would love to explore South America, the more temperate regions, and Africa, the more temperate regions. We would love to work in Australia, of course. I think this is a line of work that needs growth. We are already able to provide clear examples of the high threats, sort of immediate threats. And so I believe that we're now working with the Forest Service on expanding this work. We'll see where it goes. You know, I have to say that the COVID situation is not making it any easier. It is, you know, now wanting to really work collaboratively with people around the world and not being able to travel anywhere is just crippling, but we're hoping to get over that soon.
Thank you.
Hello, Jerry, it's Alain. It's a pity we cannot meet directly, but I have one question, which is joining the one of Jean-Claude. You centered your activity on China, as we are doing too. But if we take into account climate warming, maybe it would be more interesting to have a deeper look in Southeast Asia. I mean, Thailand, Indonesia, and especially regarding Florida and the climate in Florida. And second, but you just replied, so it's difficult now to do such experiments in China for all of us. So how are you coping with this?
Oh, and exactly. We have the same issues that you guys have. It's been, in China, it's difficult not just because of COVID, but politically, increasingly difficult. And so we are definitely looking for collaborators outside of China. We've been looking at Taiwan, South Korea, but also a little bit further south, Thailand. You know, I would love to work in Indonesia. But again, politically, it's virtually impossible. If you watch the development of the regulatory apparatus there, it is increasingly restrictive, especially under their new president. They're restructuring the entire ministry responsible for science, and it's going to be even more restrictive from now on. It would be difficult. But there are many other places in Southeast Asia where this would make sense, and places which have congeneric trees with Florida that are not present in China. Now, I have to say, though, that our work in Yunnan province is near the XTBG garden, which is fairly tropical. It's not exactly the humid tropic, but there are many trees there, many species, many congeneric tree species with the Florida subtropical and tropical realm. So at least we have at least one place in the tropics. And if you guys are interested in expanding your operation into that realm, we will be very happy to work together. As you probably know, we had just signed a memorandum of understanding with INRAE. We had a meeting about it earlier this week, and I'm sure you're aware of that. Your colleague Carol was there, and I would love to explore opportunities to work together on some of these sites.
Jiří, thank you. We have two questions which were asked online. Can you see them, or should I just read them?
I can see them, and I am happy to restate them. Yes.
Yeah, I just read them through the audience here because it's they can read that probably. So Geoffrey Williams, who's going to give a talk later today, asked, could there be other factors that are masking potential pests in Asia or environmental factors that might limit their impact on US species in their native range?
Absolutely. Oh, there are so many. You know, one of the biggest issues that we have, one of the biggest weaknesses of our approach is that we are only planting small trees. We can't really plant a forest of big trees. That would be the most appropriate experiment, but it's impossible physically. So that's the one thing, that some pests or fungi are going to be most prominent in the, not in the sort of background abundance, but in the outbreak abundance, which can only happen if there's a whole tree, a whole forest that's stressed out. There are many, of course. There are the whole biotic buffer that there is, you know, parasites and predators that are making some of these insects low abundance in China. But then when they are released from that predator pressure, they can go nuts in the United States and become very abundant. You know, that's the case with the laurel wilt beetle, the Celosandrus glabratus, which is relatively rare in Asia. It's really hard to find it. There were very few of them in collections before it ended up in the United States. And there it is now vastly more abundant than in Asia. So absolutely, there are many such limitations. Now, on the other hand, it's not all bad, because these experiments show that we can replicate the pests and pathogens that have already happened. We are seeing them again in our sentinel gardens. We can replicate the effect of the fungi. We can replicate the effect of some of the borer beetles. So even though the study has limitations, environmental limitations, the most important thing is always the interaction between the herbivore and the plant, and that we can replicate.
Thank you. I think you already answered the second question by Travis Marzico, whether full-sized or mature trees would actually matter, and I think you answered that already.
Yeah, it matters. The good news is about the fungi, that the fungi, we've tried that and other people tried that, and it looks like, you know, like the Dutch elm disease fungus, the Ophiostoma novo-almi, is actually most effective with marginal statistical significance in young trees. Samplings are too small. Like if they don't have a fully developed xylem, they would be too small for experiments. But if it's already a tree of a couple of centimeters in diameter, that is already suitable and very representative as a testing material.
So we have one last question here from Enrico.
Hi, Yuri. I just wanted to add that testing small plants could be important to understand the effects on regeneration in target environments. So that's also very important.
That's right. That's right. Yeah, thank you. And before we all go, I just wanted to share the screen with you once again and show you one more thing. There's more that I wanted to show you. Mostly it's our work on the European beetle. If you have a moment, I hope you can see that we put up a new website called theclimatebeetle.org, where we're discussing a lot of these issues with beetles and forests and silviculture and climate in a very sort of public-friendly way. So take a look. These are all films. These are all high-quality films made by professionals, not by me. And there's some cool stuff like about the beetle and things, but I wanted to invite you. These are all films, I think pretty interesting ones. Some of them are actual sort of virtual reality where you can go in and explore the...
Okay, I'm going to show you a couple things in this forest. Let's walk over here to these horses. We got two horses working the forest. Black one, white one. Somebody's cutting trees. They're falling right at the moment.
Anyway, so I just wanted to invite you to have a nice watch after the end of this conference. Thank you.
So thank you very much, Jiří. It was a pleasure to have you here, and we hope you stay with us for the rest of the meeting. And
absolutely.
Yeah. Now back to your breakfast. And we'll have coffee break now. And thank you, all the speakers, and thanks to the audience as well.
All right. Well, we'll start again. I'm Jean-Claude Grégoire, and I will be moderating this session. And we are something like 15 minutes behind schedule. So we'll just have 15 minutes more for move on with 15 minutes of bonus, so to speak. So we have 6 presentations today, and among them 2 remote presentations. And the first presentation is by Lucas Seehausen, and the title, should we read the title, or is he going to read the title? You do it? Perfect. So the floor is yours.
Thanks.
Yes.
Perfect, thank you. So yeah, I'm Lucas Seehausen from Kabi, and the talk's gonna be entitled "A Global Perspective on Biological Control Policies Between Patchwork and Harmonization." There's something about management in the title there, but that's I don't know how it got there. Let me start by asking a question. Do you remember Enrico Bonello's talk from today after lunch, the one after lunch? Remember it? Good. What he said. Thank you. Okay. No, there will be similarities, but I'm going to focus more on biological control, and so a little bit more focus on this. And this is a work that I did together with Catarina Afonso and Manuela Branco from ISA in Portugal. So first of all, no panic. Yeah, it is about legislations and so on, but I'm not going to read you the text of them or something like this. So you can stay here. You all had a coffee, and we went even outside. So no worries there. So why should we talk about policies in biological control? There are many reasons, of course, but one is that we had to have this project, HOMIT, Holistic Management of Forest Pests and Diseases. It's an EU project, and in one of the tasks, we were working on bridging the gaps between policies and practice, and we wanted to do this on the legislations of biological control. When I say biological control, I mean in this context here, classical biocontrol, so the import of biological control agents into a country where an invasive species has become problematic. Maybe we can start with an example. And this is here the time before regulation. So I want to explain you how we got from no regulations to regulations. And the example I want to give here is the introduction of the Australian Vidalia beetle to control the cottony cushion scale in citrus orchards in 1888. So one of the first attempts of biological control, and this one happened to be very, very successful. And we can say that this was one of the first ones that actually was very successful, and and it was kind of like the starting point where people started more thinking about this as a possible pest control a method that can be used. So you can look at this as well in this graph here. We have the number of introductions per decade, introductions of biological control agents, classical biocontrol agents. You can see that from the very beginnings of the first trials of this method to the 1930s, there was a very steep increase of cases, so very well used and more used pest management practice. And then we had a decline. This coincides with World War II. This has to do with the war itself, but also because at this time, chemical insecticides became largely available and very cheap, and were used a lot as a pest control method. Then we can see that that the cases go up again. And this is because of a movement, an environmental movement. You may all know the book Silent Spring that pointed out the big risks and damage chemical insecticides had done already to the environment. And then in the 1980s and in the 1990s, there was a big debate among scientists, but also the public. And we can see a shift in perspective from the benefits to the risks of biological control. and that led to the implementation of legislations in the end and regulations in many countries around the world. And unfortunately, what came with that was the big decline in classical biological control being used as a control method, as you can see in this graph. If we talk about legislation at the national scale, we have to point out a couple of countries that were basically pioneers on those legislations, have a long history of them. And one is definitely Australia. Australia had its first Quarantine Act in 1908 already. It was later replaced by a Biosecurity Act. And it was a piece of legislation about protection from negative impacts associated with entry, establishment, or spread of exotic pests and diseases. It also is one of the only countries that actually has a biological control act, so an act that is directly relevant to biological control, and it includes the requirements of studying the risks to the release of an agent organism may pose. And a third piece of legislation that is important for biocontrol in this country is the Environment Protection and Biodiversity Conservation Act, which provides a legal framework to protect flora, fauna, ecological communities, and heritage places in Australia. So relatively complex already. If we hop over to New Zealand, it is fairly similar, but there are two main pieces of legislations. And they also have a very long history with those legislations. So we also here we have a Biosecurity Act and the Hazardous Substances and New Organisms Act that also requires risk assessment for classical biocontrol agents to be imported and used. Although those were some of the first countries that actually thought about this and thought about this long and hard, as Gauss and all pointed out, there are still misalignments between the lawmakers, the implementing agencies, and the science community, as well as various inconsistencies and practicalities. So also, even there, not everything still works so very well. We can go over many examples here. I don't want to do this. However, I want to talk about Europe. And in Europe, well, it's complicated. You can imagine already so many little countries, and they all want to be have their own kind of legislations, and it is complicated. So this is a fairly outdated map here, but this one that got published by Hunt et al. In 2011, and shows that this is really a patchwork of regulations that were implemented in preparation or not even a regulation in place. To explain you a little bit better, there's a photo missing, I don't know why. I can explain you the case of the Asian chestnut gallwas, Durycosmos coryphyllus and its parasitoid. Here you have a timeline. The insect was first reported in Italy in 2002, and it was very fast that Italy decided to import Torremo sinensis, a parasitoid from Japan, and this was just a couple of months before Italy forbid completely the import and use of exotic species in the country. After a very short risk assessment of only 2 years, the parasite was released in Italy the first time in 2005. It was not before 2011 that in France, the insect was also released. And only 1 year later, it was actually forbidden by the Swiss government that did a risk assessment and said that this is not a safe insect and could have could be problematic for the environment. Nevertheless, other countries thought very differently. They did not only assess the risks, but also the benefits of releasing such an insect. And it was thereafter widely distributed and released. The latest one is in Greece that released it after a 4-year span of risk assessment and found that the benefits of releasing it are higher than the drawbacks that it could have for the environment. So you can see on this example, there are many countries with many different views on risk assessment and the use of biological control agent. You can very well, because they're so close together, you can very well think about what are the consequences of releasing or non-releasing. Then going from the national perspective to the more international perspective, there's some things that one has to understand if you want to talk about the efforts of certain institutions to harmonize a little bit this patchwork of legislations. And I have to talk in this context about a very important treaty, the International Plant Protection Convention, This is a 1951, so fairly early multilateral and intergovernmental treaty signed by now over 180 countries. So it's fairly important. It is overseen by the United Nations Food and Agriculture Organization, or also called FAO, and it introduced the so-called International Standards for Phytosanitary Measures, or ISPMs. And those, to show you the importance of those were actually adopted in the World Trade Organization's Sanitary and Phytosanitary Measures Agreement. So taken very, very seriously. Why is this important to biological control? Well, in 1996, the FAO developed a code of conduct for the import and release of exotic biological control agents. It's a very important document because it describes the procedures for the import, export, and use of exotic natural enemies. And it was specifically designed for countries where national legislation does not exist or is inadequate. So kind of going towards this harmonization of this patchwork. And only a few years later, the code was actually transposed to the ISPM #3 guidelines for the export, shipment, import, and release of biocontrol agents and other beneficial organisms. So showing again that it was taken very, very seriously. Also, not only on the international scale, but a little bit smaller again on the regional scale, we can see attempts of harmonizations of legislations for biological control. And there are many examples that I could name you here. I want to focus on one, this one here. And this is the European and Mediterranean Plant Protection Organization, also called EPO. And this organization actually developed, between other things, the so-called phytosanitary measures based on the FAO's code of conduct that I just talked about. There are standards and describe ways of pest risk assessment and risk assessment for biological control agent and how they should be regarded for the introduction and study for potential biological control agents. So this was kind of like an overview, and I'm very well aware that this is it's a subject that one doesn't necessarily want to think about so much because it's complicated, and we all don't like really the policies. We often conceive them as something that is hindering us in our work. But I do want to encourage you to think about those legislations actually as useful tools, because in the end they enable us to work very safely on this subject and to put certain standards on the work that we can do this very safely. In order to promote biological control and create awareness of this use control method, there's also another way that we can help, and this is to get involved and influence policy making, so to directly get involved in the subject and work on it. Another way, the third way, is to share experiences, resources, and information. This is basically what we're doing right now here. Thank you for being here. And a very, very and ever more important thing to do is to communicate with the public, because only if we inform the public about this useful tool, and we now can do this very safely, they will then create a support also at the policy level that we need in order to actually do it. So to bridge this gap between the legislation, the policy, the politicians, and us as biological control practitioners. So I think it is now a very good time that we're thinking more and more again about how we can do safe pest control and use less and less insecticides, and that we can reverse this trend and work on doing more and more biological control again. And so I think those legislations can actually help us in the end to do so very safely. And if you didn't listen, or you want to know more about it, this presentation will also be soon available as a chapter in the book Biological Control of Insect Pests in Plantation Forests, so you can read it up. I want to thank the Homewood Project that supported this project, and you for listening. Thank you very much.
Thank you very much. It was really interesting, and you are fully in time. So there is time for questions. No question? I have one question at least. You showed us the last curve, which was coming up. But do you think that new legislation might open the field for new attempts at biological control?
That's actually a difficult question, because I do think that new legislations, if we are influencing them, if we are explaining to the policymakers what needs to be changed and what needs to be in there, only then we can actually start doing more and more cases. I can only say so from the example of Switzerland, where we just applied for releases of the biocontrol agent Ganaps brasiliensis against Drosophila suzuki. And it was a long process, and we actually need to talk to several offices, federal offices, to actually make them understand first what we wanted to do and why we wanted to do it. And only then there came suggestions from them how we can actually make this possible. Switzerland is by default very strict with biocontrol, and as you saw in the example, even though other countries might readily do it, doesn't mean that Switzerland will also do it. So it shows me that if we engage directly with those policy makers and explain them what we need in order to do something like this safely, then it is possible. However, of course, in some other countries that might be other situations, and not so easy to do that actually.
Any other question? Then see, Sandy has a question.
I was just wondering, what was the reason for the rejection of the Asian chestnut gall wasp parasitoids? Yes.
A very interesting question, and there's actually a paper published about this by the Swiss government. This is very interesting. If I remember well, there was a multitude actually of things that, in the government's perspective, were speaking against the release. One of them were non-target effects on gall wasps that are indigenous in Switzerland. Then the possibility of hybridization with other native parasitoids that are closely related. There was a third one that I cannot remember right now. But the interesting thing is that there was a lot of studies after the release, especially by Italy, and they actually found that those things were true. So there were non-target effects, only important in some environments and less in others. And there was actually apparently hybridization with the indigenous parasitoids. But nevertheless, even the airport says now, yeah, we should not only look at the risk that might be in some cases minor, but also on the benefits. And so many countries decided, despite those risks, of non-target effects, for example, happening, it has much more benefits for the environment if we release this parasitoid.
Maybe just on this topic, I want to point out a kind of paradox, because Switzerland forbade the release of the parasitoids, but they were already released in Italy and in France, and they are spreading very fast. So in a sense, it was wasted time, I think. But Enrico, you wanted to ask a question?
No, actually, I wanted to make a comment, and that is that you talked about engagement just like I did, right, by us. And I would suggest that UFRO would be a good organization to get help from. If you want to engage in your own country, you could work through, working parties like this one, for example, I think, for example, to gather support. Huh? Right. I mean, I'm just saying that if you want to contact your legislators, for example, and go talk to them and so on, you could potentially get support from a group like this one.
Okay, thank you. I think now we should go to the next presenter, who is Raffaele de Andrade, who is going to, from the University of Maryland, who is going to talk about ash decline and emerald ash borer biocontrol.
Hello, everyone. I'm Rafael, and I'm a postdoc at University of Maryland. I'm here to talk about my research on ash decline and emerald ash borer biocontrol in the continental United States. And the emerald ash borer doesn't need much introduction here. It was first recorded in 2002 in the United States, but probably arrived earlier than that. And it's threatening billions of ash trees in North America. And among the efforts to curb its invasion, it's the introduction of native range parasitoids, namely the Tetrasticus planipenise, Obius agrilli, which is an egg parasitoid, and two species of Spatius, which are also larval parasitoids. And in terms of the stages of an invasion process, the impact of introduction is measured by its capability to reduce the target species, but more specifically in our case, its ability to reduce EAB populations, and if that reflects on reduced ash decline. So our group at University of Maryland in the Department of Entomology, we are assessing the ecological consequences of the emerald ash borer invasion and also assessing the impact of biological control. And my specific research is asking whether biocontrol can mitigate this ash decline at a continental scale in the United States. So for that, we are leveraging the largest biocontrol database in the United States, which is the Map Biocontrol portal, and also the forest inventory analysis data set. The Map Biocontrol is a portal in which multiple users will enter data on parasitoid release and recoveries. And it has its limitations because it depends on the goodwill of the users to enter the data, especially recovery data. And so it has its gaps, but we work on try to work around that. So after some crunching and digesting of the data, we end up with a data set that shows the location, the year, species, and the release effort of these parasitoids. So we can plot them like this, if you look at them by species. And here the colorful circles are the releases, and the dark, the black dots are the recovery records. And the circles that they are proportional to the release effort. And then you can see that Spatius gallini has a much lower release effort. And that's because while the other species has been approved since 2009, Gallini has been approved only 2015. So it's been going on for less time. So we have less total individuals released. We also have the data set of the year of emerald ash borer detection by county. And county is a subdivision of states. It's the equivalent of a municipality almost everywhere else. And this is zooming in the state of Maryland, where we are based, and where the locals are very proud of eating crab from the Chesapeake Bay. And they are convinced that they invented crabs, but I don't think they did. So the detection by county looks like this. The grain of the data can vary a lot because the size of a county can vary a lot depending where you are in the United States. And like I said, we're also using the forest inventory data set, which is in operation since the 1930s. And the plots that we use to assess ash decline are revisited every 4 years. So that makes for a lot of data that Luckily, we have our FIA package to make things a lot easier. So again, zooming in Maryland, if you look at the FIA plots that have or at some point had ash trees, they look like this. And when you retrieve from map biocontrol the release locations, it looks like this. So you have somewhat a mosaic of spatially mismatched data. So what we are doing in our research, we are creating these spatial buffers around each release location. First of 10 kilometers, and then 20 and 30 kilometers. And then when we overlay that with the FIA plots, we end up with plots that fall inside the biocontrol buffer and plots that fall outside the biocontrol buffer. And then we can see if the ash trajectories in those differ in any way. So this is how the whole data set looks in our case. When we zoom out the whole extent of the EAB invasion. So the biocontrol program is in second decade, and there is no broad scale assessment of its efficiency and impact. So my specific question is simple, is if FIA plots close to the release points show us lower ash decline. And for that, we are using the generalized additive models with the percentage of ash. And as the years progress since the detection of EAB, and we compare the two trajectories. So our axes have the year since EAB, they're all aligned. The plots are aligned by the year of detection, which appear in the 0, and the percentage of ash on the y-axis. So when you plot the actual data, it looks a bit scary like this. But when you see the predicted values of the model, it actually behaves a lot like we expect, which is the percent of ash quite stable up until the detection of EAB, and then it declines. So here we're looking at the trajectories of the release by species. And we are comparing the trajectories in orange of the ones that fall inside the release buffers, and in green, the ones that fall outside the release buffers. So at first, it looks like biocontrol is not doing much to reduce the ash decline, except for the latest species, Patius gallini, which by the way is the one that was the least released, is showing a significant difference in these trajectories. And it also shows it's not wise to read too much into the very end of these predicted values, but it shows some suggestion of increased recovery or recruitment after 15 years or so. So if you look at the map on the top right corner, if we increase our buffers, we now include plots that are further away from the release into the biocontrol treatment, and we don't see anymore the effect that we were seeing with Patius gallini. And as expected, the same if we increase the buffer to 30 kilometers. So in conclusion, the biocontrol does not seem to prevent initial ash decline. Maybe that suggests that some studies on release timing could be made. Perhaps if releases are made too early, there's not enough EAB density to sustain a parasitoid population. If it's made too late, perhaps the ash decline has passed a Rubicon and there's no way back. But we see promising results with Patius gallini within the 10-kilometer radius. We indeed see a higher dispersal and establishment capabilities of these species. We have a paper submitted to the Journal of Economic Entomology on that. And some characteristics of these species, like the much longer ovipositor, perhaps explains some of the higher success. It can, with the longer ovipositor, it can attack larvae in much larger trees that has a thicker bark. So maybe that reflects on a much better efficiency. And the possible increased recovery and recruitment within the biocontrol buffer. So, but that requires further analysis. The recruitment data is a bit more sparse for this kind of analysis. And of course, we need more time to see what happens with these plots. So the study has been a collaboration of a lot of people, including the university in Delaware. But this is the main group at University of Maryland. And that's it. Thank you.
Thank you very much. There's time for questions.
Thanks for a great talk. I'm just curious if you've looked at how the effect, or if the effect has varied at all geographically. For instance, Spatheus grilli is not being released north of the 40th parallel anymore because it wasn't being effective. So I'm just curious if you've looked at that, and if so, how have the results possibly varied geographically for the different parasitoids?
No, not yet. We didn't look at this. We run our analysis first in Maryland to see if the script works, and then we run with the whole data set, which takes a lot of time. But we haven't looked at this specifically in each region, but it's a good point to try.
Yeah, thanks very much. Really interesting. Do you have information on the host range of these parasitoids, if they attack other Agrylus species?
They don't seem to attack. You mean other species in North America?
Yes.
Not that we know of, but
so they're all specific to Platypenis.
As far as I know, but maybe. Yeah.
Maybe you have some data, some information on this in your database, or are you strictly only looking at Platypenis?
No, that we do. This database does not include this specific information. The sampling for recovery is made via different methods, such as debarking of trees and then looking at the larva. But I'm not sure. I don't think there are any records of these parasitoids attacking other species, as far as I know. But it's something I would need to check.
Excuse me, this is Faith Campbell. APHIS did very thorough risk assessments on all 4 parasitoids before approving their release. They have a lot of data on North American Agrylus and other kinds of insects that they don't attack.
Yeah, so there's the voice has responded.
So did you look at the results by species of ash, or would you actually expect dramatic results if there is no host resistance at all? Do you think biocontrol without host resistance, how effective can it be? Maybe it can be somewhat effective, but do you really expect to solve the problem if you have very susceptible hosts?
We know on this analysis, we look at all ash species. And I believe green ash and swamp ash would be dominant in most of the regions that we analyzed. But we did any analysis separating the ash species. For each plot, we have revisitation period of 4 years. So normally, whenever you subset the data, it gets very sparse and the models don't converge anymore. So at this point, we are not separating ash species.
The second part was, would you expect what's the maximum that biocontrol can be expected to achieve in the total absence of any resistance within the whole species? Can it work entirely by itself, or would you advocate perhaps developing some host resistance as well?
Yeah, so I think the numbers are still to be evaluated in terms for the Spatius gallini species, but the others, it's a marginal mortality that we can observe. For instance, the mortality caused by woodpeckers is much higher than what we see with the parasitoids. So it would certainly be welcoming with other approaches to curb the invasion.
Yeah, so this is actually an issue that's close to my heart because I worked on emerald ash borer for a long time. And I really want to commend you for conducting this research, because I think it's the strongest evidence so far of whether or not these parasitoids are having an effect ultimately on survival and recovery of ash. And it seems to me that at best, the effect is dubious. And APHIS has spent millions of dollars on this program. And I want to put you on the spot a little bit here and ask you, if it is true that it's dubious in value, would you have recommended APHIS would embark in this kind of program or not, in terms of cost benefits?
Yeah, so I think it's hard to go back in time, but I feel like my recommendations would be to try to improve if release timing could be improved. I think we work with the Department of Agriculture, both the US and in Maryland, and how the releases are made are very limited by logistics and what's available and what's being reared and how much is being reared. So I think these things have been defining the releases much more than what science tells it would be better. So I think, yes, it's a worthwhile program, but it has to be made in a better way. It has to be improved. But I wouldn't want to be the guy that says, it doesn't work, stop it. Thank you.
The reason I'm asking, sorry, just one last point, is because APHIS and again, for the international audience, APHIS is the Animal and Plant Health Inspection Service in the United States. APHIS has decided to go away from quarantine measures and really go all out on biocontrol, and that's why this question is kind of pertinent.
Well, thank you so much. It was really very interesting. If there are any other questions that could be asked after, and personally, I would like to ask one question after. So the next speaker is Karel Czerny, who is going to talk to us about invasive oomycetes in Czech forest nurseries. No?
Hello, everybody. I would like to present here some notes to Phytophthora presence in Czech nurseries, in forest nurseries. So we have in this country 6 widespread naturalized pathogens from this genus. They usually infested forests, wet forest types, for instance, alder cars, or riparian stands, or floodplain forests. I don't know how you see it. There is a group of Norway maples in a floodplain forest colonized by Phytophthora plurivora. But we realized that these pathogens are spreading not only via water, but also by other kind of movement. for instance, and then they are able to infect also mesophilous forests, for instance, beech forest. And they are spread via, usually via maybe forest machinery or with visitors along pathways. For instance, in this case, there is a parking place of visitors in forest, and in front of parking place, sorry, there is a group of affected beeches. And the most important way of introduction of phytophthora into forest are, of course, forest seedlings, seedlings of forest trees. And we obtained 2 projects, sister projects, and we would like to identify the real Omycetus infestations in forest nurseries. And we would like to identify also potential influence of technologies used in nurseries on infestations. And at the end, we should develop some methodology to minimize the impact of pathogens. Very shortly to methods, we covered all the area of the Republic. We investigated 25 nurseries in different conditions using of different technologies. We isolated from more than 30 host taxa, and isolation was classical on selective media. We used also baiting method, filtration. Determination was classical, sequenation of ITS reagents, and so on. Also, we tested also efficacy of fungicides and some biological preparations also. And we also evaluate impact or influence of environmental factors, usually with use of GIS in ARPUS software. Of course, we used also important literature sources, especially work of Jennifer Park, which was very interesting and important for us. Main outcomes. We investigated infestations in 25 nurseries and isolated from more than 10,000 of host plants. And we realized that more than 90% of bear production of forest nurseries are positive for presence of Omycetes. We identified more than 40 Omycetus taxa, and 1/2 of them were Phytophthora. And there is their list, this nice garden, of course. And there are frequencies of the most important Phytophthora pathogens. Most of them are alien and invasive in the country. The 3 first are Phytophthora plurivora, Cambivora, and Gonapodides. And of course, these species are very frequent in our forests, also usually hygrophilic forest. But it looks that spreading of Phytophthora is of older data, and probably the process maybe take for 1 or 2 decades, because the spectrum is the same in forests as in forest nurseries. Some outcomes of irrigation water, we divide sources into 3 types, into wells, which were without contamination, of course, and big surface, big sources, maybe for instance for rivers or big ponds, were usually heavy contaminated, and presence of invasive pathogens. It was sorry, presence of invasive pathogens was very frequent in them. However, in low frequencies, of course. Classical picture of forest nursery. Most of these fields were contaminated. We sampled all this field, for instance, we taken more than 20 samples, and all of them were positive, but the disease manifested only in wet areas, on small depressions, or on places with heavy soils. Another problem with Phytophthora campivora is a beach field somewhere in central Bohemia. And this field was also completely infested, and disease manifested only in a small depression in front of the picture. The most frequent pathogen is Phytophthora plurivora, of course. This well-known pathogen, and it is now spreading into our mesophilic beach forests. This pathogen is very dangerous. However, it is not naturalized in the country, but it creates small, stable local populations, usually in gardening centers or gardens, and in some nurseries. In picture right, this field of some nursery in northern Bohemia, and the pathogen survives here for more than 10 years without problems, and the owner isn't able to eliminate the pathogen from the bed. Some influence of environmental factors on presence of Phytophthora. We confirm conclusive influence, for instance, of hydrological soil, hydrological group of soils and soil type, and probably many other factors could have also some maybe minor effect. This picture describes influence of soil type on presence of oomycetes and disease caused by oomycetes, and you see that plants on, for instance, Chernozems or sandy soils are less affected. On the other side, plantations on glade and fluid soils, glade soils are usually heavy infested. It is very important for management, of course. Technology. We found conclusive influence of growing technology. Bare-rooted plants were usually heavy infested. On the other hand, quick-potted plants were infested only in a few percent. Very important is, of course, source of irrigation water, length of growing, and so on. When we sampled the operation of some nurseries, we realized that first contamination usually was present shortly after mixing of substrate, but substrates were in all cases sterile, and the contamination came from water, from irrigation water. Quick-potted or containerized material was less infested, and all pathogens after 1 year of cultivation or growing, of trees were all from water, probably because these pathogens are frequent in sourceways, water sources. On the other hand, bare-root material was highly positive, and the spectrum of pathogens was quite different from the first one. Growing time is big importance, and during time of cultivation or growing, we found increasing diversity of pathogens and change of dominance in oomycetous communities, and so on. Something to chemical control. We tested all accessible preparations in the country, and in a big set of experiments. Here is an experiment with the beet seedling. It lasted 7 weeks. And in the red line is positive control in infection of soil infection by Phytophthora plurivora, and in bottom is blue line with negative control. And we have good outcomes with systemic preparation, phosphonates usually, or metalaxyl or dimetamorph. Frequently used preparations as copper or propellant were less effective, but they are frequently used, which is mistake, of course, and so on. We tested also a set of biological preparations, some stimulators or fertilizers, and no one of them were effective, with one exception. These preparations with Trichoderma kenningi and Hartzianum, but this preparation is not registered in the country, of course. And some conclusions. All investigated nurseries and growing fields were contaminated. We identified extremely high proportion of contaminated samples in Beirut production. In fact, most of them, more than 90% of production, was infected by Phytophthora or omycetes. We were horribly surprised by this outcome, especially in the point of view that in the country we have more than 1,500 of hectares on nursery plots, and most of them are probably infested. Every year total production of seedlings is more than 200 million of seedlings, and more of them are probably also infested. What to say? Very likely artificial regeneration is a very risky operation, and during past 20 years we regenerated more than 400,000 of hectares of forests, and probably many of them are now contaminated by omycetes. But to be honest, we don't know what is going on in these areas now. It is question for next investigation, of course. But because omycetes are able in these conditions survive and cause losses, can say that omycetes pose a time bomb in our forests. We developed some methodology for control in nurseries, but maybe it is too late. But there is only a few important rules. Nurseries shouldn't use resale plants, and they don't grow ornamental plants. They should especially product container grown production with air pruning system on meshed up benches, because there is no contact between containers and soil. Irrigation have to be also contamination free, sterile substrate, and so on. Of course, very important is shortening of growing cycle to less 1 year. And sorry, this, in fact, it is all. There is only small, simple flow chart developed for nurseries. Thank you very much, and sorry.
Thank you very much. We have just time for one quick question. Simone.
Thank you, Karel. Just a question, do the nursery owners or managers listen to your recommendation? Because you found a lot of Phytophthora species, including dangerous one like Cambira and Cinnamomi, but if I'm not wrong, none of the species is considered as quarantine organism. So are they willing to do something, or they just ignore what you recommend?
We cooperate with many of them. We cooperate with many of them, and some of them were not happy, but they would like to change operations to producing more adequate and not infested in so so infested material, but is a very demanding process for them and very expensive. But some nurseries now produce maybe 1/3 of production via container grown material, and they would like to increase number of saplings produced by this way. But I think that the support of Ministry of Agriculture is needed in this process because, for instance, no of our forest nursery, if I remember well, I don't have filtration of water. It is a big problem, of course. Thank you.
Thank you very much. Now it's time for René Eschen from CABI to tell us about small-scale sampling intensity in Sentinel nurseries.
Thank you. Good afternoon. Thank you that you're still here. I see some people leaving. It's nice that you're still here. So I'm going to talk about Sentinel nurseries, and that has a bit of overlap with what the last keynote was about. I'll explain to you what is different about what we do in a minute. But it's about finding new things before they are being introduced. So a number of talks have already addressed my first point here. So there's a lot of international trade, and that has been set to increase the number of pest introductions around the world. There have been very nice talks about this. There's very good illustrations, so I decided not to put any graphs to illustrate the point. I think this is something we all know. One thing that wasn't really shown and addressed is how complex and how dynamic these movements are in the trade. And so I put this graph from somewhat older publication showing that over the years, European imports have that traditionally were dominated by imports from the United States. Live plant imports have become more and more also influenced by imports from Asia, of course, mainly China. And the other important thing is that many of the established pests and diseases or agents of disease basically weren't known, and therefore they weren't on the radar of plant protection organizations. And this is very important because plant protection organizations are, of course, responsible for the implementation of plant protection measures, phytosanitary measures, and these rely on evidence. So if you don't know of a species and you don't know of its impacts, then it can't be regulated. So the solution to this is obviously to find potential pests prior to export. And our solution to this is Sentinel nurseries. As I said, this is very similar to what Vajiri spoke about. So we're talking about Sentinel plantings, and we distinguish two types depending on the origin of the plants. So Sentinel plantings are basically plants that you expose to pests and you monitor them on a frequent basis to find insects or diseases, and so that you can identify them. And the two types that we distinguish is Sentinel nurseries or in patria plantings, where you plant species that are native to the location of the Sentinel planting. And the purpose of this is to identify things that may be transported on these plants. So it's the likelihood of introduction to an importing country that you're trying to assess. And that is what I'll be talking about, and it's slightly different from what was mentioned earlier, which we call ex patria plantings or Sentinel plantations, where you plant species that are considered to be of value to importing countries. And these plants are then exposed to species or to pests and diseases that are native to the exotic range, if you want. And therefore, these these sentinel plantations can be used to assess impact. And we've seen very nice examples of that previously. Now, obviously, we all know that the presence of organisms associated with trees is very diverse, both in time and space. That's nicely illustrated by this graph on the left from a paper by Alain. And what you see here is the number of species they identified on tree species in China. You can, in 2 different sites. You can see that not only does the number of species that they found increase over time, but the total number of species that was identified is very different between these 2 sites. And a few years ago, we wrote a paper where basically the question was, what do you need to do to capture this variation? And we, in that paper, we suggested sampling designs for the assessment of species diversity with a particular tree species. And we used a few case studies to explore where the diversity of the variation comes from, things like climate and location and this kind of thing. And one of the outputs or one of the figures in that paper was this nice, decision support scheme, if you want. And the top half is about the selection of locations in the world where you would like to have these sentinel plantings. And the bottom was basically what you would do within a plantation. And we said, okay, so you select a number of tree species or trees per species per location. And then depending on whether you're interested in fungi or insects, you kind of sample these trees. And the question that we have now is how many trees do you need in a location for assessing the total diversity associated with such species? And specifically, how many trees do you need to sample if you want to find one individual of a particular insect species? We assessed this in 2 sentinel nurseries that we established in Switzerland as part of the HOMET project. We planted 5 European native tree species, 25 trees per species in blocks at 2 sites, one in Cabby or at Cabby in the garden and one at Wesel. So it's both in the north of Switzerland, about 100 kilometers apart. And since 2019, we conduct a monthly monitoring of mainly herbivorous insects on those trees, and we collect and record information of each individual tree. And since then, we found quite a number of insects of different orders, which are still partially under identification. And what I wanted to do here is to explore, based on some of the more frequent insect species, how many trees you need to look at in order to find them. So I looked at 5 different species. I took the data that we have and basically using repeated random sampling without replication of the data, I just picked how many trees would be positive. The other thing to say, so we found quite a number of species at the moment. It's mainly leaf feeders, and that's mainly because these are, as you can see on the picture, they are just very small trees, and they're still too small to find wood-boring insects and so on. So I have 2 examples to show because all the slides would look the same otherwise. This one is an aphid, as you can see on the picture. It's an Eustia corni. It's a specialist of Cornus. We planted Cornus sanguinea. It's a European species, and it has been introduced to South Africa and North America. And it appears to be a pest because it lowers the aesthetic value of the shrubs, especially in autumn. And we found the species in all years at both sites. And these 2 graphs show data from 2020. On the left is the Cabby site, on the right is the Wesel site. And you can see 2 things here. One is that the species is most abundant in May and in September. And the other thing is that with increasing the number of trees that you sample, you're obviously more likely to find the species. And at both sites, you would be you're very likely to find a species if you sample, say, 10 trees, 10 random trees. And at the Wesel site, you're slightly more likely to find it earlier if you sample in the right season, just because it's more frequent. The second example is the viburnum beetle. As the name says, it's a specialist of viburnums. It's a European species and it's invasive in North America. If you look for the species, you will mainly find hits giving you recommendations about to use pesticides to get rid of it. We found none in 2019, so it took a while for the species to actually find our shrubs, maybe because there's not so many viburnums in natural environment around. But in 2020, we found it, and as you can see, we found very few at Cabby. Actually, we only found it once. And we found quite a few at VSL, mainly in September or in August. And so this species, you need to adapt your sampling intensity depending on the site and on the season. So, which is maybe not very surprising, but it's kind of interesting. But when it's present and it's fairly frequent, you only need about 10 trees. So basically, these data confirm the usefulness of the Sentinel nursery concept, which is largely shown by the finding of native species that are exotic and invasive elsewhere. We still need to compare the identified species with literature records to make an assessment of how complete our findings are. I can already guess that it's not very complete, which indicates that you probably need more sites or a longer period of sampling. And then the other thing is, so I only showed 2 examples. I did 5, which are shown on the top right. So there's 3 aphids, that's the top 3 species, and 2 Coleoptera. The first 4 are specialists, and the last one has nothing to do with the host on which I found it. But what you can see here is that so the bar charts show the number of trees on which it was found, and the colors correspond to the previous graphs. And what you can see here is that aphids are more likely to be found in early summer, say May to June, whereas the Coleoptera are more likely to be found in late summer or in August, and in autumn. And so you need to adapt your sampling to the target organism, which is maybe not very surprising, but I'm not aware of papers showing this kind of information. Then the other thing is that obviously we found large differences among sites, so you need to make sure that you sample sufficient number of sites with different environments surrounding it. But for species, as earlier, for species that are relatively frequent, it seems that 10 trees is a reasonable number per site. So maybe you want to invest rather in having more sites than in having more trees per site. And then the other thing is, of course, that I only looked at slightly frequent species, and less frequent organism groups such as Lepidoptera and sawflies have 2 issues. One of them is obviously that they're not very frequent, so you need to have more trees or more locations to find them. And the other thing is that they seem to be more polyphagous. And that means that even if we found them, we very often found them on one of the species and not on all the species that they may be able to feed on. So again, you need to increase sampling for that. And just to finish, I only looked at insects. I suspect that your sampling design needs to be quite different for fungi. And there it depends a lot on your methodology. If you base your methods on plating, you're sure to find live organisms. You may need a fairly large amount and maybe different media in order to find all the species. If you use molecular tools, I suspect that you overestimate presence of the species, and you are very likely to find false positives. So that's all I wanted to say. Again, this project is funded through the HOMET project, and thank you for your attention.
Thank you very much, René. We have plenty of time for questions. Questions?
I would like to ask you if you have any special information about the distribution of these host plants in the vicinity of the nurseries that you plant.
Yeah, so we planted 5 species: Cornus sanguinea, Corylus vanella, Ilex, something, Juniperus, and Viburnum. And most of them occur in close vicinity. Maybe Ilex is slightly less common, and Juniperus as well. But as you see, the aphid is either colonizing species very rapidly, or maybe it came with the trees when we bought them. I'm not sure.
I have a quick question.
Sure.
This is Yuri. I wonder, have you guys thought about focusing on looking for symptoms or damage rather than for the insect themselves, and only recording damage as a positive data rather than just the presence of the insects?
Yes. The problem is that very often it's difficult to assign damage to a particular organism. So we could probably assign it to a particular organism group, but not to specific species. And as I said, so first of all, the purpose of these sentinel nurseries is rather to identify species rather than damage. So yeah, we have thought about it. We do record it, and especially later in the season, it becomes rather difficult. And at VSL, they had a pretty decent hailstorm this year. So there's really a lot of damage because of the weather rather than due to insects.
Thank you.
This is from Geoffrey Williams, which is the next speaker anyway. What are typical nursery practices like in terms of rotation, fumigation, etc.?
I'm sorry, that was a question for the last speaker.
Oh, sorry.
So still there, left over.
No, okay. Well, we might try to arrange that at a later time. Maybe I have one question, and this concerns the possibility to have older plantations. In fact, there are several plots in Europe which are devoted to the study of forest biodiversity. There are some in Bordeaux, for instance, there are some in Belgium. Isn't there any possibility to use these plots because they're older already and you could probably find wood borers and other species?
Yes, of course. In fact, you could use any tree if you wanted to. Botanic gardens are very good. Arboreta are perfect. You could look at trees in urban settings. It doesn't really matter in a sense, as long as they're not treated with pesticides, I suppose.
Thank you very much. Well, we will go on then, unless someone else has anything. Yeah, Sandy is very eager that you go on. So we have now two remote speakers, and one of them just was on the screen, but it's not the case anymore. This is Geoffrey Williams of the US Forest Service, which is going to talk about an international sentinel network to reduce biosecurity risk uncertainty for North American woody flora. The floor is yours.
Thank you for the introduction. So the title of this project, so you've heard the title. This is still in the project development phase, and we are welcome to suggestions and feedback, as well as any information you may have on any US species that you know of planted in your country. Please reach out to me or Richard Snesko, who is the technical advisor on this project. And that's what we mean by established ex situ resources. It's just a fancy way of saying you have US tree species planted in other countries. So the driving question behind this project is we really want to know what pests, including pathogens and insects, are out there that could impact US species. And
excuse me, Geoffrey. Sorry. Could you pass to presentation mode? We are on the wrong end of your computer, I think.
Oh, okay. Yeah. I'm sorry about that.
If you cannot manage, it doesn't matter, but it would be better still.
Yeah, I can fix that. Is that better?
Yes.
Okay. Sorry about that. I didn't realize I was doing that. So the other question we wanted to know is, are sentinels really going to hold the answer to detecting these pests before they come to the US in a reliable and useful way? And I'll get into what that means in a second. But first, some definitions. What are non-native species? In North America, these are species that arrived after the first Europeans came to the Western Hemisphere. What are invasive pests? These are non-native insects, pathogens, or other biotic agents that are problematic because they cause damage and mortality. And then what are sentinel trees? And Renee Eschen really nicely defined them. But these are really defined in terms of how they're used, rather what they are. So it's more of a question of why. But sentinels can be trees that are either growing in the US or abroad from our perspective for the US biosecurity. They could be native to the US, they could be related species, but for the purposes of this talk, and at least for the beginning of this project, we'll be focusing on US species that are planted abroad, or expatriate trees. And these are really hold a lot of promise for detecting future threats. They can be used to decrease risk uncertainty and to jumpstart response and research and development, including breeding for resistance. And this approach has really been gaining traction. So there's been a lot of progress and success in the pre-invasion realm, both with regard to US species, both domestically with the US Sentinel Network, as well as on an international level with botanical gardens, Conservation International, and the International Sentinel Plant Network. Establishment of new sentinel gardens that we've heard about today. And there's even been potential future threats that have been identified, such as the sweet gum inscriber. We also know Phytophthora ostrocedri has been shown to attack US hosts in the UK. Cronartium pinei is a rust pathogen that attacks, that causes disease in some US pine species in Europe. And even attempts to assess risk for unknown hypothetical pests. We also have other resources available, lists of pests and hosts and distributions through CABI and EPO within North America, as well as just other databases that could be used to look for observations of pathogenic fungi or insects, such as the global biodiversity, the GBIF, and even tools that leverage additional data such as climate and pest distributions to come up with more comprehensive risk assessments. But like anything in science, we're making some key assumptions that could potentially pose challenges to this approach of using sentinel trees. So to go through those, I'll use the disease triangle framework. We're assuming that there are pests in the vicinity of these sentinel trees that the sentinel trees would be exposed to, that are virulent and viable populations. Also, with respect to the hosts, if these are really virulent pests, and we're talking about established plantations, the hosts could have been killed years ago with no record. Also, we're assuming that the host genotypes that are introduced are susceptible to the diseases that could come here and cause problems in the US, and that there's sufficient abundance and attractiveness of these hosts. And this is really important when you start to think about pests and pathogens with wider host ranges that may do better on alternative hosts other than the ones that we're interested in, but if they were introduced, could adapt to US hosts and become problems. Also, we're assuming that the hosts have attained an appropriate age or stress level where you'll see disease. And then from the environmental side of the triangle, there could be local environmental conditions that are conducive to disease in the sentinel range, but that are not conducive to disease in the native range, as well, leading to potential kind of false positives from a sentinel detection standpoint, as well as natural enemies that prevent detection if they maintain pests at a low level and other pests that, potentially could mask the real threats that kind of get in the way of the pests that might come here in terms of detecting pests. And so just to give an example, after starting this project, I actually realized that I did my dissertation on sentinel trees. So I studied a pest of black walnut, which is native to the eastern US. But really this pest of this disease, thousand cankers disease, has been a problem in the Western United States, where eastern black walnut has been planted widely. And along with some other walnut species, black walnut's been decimated in the Western United States. So these are sentinel trees in the West that are getting attacked by a pest complex that's thought to be native to the Western United States. And so as soon as this disease was discovered, the alarm was sounded, quarantines were put in place in all the Eastern states. And we had to walk that back to a significant extent. And even there's been multiple extension publications and outreach, making sure that growers and landowners don't fall victim to predatory timber buyers, or that they don't want to plant walnut because they think this disease is going to be a problem. It turns out, whether it be climate or biotic interactions, or the interactions between biotic interactions and climate, this disease has not become really invasive in the Eastern United States. And this is a problem because our credibility is at stake. First, we sounded the alarm on TCD, then we walked it back. And I actually found that in the future, due to changes in climate, wood moisture content may become favorable for the development of disease in the heart of this host native range. And so we may have to actually sound the alarm again. And so the question is, how sustainable will this approach be in a reactive political and social system? And the bottom line is we really need a more complete picture of these pests and the risks that they really pose. And we needed it a long time ago. And so one way to get at this problem and answer this problem is just more data. So we need to expand our sample size and geographic coverage of sentinel data and facilitate access to that data and comprehensive risk assessment in a way that can be updated and done in real time. So one really potential resource for this is the fact that actually, from a US perspective, our species, native species here, have been introduced abroad in other countries up to 100 or more years, whether that be driven by the Forest Service, the FAO, or the international development, and through private industry, or just ongoing, like within individual countries in their timber industries. And so the primary objective of this project, as it was conceived, is to gather data from, or at least get an idea of what's out there in terms of resources from established operational plantings that could be used as sentinel trees, whether they be provenance trials, progeny trials, or introduction trials, or commercial plantings, both through the literature as well as outreach. And the advantages of this approach is it's going to expand our sample size and geographic coverage for sentinel trees. There's a wide range of countries and climates and environments that enables a more comprehensive assessment of the risks they pose, that pests might pose that are detected on these sentinel trees. In many cases, we're talking about mature trees that are planted at high host density that could be sinks for pests. We could even potentially identify variation in severity of damage. And we may, through gathering the data on what's actually out there in terms of host sentinel resources abroad, actual reports of failures or of pests themselves. And this is also important because now that we have all of these plantations abroad, they could be functioning as bridgeheads to select for host transitions or host jumps, or just build up populations that could then spill over into the United States through international trade. So this is just a schematic of how we've conceived of the whole project so far. On the right hand, I've talked about outreach and literature search to get a sense for what's out there in terms of already established plantations, as well as on the left, the next step, which would be to also integrate this information with the other sources of Sentinel data and data in terms of the presence of pests and pathogens, whether that be through some of these databases, as well as tools that could be used to use all of that data once it's integrated to do rapid assessment in real time through like an online tool or some sort of graphic user interface. And as a proof of concept, we'll be focusing on pines, cedars, oaks, black walnuts, and legumes. So this represents a large number of species. We've selected within those at the subgenus or section level because that's often a high frequency for host range. And this also covers like geographically most of the US, includes hardwoods and conifers, some of the most ecologically and economically important species, especially in human impacted or managed forest systems. It represents a large proportion of potentially of US species, and many of these species have been planted abroad. And from the beginning, we'll be able to expand our list of candidate pests potentially, but also, we really want to move towards a more dynamic platform by integrating, as I said, with some of these other databases and resources, and ultimately construct a user interface. And once we know what's out there in terms of hosts, we can then use that as a base for efforts to do monitoring and look for new pests and pathogens in those plantations, as well as potential variation in terms of the provenances that might be planted if there's records of that. And just to give an example of a tool that could be integrated into this approach. Excuse me?
Just a quick example, please.
Yeah, very quick. This is just a recent publication where this Sepharis was used to predict where the highest risk might be for native pine species in the US for Carnartian pinei. And here are all of the people that have been involved so far or that may be involved in the future. And please, if you know about any US trees in your country, do not hesitate to reach out, or if you want to get involved in the project. Thank you.
Thank you very much. So we have time for one or two quick questions. René?
Hi, this is René Eschin. This is a cool project. With regards to finding records of American trees planted abroad, would you be willing to support people entering data from handwritten records? Because those exist and they go back very long.
Absolutely, and that's, as much as we can expand this project to support that type of work. Once we get the ball rolling, I think that's going to be a really important component of the project. And so we'll really be relying on building trust and collaboration. And thank you for that idea. Yes, if there's any written records and file cabinets, those will be of use and really important in this project.
Thank you. Another question? No? Okay, thank you very much then. And then the last speaker today, and we apologize for the delay, but there was a group picture and this is why we are slightly late. So the last speaker is Caleb Keim from the Ohio State University. And it's again about sentinel planting, a reciprocal sentinel planting approach for assessment of risk from invasive alien tree species. The floor is yours.
Okay, thank you for that introduction. I hope everyone can hear me well. And again, this is a project I've been involved with for my master's at Ohio State. And just before we get started, I just wanted to provide a couple of acknowledgements up front. Dr. Enrico Bonello is the project lead behind this and the main originator of this idea. And I just want to mention all the other project collaborators involved right up front. So I won't spend too much time on this since we just heard a couple good presentations about sentinel planting. So I think the idea is pretty fresh in everyone's mind. But what we did was a little bit different approach. So as the title mentioned, this is a reciprocal approach to sentinel plantings. And what that means is we work with international collaborators, and there are sentinel gardens established in 4 different participating countries. So we have 2 established sites planted with the same species in the US, 2 planted sites in Europe and Sweden and Italy, and an established site in China. And very simply, at each of those sites, in North America, we planted European and Chinese species. In Europe, North American and Chinese species were planted, and in China, North American and European species were planted. For the focus of this, I am just going to be discussing the established plantation in Columbus, Ohio. So the objective of this project was really to understand how potential invaders would interact with target species if they would be introduced into potential sink environments. And ultimately, we know knowledge of such relationships could prove crucial to the countries of origin, and ultimately aid in risk assessment and prediction of threats. So as I mentioned, there were 2 established planting sites in the US. For reference, this is where those would be on the map. We have the site I'll be talking about is here in Columbus, and the other is in Portsmouth, New Hampshire. So a complete randomized block design was used for the experimental setup. And as you can see here, there are 6 by 3 meter plots planted with each of 6 Asian species. And sorry about that. And this shows the 6 planted European species. So after trees have been planted and allowed to establish, visual surveillance of planted trees was conducted on a regular basis. For 2020, this occurred every 2 weeks, and for 2021, this occurred on a monthly basis. During these visits, we assessed insect feeding damage, as well as pathogen symptoms. Such damage was recorded. Any live insects were collected or identified on site. For symptomatic plant material, this was collected, brought back to the lab, and isolated. And the pipeline for identifications from isolated fungi was that of DNA extraction followed by targeted ITS, PCR through the ITS region and Sanger sequencing. So with that, I just wanted to quickly move on through just some of the results we've seen so far. This table lists the observed insect species we saw in 2020 that have been identified. There are a number of aphid samples that were collected, as well as some psyllid samples that have to be outsourced to a specialist. So we're still waiting on those to be identified. But so this is just listing the species, the number of occurrences we saw that species, and then the obviously the potential, or obviously the observed tree host those occurred on. This is a very similar table in 2021. And again, we're seeing some of the same species occurring again, which would be expected. For the fungi, as I mentioned, only an ITS, only the ITS region was used. So we couldn't really definitively get to species level identification alone through this. So really just what I have here is genus level identification. We would need a multi-gene or other approach to further arrive at a confirmed species identification. So yeah, just listing here some of the observed genera we've seen, kind of the known lifestyle of those genera, and kind of if this is, and I have on the right, if these had been reported in the literature as occurring in those species or not. And this is a similar table for the 2021 isolates. No, these are not complete, as we still have a considerable number of isolates. I was sampling up until the 3rd week of September 2021, so those were not, those are just kind of begin growing. So we should have more information later. So I just wanted to highlight a couple of the a couple of the observed insects that were of interest, of potential interest. The species of the question mark, it's a pretty common polyphagous feeder in the family Ulmaceae. But as far as literature searches, at least from what I could find, this had not been yet reported on Zelkova serrata, so I mark that of interest. And likewise, the white-marked tussock has a very wide host range, including species of Tilia, and likely would occur on Tilia cordata, but again, this hadn't been seen in the observed literature, the best as I could tell. In general, for the fungi, a lot of what we're seeing were endophytes or saprophytes in leaf tissue, common molds. But we did a couple of the interactions I thought were of interest, so we're going to try to look a little further into those. We did observe the Botrosphaeria in Tilia cordata, and the genus Botrosphaeria is of interest due to its quiescent lifestyle and kind of its ability to act as more of a latent pathogen. So as far as literature search was conducted, this didn't appear to have occurred on Tilia cordata previously. A similar case was observed with Pistaloceopsis on Colearteria paniculata. This hadn't been previously reported on any the host Colearteria paniculata, as far as I could tell, and these were associated with symptoms of a leaf spot. So yeah, then just in summary, a lot of what we're seeing was known common occurrences, but we won't argue that this approach does still have the potential to detect many novel relationships, and there's a lot more that can be understood in the future. A couple ways, that next steps we're looking to improve this process. As I mentioned, the ITS region alone, we're not able to get species level identification, and we really need a good species level identification to better understand the fungal host interactions. So for this, we actually have an established contact at Ohio State, for which we will be conducting a whole genome sequencing of selected species. And finally, we know the effectiveness of sentinel plantings can be further combined with the use of high throughput sequencing. What I did, this was a targeted culture-based approach. So if you combine this with a high throughput sequencing, you're going to be able to observe more of the endophytic communities as well. So this is an ongoing process with our collaborators, we actually have already collected both asymptomatic and symptomatic tissue this year in June and September. So we're in the process of extracting the DNA and processing those samples for the future. So I don't have.
Can you conclude, please?
Yeah. Okay. So I just want to, yeah. So in summary, I just want to conclude and thank you for your attention. And if you have any, I'd be happy to take any questions at this time.
Thank you very much. It's the last talk of the day, so we have time for questions. Yes.
All right, thank you. Yeah, I see the question from Jeffrey Williams.
Sure. So this is Akif Eskalan from UC Davis. Quick question. Thank you very much for the nice presentation. Botryopsida and Pestalozziopsis are known to be wood canker pathogen on other woody hosts. I wonder if you were able to observe any wood canker on your samples.
Not on those species, but yeah, no, that's one thing that I didn't mention it in there, but for the Botryopsida, that is very much of interest is, that its ability to exist as an endophyte in the leaves, and then, it's possible this could occur later and affect trees if the study were to go on longer, or if a wound or other stress, event occurred on those species. So it's interesting. It'll be interesting to see if anything does occur later. As of now, I haven't observed any symptoms associated with canker on those species. Most of what I saw was foliar. Thank you.
I can see a question online. Can you read it perhaps on your screen?
Yeah, from Jeffrey Williams.
Yeah, I will read it. Oh, can you read it then?
Yeah, I could see it. Thank you. Are these new pests? Are the ITS sequences or insects different from those already known from the US? Most of these are pretty common fungi as endophytes or weak pathogens. Was the sampling targeted at the symptomatic tissue? Yeah, so to answer your question, and I think I tried to make that clear, like most of what we're seeing, at least for the fungi, were known common occurrences. And yeah, so what I would do is I would collect, I'd identify symptomatic tissue. I would isolate from the symptomatic tissue, and those are the cultures I got. In many cases, multiple subcultures would occur, and I would isolate those. So, I am getting a lot of endophytes as well as saprophytes in there too. So, in general, like I said, most of what we're seeing were not, new pests, but it is giving us a better idea of the kind of the general associates of the leaves.
Okay, thank you. Any other question? No, fine. Then this will conclude our afternoon. Thank you very much for all speakers, to all speakers. And I think the last round of applause is absolutely necessary at this point.